Good morning, everybody. We can ask folks if they could take their seats. So welcome once again. Welcome to all those who are new with us this morning. On behalf of the Ontario Federation of Anglers and Hunters and Fitzsimmons Financial Group, I am pleased to welcome you to the conference. For those of you who have been an OFAH conference at OFAH conferences before, we're doing things a little bit differently this year. Instead of covering a variety of fish and wildlife topics, we are focusing on this year's conference on one of the greatest conservation threats we face today, and that's chronic wasting disease, or CWD. During the next few days, we have an incredible lineup of speakers from across North America who will be sharing their knowledge and their experience and advice on how to tackle chronic wasting disease. And we really have to hand it to some of these speakers. That's not in my notes, but with the Max 8s being grounded, some have gone through some pretty incredible efforts to get here, and we really appreciate that. In addition to the formal agenda of informative presentations and thought-provoking discussions, this conference will offer excellent networking opportunities. So I encourage everyone to take advantage of the evening social events to renew old friendships and make new ones. These relationships will be the foundation for building a coalition of like-minded individuals needed to find a path forward and sustain the momentum needed to influence positive change. We have conference attendees from every province between British Columbia and Quebec, as well as Minnesota, Wisconsin, Michigan, New York, and Pennsylvania. We all have diverse backgrounds and interests, and will be able to offer perspectives on agriculture, hunting, First Nations, environmental issues, human health, wildlife management, science and research, animal health, and policy. Regardless of where you fit, we all have a common goal. We want to minimize the negative impacts of chronic wasting disease. Deer, moose, elk, caribou are an important part of our identity. Whether you are a naturalist, medical professional, hunter, veterinarian, photographer, outfitter, or someone who simply wants to sustain healthy wildlife populations, we all need to work together to address this conservation threat. Today and tomorrow, we will have a tremendous opportunity to listen, to learn, and to establish a common understanding of where we've been and where we are, and most importantly, where we need to be. We don't intend for this discussion to end here. We want the momentum created here at this conference to spill over beyond this event, to create awareness and to influence positive change. To raise awareness, we hope everyone will leverage their respective networks to enhance the profile of chronic wasting disease across Canada and North America. While you are here, please share the highlights of the conference. The OFH will be active on social media, and simply sharing our posts can extend the reach and awareness for chronic wasting disease. When you leave, please keep the conversation going. We need to get people talking. We need Canadians to care about chronic wasting disease. Tackling this threat requires a strong and coordinated effort where we all work together to influence timely and effective actions on chronic wasting disease. At the end of this conference, we will produce a summary that will hopefully describe our collective path forward. But first, we have 2 full days of engaging presentations, discussions, and social events that will help get us there. I want to wish everyone a pleasurable and informative conference experience. Thank you for joining us. Your voice matters in the fight against chronic wasting disease. So now it's my pleasure. We are very fortunate to have both Ontario's Minister of Natural Resources and Forestry, the Honourable John Yakubuski, and his Parliamentary Assistant, MPP Toby Barrett, in attendance today. It gives me great pleasure to introduce Minister Yakubuski. He is a progressive Conservative member of the Legislative Assembly of Ontario who was elected in the Eastern Ontario riding of Renfrew, Nipissing, Pembroke in 2003. Before being appointed as Minister to Natural Resources and Forestry this past November, he was the Minister of Transportation. Other roles he has taken during his political career has been critic for labor and training, critic for energy, critic for community safety, chief opposition whip, as well as opposition House leader. As a longtime resident of Barry's Bay in the heart of Madawaska Valley, Minister Yakabuski understands and appreciates the importance of our natural resources, our fishing, hunting, and trapping traditions. I think everyone up there must play hockey too. When I was with the Ministry of Natural Resources, we had these big tournaments, and it was always Algonquin Park and Whitney that was the team to beat. So I want to welcome and have everyone welcome Minister Yakabuski.
Thank you very much, Kerry, for that kind introduction. Is this stuff I'm supposed to throw out to the fans here? Is that... Anyway, it was great to be here this morning. And Kerry, you mentioned the number of people from across Canada and also in our neighbor, the United States, from different states. So it's so fantastic to have that kind of attendance here and those kind of people coming from so far away to discuss those challenges that you'll hear about at this conference. And if I may, I'm just going to slip these off here if I could. What a beautiful morning. I was in Myrtle Beach last week, so I'm just trying to be nice, you know. Anyway, it is great to be here, and we had a 15-hour drive back yesterday. Left at 1 in the morning. Got back to Mississauga to my sister's, our rendezvous place, my son, 2 of my brothers, and we had gone down, and it took us 15 hours, all stops included, so it was a very smooth ride back. I'd like to acknowledge that you did already, Kerry, but my parliamentary assistant, there's Toby Barrett, who's been doing a fantastic job, and he's been a member since... Toby's got 8 years on me. He was elected in 1995, so he's seen the file from the government side of the House on 2 occasions and doing a fantastic job. And also, Toby just finished up doing a bait fish study and analysis from going all around the province, and he's about to tabulate all the results from that and the recommendations. So thank you, Toby, for the work that you've been doing, and also his tireless support for angling and hunting across the province. As someone who enjoys hunting and angling myself, it's great to be among a group of people who also enjoy those pursuits. Those who are committed to a future with healthy lakes and forests and opportunities for all Ontarians to hunt and fish. These activities are important to Ontario's economy, with hunting contributing more than $430 million and recreational fishing bringing in over $2.2 billion. Going hunting and fishing is a great way for families to connect, to enjoy the great outdoors, and is certainly something that my family takes advantage of. So let me just tell you a little bit about my family. My dad, who was also the MPP for Renfrew South, was also the parliamentary assistant to Natural Resources for a number of years. I was talking to this gentleman here who was at some event with Leo Bernier and my dad. That'd be many years ago. Leo Bernier, who of course was the minister and known as the King of the North. But Dad was very proud to be the parliamentary assistant in that role for a number of years. But my dad, before he got elected into politics, used to fish every Wednesday afternoon and every Sunday afternoon. Wednesday afternoon in Barry's Bay, where I come from, back then, stores were closed. The stores closed on Wednesday afternoons. My dad was in the retail business, and stores closed on Wednesday afternoons, and he went fishing. And I remember catching my first fish at Stopas Creek. and my dad had a bamboo rod. It was a good long one, probably about 13 feet, because he said, you've got to be able to get over in that little hole over there, because they're in there, trust me. Caught my first speckled trout at Stopus Creek. When I caught that, you'd think that I just landed one of these, he used to call them the lunkers, the big lake trout or something. You think I'd landed a trophy, because it was. For me, it was a trophy. And that's how kids feel. And so when I was up at Chemong Lake earlier this year, and those kids are pulling out those little crappies, they were just like, they're so excited because for them it might have been their first fish. You know, when I was in 1967, the high school opened in Barry's Bay. First time we had a district high school. So we had to bring in a lot of teachers from outside of the area. And many of those teachers came from urban parts of the province. Some of them came from Toronto. And they had to be educated themselves. Because what they didn't realize when they got there, don't schedule anything of any importance during the first 2 weeks in November. It's deer season, folks. The place will be deserted. And it essentially was, because everybody was gone hunting for those first 2 weeks in November. So the basketball, the volleyball, it was volleyball in the fall, volleyball, inter-school stuff, and this was all through Renfrew County, and Gord's out there somewhere, I saw him there. Gord would understand this. Every high school in the county was the same thing. You don't do anything in November. It's deer season. Forget about it. And our kids they're a lot more involved than me because they have more time. After my dad got elected into Parliament, he didn't have time to go fishing on Wednesday afternoons. So he lost some of that, but he never lost the love of that pursuit. But our kids are very avid outdoorsmen. This year, 3 of our children were all successful. I remember I was getting on a plane back from Newfoundland. We had a fisheries conference there. And I was sitting beside this lady, and she was wearing camel pants. And I thought, oh, this is good. And I brought out a picture that my wife had just texted me of our daughter kneeling over a 12-pointer that she had just downed with a crossbow. And I said, oh, you got to see this picture that our daughter just got this morning with a deer. I showed her pictures. Ah! I'm a vegan. Things got a little quiet for a while. But I looked at those camel pants and they said, she's one of us. Apparently you can buy those things anywhere. Or you know the old saying, don't judge a book by its cover. Don't buy a girl by the pants she wears. Don't judge a girl by the pants she wears. Our daughter-in-law, I got to tell you this one for all you gentlemen out there. Zachary's wife, Stephanie, this is a few years ago. She was the shooter. 2 deer, 2 bear, 2 moose, 1 hunting season. Don't mess with Stephanie. So we have a great history, and we're proud of the people that love these pursuits. And as far as the fishing goes, they're out fishing as much as they can go. They're running a little tourism operation now themselves as well, so that keeps them busy as well. For over 7 decades, the Ontario Federation of Anglers and Hunters has been a very effective voice for those who love to fish and hunt, like my family. Like you, our government for the people is working hard to promote these popular sports. They are part of our culture, part of our heritage. To make it easier to enjoy our plentiful natural resources, to encourage safe and responsible hunting and fishing, and to champion the sustainability of our province's fish and wildlife. We are working for the people because we believe that every decision made by government should put people first. Last June, the people voted. You voted for a government that would respect your money, that would clean up the financial mess, that would bring good-paying jobs back to Ontario. A government that would grow the economy, make life easier and make life more affordable. These priorities of ours apply to every ministry, including the Ministry of Natural Resources and Forestry. Before I talk about what we're doing about chronic wasting disease, I'd like to mention some of the things that our government has done and is doing to make hunting and fishing easier and more accessible. Last fall, my colleague and predecessor, the Honorable Jeff Urich, announced the introduction of a new fish and wildlife licensing service that makes it easier for anglers and hunters to buy licenses and for private license issuers to serve their clients. Now, there's only one version of the outdoors card instead of different types of cards for anglers and hunters. The new service is available over the internet, in person at approximately 100 license issuers and 60 Service Ontario locations, or by calling a 1-800 number. This has made life easier for anglers and hunters. And the improvements haven't stopped there. You are now able to carry a single license summary or keep it on your mobile device, instead of needing to carry individual hunting and fishing licenses. One of the first announcements I made as minister was the steps our government has taken to thank our armed forces members and veterans. They can now fish for free all across the province all year round. This is a small token of our gratitude for their dedicated service to our country, and a gesture that will hopefully inspire our servicemen and women to get out and fish and to take their families with them. We are encouraging all families to do the same. This year, our government has doubled the number of license-free fishing days. All Canadian residents can now enjoy free fishing on the Mother's Day weekend and the Father's Day weekends in 2019. These are in addition to the free license fishing events on Family Day and the license-free event, a week-long event at the end of June. It was great to see so many families come out to the OFAH Family Ice Fishing Day last month. I understand it was a record turnout this year. More than 1,100 people ice fishing on Chemong Lake. Hundreds of families bonding over an Ontario tradition that our government, your government, will continue to support. We also recognize that for our future generations to be able to continue enjoying our natural resources, we must promote sustainable fish and wildlife management. It is important to note that all hunting and fishing revenue goes into the Fish and Wildlife Special Purposes account, which is used for important management programs. And I do want to point out that managing those programs is part of our ministry, and we have a number of ministry staff here as well, and I thank them for their contributions and service as well. Some of the concerns I've personally heard from hunters recently have to do with moose populations, hunting opportunities, and the moose tag draw. As I announced last fall, my ministry is conducting a comprehensive review of our moose management program. An advisory committee will spend the next 2 years looking at how tag quotas are developed, how hunters apply for tags, and the current allocation process. Many of you will be familiar with the Ontario Moose Bear Allocation Advisory Committee. We have restructured this committee and given it a new name, one that I believe better reflects their new mandate. It is now known as the Big Game Management Advisory Committee, or as I like to call it, Big Mac. The initial task of the committee will be to participate in the moose management review and develop recommendations on moose management with a focus on how quotas are developed and how tags are allocated to hunters through the draw. As the review unfolds, we will continue to listen and engage hunters and stakeholders to inform future decisions on tag quotas. Once our initial review of the moose management has been completed, the committee will provide their advice on a variety of big game management issues. We have identified well-qualified appointees, many of whom have worked with or for OFAH over the years, and I look forward to receiving their recommendations. Our goal is to make moose hunting fairer, more accessible, and simpler for hunters. But we also want to ensure a sustainable and healthy moose population and continued hunting opportunities for generations to come. In order to also ensure a healthy deer population, our government recognizes the importance of addressing chronic wasting disease. We have taken steps and will continue to do so to protect wildlife populations in Ontario. As I'm sure you all know, CWD is a progressive fatal brain disease that can strike deer, elk, moose, and caribou. To date, it has been found in 26 U.S. states and 3 provinces, most recently in Quebec. While CWD has never been detected in wildlife in Ontario, my ministry has been diligent in its surveillance efforts for many years, testing more than 12,000 wild deer and elk since 2002. In 2018, we focused our testing in wildlife management units in southwest Ontario. We also conducted increased surveillance in Wildlife Management Unit 65 in southeast Ontario because of the report of CWD on a farm in Quebec near the Ontario border. The good news is that CWD has not been detected in any sample in Ontario over the last 17 years. But we know we cannot become complacent. As we begin 2019, we continue to evaluate the CWD risks to determine which wildlife management units will be the focus of our surveillance efforts this fall. And although we conduct testing in the fall, we continue to seek samples from sick and strange acting cervids anywhere in the province at all times of year. We're also updating our response plan and associated rules under the Fish and Wildlife Conservation Act to make sure we have the right mechanisms in place to minimize the risks of CWD entering or spreading in Ontario. I'm very encouraged that you have several discussions planned on CWD over the course of your conference this weekend. Because in the coming months, my ministry will be asking the OFAH, hunters, and the public for input on this important issue. We continue to welcome your feedback so that we can continue to manage this effectively. Hunters and the OFAH are important allies in our efforts to prevent and, if necessary, respond to this disease. I want to say to you, the OFAH members, I share your passion for hunting and fishing, and I am committed to working with you to build on our province's rich traditions. To the OFAH executive, thank you for your ongoing support and for so capably representing our province's anglers and hunters. I will work hard to maintain the strong relationships we enjoy today. I want you to know, the Ontario Federation of Anglers and Hunters, your membership, and all hunters and anglers out there, that you have a friend in the Minister of Natural Resources and Forestry, John Yakubuski. Thank you, and enjoy the rest of your conference.
I want to thank John for taking time out of his busy schedule and leaving warm Myrtle Beach to come up here with damp, cool weather. But we really appreciate the fact that you have taken that time.
My pleasure. Thank you. Thank you. Here, Carrie, I'll put those back for you.
Oh, you don't need to. They're for...
No, my mother always told me, if you take something, put it back where you got it.
He listened. Okay, so I'm going to ask Keith Monroe, OFH wildlife biologist, to come up, and he's going to start introducing some of our guest speakers.
Thank you, everyone, for coming. We have a host of fantastic speakers today, and I'm sure we're going to have a lot of questions. We have time scheduled at the end of every talk for questions, and the way we're going to do it is we will have a couple of microphones circulating, so if you're interested in asking a question, just hold up your hand and an OFAH staff person will bring that to you. Chronic wasting disease is a complicated issue and frequently involves aspects sort of outside of our normal hunting, fishing experience. So the goal of this morning's session is to provide everyone with the foundational knowledge on chronic wasting disease so that we can fully embrace and understand the rest of the conference. So to do that, I'm happy to introduce our very first speaker. Dr. Evelyn Merrill is a professor in the Department of Biological Sciences at the University of Alberta. She received her master's from the University of Idaho and her PhD from the University of Washington. She has spent 40 years researching cervid foraging and habitat ecology in a diversity of ecosystems across North America. She and her students' current research includes an 18-year study on the dynamics of a partially migratory elk population in the Rocky Mountains of Alberta and the spatial spread and population transmission of CWD in the prairie provinces of Canada. She is a fellow of the Wildlife Society, served as the president of the Canadian section and the Alberta chapter of the Wildlife Society, is currently the Canadian section representative to the Wildlife Society. She was the editor-in-chief of the Journal of Wildlife Management from 2013 to 2016, and in 2018, Evie was awarded the Wildlife Researcher Award by the Rocky Mountain Elk Federation. And also, when I asked her to send in a bio, in addition to all that, she somehow has time to spoil a set of grandchildren as well. So, Dr. Evelyn Merrill. There's your clicker for your slides.
Okay, great. Okay, it sounds like you can hear me. Thanks very much for the invitation to talk today about a very difficult issue. I am pleased to be among a number of speakers who I really respect. We're going to hear about prevention from Dave Westaway, and we're going to hear some inspiring words, I'm sure, from Daryl, who's been thinking about this issue for a long time. And I'm really want to hear from New York, who has done something that all of us have not been able to do, and that is tackle the CWD problem. But my role today is really just to start the conversation. I am not an expert on all areas. I am actually a field biologist, so I'm going to be talking to you today about some things that really you will hear later on the experts talk to you about. I do want to acknowledge my co-author here, Michael Samuel. Mike is at the University of Wisconsin in Madison, has been dealing with chronic wasting disease in Wisconsin for many, many years, and he has provided me some data for some comparisons that I'm going to show you today. Okay, so I'm going to cover a lot of ground to get a foundation here for these basics, which is what I've been asked to do. I'm going to touch on prions and prion diseases, a bit of history of CWD, something about the diagnostic tests that you might be interested in, transmission and factors influencing, some surveillance and spread, demographic consequences, and management challenges. And we'll explore each of these topics in more detail as we go. But please remember that it's really, my knowledge goes from less about prions to more about wildlife. So prions are basically a proteinaceous infectious particles. And the basic idea is that they are a cellular protein and are normally produced in all mammals in a normal form. And the PRPC cellular form actually is composed of about 250 amino acids. It's coiled in a alpha helix. It is soluble when it's in this form. It's unstable. It's easily digested by proteinase and short half-life. It does not accumulate in the cells. Those are all the good qualities that we want. But the alternative fate of the prions is that it can be converted into a disease-causing form. And this is called the PRP-res, which is the resistant form. It unfolds and then refolds into alternate B-sheet forms. It's resistant to proteinase enzymes, and it does accumulate and causes the plaques that you see in different pictures that I'll show you today. It's associated with neurological and especially in the brain damage. Now, those two forms then are what we're going to be talking about even when we're talking about whole animals. The idea then is that conversion occurs as a chain-like reaction where the abnormal PRP, CWD, enters the organism likely by ingestion or some other means. This actually seeds then and binds to an existing PRPC, the normal form, and causes it to misfold. So it's, again, not a bacterium, not a viral. The cells then are low, and they make more cells as they start to contact and act like an infectious disease. And this cycle continues through the accumulation of PRP-CWD in the body. Now, there are some intermediate forms, as I understand it, that are maybe part of the reason why we have such long incubation times in CWD. But this is the basics when we start talking about animals, even on the landscape. Now, prion diseases, of course, are known as transmissible spongiform encephalopathies, or TSEs. And they are a group of progressive neurological diseases that many of you know about. From the animal side of life, there's scabies in sheep and goats, and really was the first prion disease to be detected and eventually named. As early as in the 1700s. We have BSE that we've all known about, and Canada has brought this to their attention when our borders were closed to shipping out cattle to the US and other countries. And now we are smack dab in the middle of the chronic wasting disease, what I would call an issue that is looming before us. But there are, of course, and we'll talk about these later in terms of what the human threats are today, human prion diseases as well, such as CJD, and you are probably familiar with the different variants of that. And then, of course, the BSE, the humans can get, and mad cow disease, and even Kuru, which means literally that name comes from the word to shiver in the Fore language in Papua New Guinea. There are some other forms that also affect other wildlife species that you might hear about, transmissible mink encephalopathy and a feline spongiform encephalopathy. So there are different forms that we'll be talking about today, but really our focus will be on CWD. Now, there are alternative views rather than terms of the prion theory for CWD, and this has been seen lately in the news and on media here about CWD being caused by a cell wall deficient bacterium, Spiroplasma, and this has been studied at Louisiana State University by Dr. Bastian. And this is one theory, but it's not one that's really at the forefront of the consensus today. And I guess today I'm going to be talking about CWD from the prion perspective. And this is also the Association of Fish and Wildlife Agencies have really come out and made a statement that this is the way that they are approaching this disease. So I will, based on that, stick with this approach today. Now, most of you already know about chronic wasting disease, but just to bring you up to speed again, if you see an animal in the field and it has strange behavior, if you see that it's really lost a lot of weight or wasting, as the name comes from, if it's stumbling around, or ataxia as we call it, so it has a lack of coordination, it's listless, it has its ears drooping like in the picture of that elk, it's excessive thirsty or urinating, drooling, all kinds of weird behavior. And it can also then be less afraid of people. And we don't know whether in the clinical stages actually may be avoiding hunters even on the landscape. And if you see that behavior, then it's likely that the animal will die, and if it does, then we should test it for CWD and look for these plaques in the brain and the brain stem. Now, CWD, what are we up against here? It obviously is this TSC that I've been talking about in moose, elk, deer, and in caribou or reindeer. It, though, has some characteristics that makes it a particularly difficult disease to work with. First off, unlike some other prion diseases, it's highly contagious. Now, when I say that, it means that the prions can be infectious very quickly in the body, but it doesn't mean that it plods along on the landscape. We don't have outbreaks like we do with botulism and other diseases that we work with in wildlife. Unfortunately, there's no full resistance to it. All animals and cervid species are actually subject to being able to get CWD. And once they get it, it's 100% fatal, which makes it a very difficult disease. The time to death, it has a long incubation time. It's about 1.5 to 2 years. In deer and moose, and maybe up to 4 years, a little bit slower in elk. Now, one of the things that makes it a difficult problem also is that it resists inactivation. And things like ionizing radiation or temperature or even formalin, which we put some of our samples in, and proteinase and nuclease treatments, all of those it's resistant to. The point being that it persists in the environment, and that makes it very difficult to deal with. Now, prior to 2000, it was really considered a Western disease within North America. And as you can see here on the map that I've shown you, the gray area in the middle of North America here, that's Wyoming and Colorado. And as you probably know, it was first detected in a captive research facility in Colorado, the foothills there. It was found in mule deer, and that was as early as 1967, although it really wasn't known what it was at that point. And in captive elk in Wyoming in 1979. And then later on, we started to detect it in free-ranging animals, deer, elk, and moose in the 1980s in Colorado and Wyoming. But today, as mentioned, it has, and it's a moving target because in the US already, it's 26 states that we found CWD in. In Canada, it was actually found in the Toronto Zoo in 1974 in a mule deer. So that was the earliest case in Canada, in fact. In Saskatchewan, it was found in farm captive situations in 1996, but it wasn't found in the wild until 2000. In Alberta, in a captive situation in 2002, but then it wasn't found until 2005 in the wild. And Quebec now, as mentioned earlier, it has been detected, and that's close on your doorstep. Korea actually in 2000 received elk from Canada, and it was traced back through time that they actually are another area overseas where CWD has been found. Now, more recently, what's been in news is that CWD is no longer just a North American issue. It is now more global in the sense that CWD has now been found in Europe. In March of 2016, reindeer in Norway, in that red spot on the graph that you can see in Norfella mountain area, it was found in reindeer. They went in and did a very aggressive cull in that area for the reindeer. And since that time, they have found it in 2 moose and actually 3 moose as of now, and 1 red deer, which is the equivalent to our elk. And so they have been surveying very aggressively for CWD in Norway. But most recently, though, it's been found in Finland in a moose where that little Google dot is, right near the Russian border. So CWD is no longer just a North American issue. It is now something that has been in Korea, it is now in Europe, and we are now collaborating with many people who are afraid that it will spread even farther. In terms of the detection and diagnoses, well, we know that it accumulates, the prion, CWD prions accumulate in the systemic and in the neurological areas of the deer. In particular, the lymph nodes, as you've probably heard about. And part of the lymph nodes, we can detect it earlier in those areas than in the actual neurological areas. In fact, it is until it invades the brain and some of the neuro invasion occurs that we can detect it in the obex and other portions. So in fact, when we go to the field and we test some of the animals, the ones that we radio collar and then try to understand their movements on the landscape, we use two major approaches for live sampling animals. The first one is a rectal biopsy, and that's the one on your left. And this is a easy technique to use in the sense that you only have to locally anesthetize the animal and also sedate it, so it's a less invasive approach. If you want to get a better sample, though, in a live animal, you have to then do the tonsil biopsies, which is the one on your right. And you actually have to put the animal under. It's much more invasive in terms of getting those samples. But it is a much better approach to actually detect CWD. Now, when we do surveillance programs, and surveillance can be either passive sampling, meaning if you find a wasted animal in a farmyard, we send it in for testing. There are many surveillance programs across North America looking for CWD, trying to detect it before it gets too far along in a certain jurisdiction. Or we also have, in some jurisdictions, we have herd reduction programs. And from those, we then get from hunters or from reduction programs by the government, we get heads or lymph nodes. And we can then look at, as we do in Alberta there, we look at the retropharyngeal lymph nodes, and we try to detect whether CWD is present in those samples, in deer and moose, and in particular in the obex and in elk. So there are a number, and I'm not the expert on these different techniques of different approaches, and we're trying to improve those in terms of detecting it in biological samples. The immune histological chemistry remains probably the gold standard still for looking for CWD, but we are evolving more rapid, high throughput ELISA tests that are now being developed. And other techniques with the goal eventually that it would be nice to have a real field technique that we could give people in the field to actually test their own animals. But we're not there at that point. That is one area of active research across North America, and it would be great if we could get there. Now, to understand the disease and what to do about it, one of the most important and in my view, one of the least well understood in terms of all the complexities that go along with it are the routes of transmission. Because without understanding those dominant routes, and it may change between the timing of the disease and the location and the stage of the disease, we're not going to be able to address how to manage it. Now, if you think about how this is happening, we really have different components of being direct contacts among infected animals and indirect contacts. And direct contacts, of course, can go through body fluids such as saliva, whether a male is urinating on another male, so it sheds the disease or the prions on different individuals. That is the way it can happen. They can also be put into the environment where, as I mentioned, it is very persistent. It can accumulate in areas where there's high use, either in winter ranges, as we suspect, or around areas such as grain bins or other high concentrated areas where animals are being fed. And so the environmental passage is another important aspect. But how the animals actually take it up and what we can do about dispersing animals on the landscape and keeping them from contacting the disease is indeed a real challenge for us. So what are the dominant routes? Well, we're not really clear at all times, and it changes probably with disease progression. Vertical transmission from offspring to adult and across the placenta may occur. It does occur, but it probably plays a minor role. That means that we have young that are being produced, healthy young that are being produced and brought into the population. But the social dynamics is probably a large part of what goes on between groups of different individuals as well as within the groups. And we have supported this with some of the genetics work that we do. We know, for example, for mule deer that they use a social dynamic where different animals, different offspring from a particular dam will will establish a home range around its mother, and therefore if we look nearby we can find related individuals. And in fact, as this graph shows you on the left, that in space, we do know something about the genetic relatedness, and we find spatial structure out there. Those animals then, ideally, we can assume that they are contacting each other, and probably the disease would go up in that spatial structure. And in fact, some of Kathy Cullingham's work has shown that as you relate pair-wise different animals on the landscape, once you account for sex and age, that yes, if you are related to an individual, you are more likely to have the disease that not your offspring, but someone related to you nearby will have the disease too. Now that's important because it does give us a key about how we might find infected animals on the landscape. The environmental understanding of and the evidence for really comes from the penned work that we have done. And it is true that if you don't have individuals where direct contacts are going on, we still have carcasses, and we move bedding from infected animals to non-infected animals, and it's only through those routes that new animals can get infected. So we know that it can be an animal can get infected via the environmental pathway. And unfortunately, there are mounting evidence and many studies now that start to point to the reservoirs on the landscape in terms of where these sources are. We have soils, and we all know that deer and elk and other cervids will eat, whether it's intentionally, such as mineral licks, or as part of their diet out on the landscape. They take in soil, and we do know that prions bind to soils, different types of soils in particular, and it can be very stable and actually increase in the infectivity. So soils are one reservoir. And in addition, studies have now accumulated evidence that if you simulate heating and wetting and drying, perhaps some are degraded. Microbial and lichens now, there's some evidence to suggest that they can reduce some prions and alter them that may make them less available, so that's a possibility. And humic acids can increase CWD and has reduced some concentration as the humic acids increase. So they may, however, then reduce the infectivity. So we have a lot of small bits of information on how this all works. But the soils are not the only reservoir. We found PRP res in water, and so that can also be on the landscape a source, unfortunately. Of great interest, of course, because the cervids that we work with and are talking about are herbivores. They eat plants, and so CWD and other prions can be absorbed by plants, and they have been found in the roots, stems, leaves, so that this is another infection pathway. Lichens extracted may or may not inactivate prions, and in fact Norway started to put restrictions on haying in selling the hay in certain areas where CWD is known, and they started this in just this last year. So plants are a source out there. Now, vectors in terms of moving the prions around, again, it is complex. We do have evidence from experimentally inoculated animals that have had infectious prions in their feces. In particular, there's evidence out there for coyotes, which we know are just move widely and are broadly distributed across North America. Corvids are another species that could move these prions around. So it's not just carcasses on the landscape, it's who's feeding on those carcasses and where they're taking some of the materials that are coming from dead infected animals. Now, these studies in total and accumulating what we know about it really give us a proof of principle that these pathways are out there. But we still really do not have a good handle in terms of the reality of what's going on in the landscape. Realistic modeling of this is not an easy task. There are good models out there, and one of the insights that come from these modeling exercises is, again, how at first, when you first detect CWD, the major pathway may be animal to animal. But over time, as the environment and these reservoirs build up, the whole dynamic of the disease progression may change because of the increase in the environment. And that's worrisome, and again, another reason, as Daryl has mentioned for many years now, that we really need to tackle this problem the best we can. So over time, environment may be our biggest worry on the landscape. Now, prevalence, because of this transmission that's going on, no matter what pathway, even though we don't know exactly how it's working, it's going on. And as you can see, as we accumulate in states and provinces where the disease has been detected now for many years, we can start to see the patterns on the landscape. And just to take two contrasts then, in Wisconsin, for example, if you look over time, and those red spots are the hot spots, meaning the highest prevalence levels, there really have been 2 outbreaks in the Wisconsin-Illinois areas. And they're beginning to merge. One of the things that they have noticed, though, in this area, that the rivers do tend to be a barrier, a semi-permeable barrier. And so you are getting patterns on the landscape that are due probably to the behavior of the animals, not just the reservoirs as well. In Alberta, that pattern is very different from Wisconsin, where it is kind of moving out from a centroid. In Alberta and Saskatchewan, we find it moving along river courses, and this may be because we find it in mule deer, and the mule deer winter habitat is along these corridors, as you can see in the little picture I have down there. That's where we find our mule deer in the winter in high densities. So landscape patterns are another component of this whole story, and something that I know my lab and my students who are interested in animal behavior are trying to focus into, because if we can understand the influence of landscapes on the transmission and spread of disease, then maybe we can focus our efforts. Now, there are some other differences that stand out, and might or might not, you may or may not be aware of them depending on what species you have in your area. But it's very clear in our area where we have whitetail and mule deer that overlap in Alberta, that in fact 86% of all CWD positive cases since 2005 in wild ranging animals, which was our first case, have been in mule deer. And we're not clear why that is. We can't rule out anything at this point. This is a pattern that has been consistent year after year. We're now up to 1,000 cases, and that has not changed in the 15 years that we have detected CWD. Now, if that's a behavior change, if it's a landscape grouping change, if it's a behavior, or it's something about the animal and its acceptability, we're not clear at this point, but it's a very strong pattern. It's clear in other jurisdictions, but it's not absolute, as I'll talk about in a bit. The other factor that we know is that genetics makes a difference. And while all cervids and individuals are susceptible to CWD, there are different genotypes that are more or less resistant and susceptible. However, that has different implications. For example, in some of these genotypes, they may get it, but they may not die as quickly. So they are in the landscape for longer periods of time and may be spreading the disease more widely. The idea that there is potential evolution of resistance and it may occur, but it's likely to take decades where our management now really needs to be directed more toward what we can do to slow down the spread and the increase in prevalence on the landscape, and not wait for it to take its own course, at least in my view. What genetics may allow us to do is understand the spatial structure and how our genotypes exist on the landscape. In Alberta, this is something that we're looking at. We're finding that there's more structure in the whitetail than actually the mule deer at this point. And so once it gets into our whitetail populations, we may be able to tell you in which direction it's more likely to spread. But in the end, what we don't know about the changes in genetics of animals that survive chronic wasting disease is how their resistance to CWD will then be linked to other fitness components such as survival and reproduction. And that makes a very complicated story as a result. Sex is also something that's very consistent across jurisdiction. Here are two graphs where you look at time on the x-axis there, and then the two bars for each of these groups. Again, the left-hand side is Wisconsin, and those are whitetails. The right-hand side is Alberta and mule deer, but the patterns are very clear between both jurisdictions here. Males have it about 2 to 4 times more than females. And again, we're not clear why that is exactly. It could be behavior. It could be something about the actual dominant routes of transmission. Males tend to be curious. They fight with each other. They spread their body fluids. In some of our modeling, one of the things we found that if we take sexual segregation, meaning when the males go off and interact among themselves versus staying with the females, that we actually lose this sex difference. So we think it has something to do with the fact that males and females actually go off and interact separately. So CWD males, at least in the whitetails, based on Mike Samuel's work, males have a higher mortality. CWD positive males have a higher mortality than CWD non-infected males, and CWD males have a higher mortality than CWD positive females. And that's not too surprising because in general, males have a higher mortality rate than females. Now, the reason I bring some of these different characteristics up is because over time, more and more provinces are going to establish surveillance programs. You already have established them, but if you get detections, you are likely to ramp up some of your surveillance. And in Alberta, for example, we have submissions of about 45 to 5,000 heads a year from the hunters. This is part of our voluntary and mandatory surveillance program. And if we can figure out the patterns of how it is on the landscape for looking at it at least at certain longer time periods, we can do something now that the mathematicians have devised called weighted surveillance, where we can then use those characteristics of sex and age and species if we want to detect it and know if it's there on the landscape and where it's moving into new areas. We can cut back on our number of samples and our testing. And so you can use these different ratios of how often it's found within certain sex and age groups to actually cut back and still get the statistical rigor for detecting it on the landscape. And this is something I think we'll all be moving toward more in the future. So what are the consequences then for bucks, for example? The average longevity for a buck without CWD, and this is based on whitetail, is about 1 more year when it's detected on average based on the studies that have been conducted. The average longevity for a buck with CWD, let's see, did I say that right, is about a year when it's not infected, and when it is infected, it's about 4 more months. And that means that we're going to have fewer and younger bucks on the landscape. How about for does? Well, the average longevity based on Mike's work is that a doe without CWD will live about 2 more years on the landscape on average based on their age structure. Average longevity for a doe with CW is about 6 more months. So we reduce the doe population, we're going to have population effects. As we see what we're learning about how it is affecting the population, we can start moving into making predictions about what's happening on the landscape. Now that's been very important for me because when I first got started in the whole CWD realm about in 2005 to 10, I would go to people and say, you know, we need to address this issue, we need to study these particular problems, and people will say, well, nobody has shown us that it's having a negative effect. But now we have that information. We know that CWD is having population-level effects, and the studies are now accumulating across jurisdictions. So for example, mule deer in Colorado, the population growth is less than 1, meaning that it's declining, and this is an unhunted population. Prevalence was about 25%. And in fact, by researching it in detail, they determined that there was higher mortality in CWD-infected deer. And in Rocky Mountain National Park with elk in Colorado, again, the population growth was about 1. The prevalence in elk tends to be lower than in other species. The population growth was a little bit less than 1, and the mortality again was found to be higher in elk. White-tailed deer in Wyoming is another very good study where they collared animals and then followed them what we call longitudinally and see when they die once they get infected. The population growth was a 0.9, which means it's declining. In this case, the female portion of the population was actually higher in prevalence than males, which is a bit different pattern than I talked about earlier. And they found that CWD-infected deer were almost 4 to 5 times higher mortality. So we are seeing that the disease is having an effect. And one more study, and it is the accumulation of these studies what makes it really convincing, is that in Wyoming, again, and this is where the population was declining and males were, again, the highest prevalence in mule deer. The females were a little bit lower prevalence, but still quite high prevalence. CWD-infected deer were dying at about 3 times higher mortality. So with that type of information, then we can make predictions about what the population is going to do over time in terms of both the total population and the age structure. So when we look cumulatively over all the information that many people in this room and many people have spent their careers working on, I have to kind of sit back and say, this is not just a wicked problem. This is a super wicked problem. It's a slow disease progression, so when you first get it, you can't sample enough to understand what's going on. There are complexities of transmission, and it may be that it changes the dominant route of transmission, changes over time, so we're chasing it. Environmental persistence is a big one. And how genotypes over time will change and add to the complexity of what we do at the beginning, at the end, we don't know. And CWD strains, this is an area that is of active research, and I'm not the expert on, but as CWD prions are passed through different individuals, the strain and how the infectivity of that strain changes and affects other animals is something that may change again the dynamics of it. No vaccine at this point. We get excited every once in a while, and I have my fingers crossed all the time, even though it would be a very expensive and difficult way to apply in the free-ranging animals. It's still something I think we need to pursue. And in terms of management efforts, they require, I think, something that we're all going to have to face, and that if we're going to have to do anything, if we're going to do something, whether it's in the House of Representatives where there's a bill on chronic wasting disease in the U.S. today, whether it's in Alberta where we're trying to get long-term political support, or as you start to work toward this goal in Ontario, the long-term political and financial commitments is really what's going to help us make inroads in this very difficult problem or super wicked problem. So thank you, and if I can, I'll either answer questions or redirect them toward the experts in the audience. So thank you. Thanks. I'm not sure where to put this.
Sean, right in the...
John Williamson's. Question I have, you didn't get into, but the association with game farm deer. You didn't touch into that at all, and it seems to me that in the past that's where a lot of it has come from or originated, and the same with Quebec, and I wondered if you could speak to that.
I think we all recognize that in tracebacks, especially in the initiation of sources in the wild, that farmed captive and farmed animals are a source of CWD. However, I I think we've moved past that, maybe not out east here, but in Alberta, it is in the wild, it's endemic now, and we need to deal with it because people have even said that it might be going from the wild to the farmed animals now because it is so endemic in portions of Alberta. So it is a very important question. I think Daryl has written on this quite a bit. The Korean cases, Saskatchewan, that's where we find it at first. Regulations need to be put in place, and the voluntary versus mandatory herd certification, those are all very direct questions that we need to deal with. But my view, and this is probably my Alberta bias, is that it's not just a farmed and captive deer problem now, it's a wildlife problem as well.
Good morning, Tony Jackson. Good morning, Tony Jackson. Thanks for your presentation. Quick question, and maybe I missed it in the presentation. Can you confirm whether or not fetuses that are born to infected does are born with the disease, or is that something they get after birth, after suckling and natural?
Great question. My understanding that it is possible. There's a study, I think by, is it Nellis? But it's relatively, I think right now we are considering it a relatively minor route. Now that could change over time, but after the offspring is born, if it's born to an infected mother, it's most likely because of all the close interactions and body fluids involved, it's going to probably contract the disease. So I think there is evidence that it actually does go across the placenta, but it's a pretty strong barrier overall. And maybe there's new evidence to say one way or the other, but that's my understanding.
Hi, Jerry from British Columbia. It's interesting to hear that Norway is doing something about curtailing the movement of hay, yet apparently there's no appetite to do that in Canada. And I know outfitters from Alberta bring hay to northeastern British Columbia in the early summer, so it's a concern to us. Maybe you can comment on that, please.
Well, first off, let me compliment you on your signs that say don't bring, your big yellow signs with blue letters that are right along the border that say don't bring in carcasses from Alberta. I know Alberta actually got, the tourism department got a little upset with those signs because it was, they thought it would affect tourism in Alberta, but the hay issue and the whole plant issue is probably just at the beginning. I think we have a lot to get on top of it, on top of whether hay from CWD infected areas, that would be in the eastern part of Alberta. In particular, there are going to be regulations. I think that'd be a great question for some of the ag people. It's not happening, you're absolutely right. I don't see it happening in the immediate future, but Norway has taken a very aggressive approach right from the get-go on this in terms of going in and culling their reindeer herds. They are taking this very seriously, and I think it's because they realize, just like from the experience in New York, that you really have one major crack at it at the beginning, and if you can't set it back on its heels right away, you're in it for the long haul, like some of the rest of us are. I don't know if that's answered your question, but I really don't think I have an answer to it. It's a definite issue.
Hi, I'm Jim McCain from Bruce County. You mentioned, I believe the figure was 86% of the cases since 2005 were in mule deer. Do you have any idea of the percentage of mule deer in Alberta versus whitetails?
Ooh, that's a great question. No, I'd have to look that up. In my study area, which is on the eastern edge, right next to Saskatchewan, we have done our aerial surveys. And in that area, I have actually calculated the density. And there are two ways to look at density. The first one is per habitat versus just for the area itself. And when you look at the, calculate it just based on the area itself, mule deer are lower. But mule deer tend to stay along those coulees, and they don't go out into some of the broad woodland areas as much as the whitetail do. So if you put them on a density level with their habitat, they come out at the same density. And the reason I was interested in that was because of, again, this close association between animals and the spreading of the deer, of the CWD and the deer. So I don't know the big picture question of that. I'm sure I could find it out for you. But even if you had those numbers, be careful with them in terms of spread of disease.
Thanks very much. I think we have time for one more question.
Please.
You had mentioned, this is on, you had mentioned that plants actually have the prion in the leaves and the stems and the roots. And I wondered if, could you just refresh us on how that mechanism works, and that make sure it's not just in the grass mites, but it's actually right in the plant. Is that correct? And how does that happen?
Okay, my understanding that there is a study with, is it barley or wheat? I've forgotten which one. Where they actually have shown that the prions are actually in the roots, in the stems, and in the leaves. How that happens, I'm not exactly sure. Whether it's in a water medium and it's taken up through the, directly from the soil, or through a water medium that it brings into the plant, I guess I'm not sure that I have the expertise to tell you on that. Great question, though. Okay.
Well, thank you very much. Dr. Merrill, we have a... I'm trying to remember. Thank you very much. So I think that was fantastic to kick us off for the conference. We're going to have a short 15-minute coffee break, and when we come back, we'll be hearing from Dr. Michael Coulthart from the Public Health Agency of Canada. Thank you. So as I went about organizing this conference, one of the really exciting things was how engaged all the scientists and policy and government and advocacy folks were on this subject. And this next speaker is a really good example of this. Dr. Mike Coulthart and I had a phone call just to chat about CWD, and as that call was going on, Mike said, I think you really need to have a speaker on human health at this conference, and I'd be happy to be that speaker. And I said, Mike, I was actually going to ask you in this phone call to be the speaker on human health at our conference. So definitely on the same page, and delighted he could make it. So Dr. Coulthart is the head of the Prion Diseases Unit and the Canadian Creutzfeldt-Jakob Disease Surveillance System at the Public Health Agency of Canada. His work has been focused on scientific and public health aspects of emerging and infectious diseases for more than 25 years. After completing his graduate and postdoctoral work in population genetics and evolution, in 1995 he joined Health Canada as a research scientist in microbial population genetics. Following the discovery of variant Creutzfeldt-Jakob disease in the mid-1990s, he was redeployed to develop a federal diagnostic and research laboratory to support national surveillance of human prion diseases in Canada. Today, he is one of Canada's senior scientific experts on surveillance, epidemiology, and public health for human prion disease. Dr. Mike Coulthart.
Thank you, Keith, and thanks to the organizers and the OFAH for the kind invitation to speak with you today. As Dr. Monroe, as Keith mentioned, I'm a federal scientist with the Public Health Agency of Canada, and initially I was hired to work on microbial population genetics. As everyone knows, those are biological species with genes. And when it was suggested to me that I worked on something that didn't have any genes, I thought, well, maybe not. And it was suggested also that I not get too comfortable in the area. This was about 20 years ago, because it was going to be a passing thing. This was before variant CJD really started to become a concern for Canada. And then that took a long time, actually, to be resolved, or at least come to the point where we feel we have control over it. And now we're concerned about a new kind of issue that may be a risk to human health, but we're really not sure about it. So if you look at the title of my talk, I ask a question here that I won't answer definitively because it cannot be answered definitively at this time. But I'm going to suggest to you that there is a structured way of thinking about human risk, even though the evidence is really not very strong yet. And the answer in simple terms to that question is a qualified guess. So Dr. Merrill did a very able job in introducing prions and prion diseases, so that will save me a little bit of time. I will briefly touch on some of the same points that she made, but then I'll move quickly into a discussion of the scientific evidence that we currently have for CWD in relation to human health risk. And there are two kinds of information here. One is data from humans directly that has come from targeted epidemiology studies as well as national surveillance in the US and Canada. And the second kind of evidence comes from animal models, where attempts are made to experimentally transmit chronic wasting disease to a laboratory animal. And most recently, there have been some findings in very active discussion from the Alberta Prion Research Institute, from Dr. Stephanie Schub's work, and also from the National Institutes of Health in the United States, in a group led by Dr. Rick Race. So I will summarize those lines of evidence, but I won't discuss other less directly connected lines of evidence from experimental work that involve mouse models, so-called test tube models or biochemical systems that can shed light on transmissibility of CWD to humans, or computer models, which have also been informative but not as fully developed yet. I'm going to end with a perspective that is largely mine, but I think it's based on the evidence on how we might think about human risk, and then suggest some public health and personal precautions going forward. Right. So, as I said, Dr. Merrill did a very good job of introducing the basics, but I'll just remind people that this is a new kind of infectious agent. In 1997, Dr. Stanley Prusiner won the Nobel Prize for his discovery and development of the concept of proteins as infectious agents. And the key is that it's a change in molecular shape of a protein that is normally made by most mammalian cells. And that simple fact leads to a host of different consequences that make prion diseases among the most difficult challenges for public health and animal health that have yet been encountered. One thing is that they appear to be capable of originating spontaneously. So sporadic human prion disease, there are cattle and sheep forms that are thought to be spontaneous in origin. So it indicates that really there's no reasonable hope of ever truly eradicating these diseases because they will probably originate repeatedly in otherwise healthy hosts. There's no protective immunity that we know of that develops as a result of an animal or a human being infected. So the phenomenon of herd immunity is not particularly relevant to prion disease. They're environmentally persistent. There's no known level of safe exposure to prions that's been identified. Careful analysis of a range of animal model studies of prion infection have shown that there's no identifiable lower limit to the amount of exposure, physical exposure to infectious inocula that would not lead to infection. So it's a difficult thing to quantify in terms of how you can handle levels of exposure. In contrast, for example, with chemical exposures in the environment, where it's generally considered that harmful chemicals are tolerable below a certain level of concentration. They have a long latency, so they can spread for a long time undetected. Dr. Merrill alluded to the presence of strains, and this can lead to significant changes in pathogenic and virulence behavior. They can adapt, and in our particular context here, talking about interspecies transmission, this leads to the possibility that a prion agent that is otherwise not very virulent for another host species can, with enough exposure, overcome the resistance of that other species to the infection. And once the prion has been able to initiate the propagation of the prion protein in the new host species, it is already largely adapted because it's the new host species prion protein that supports the ongoing propagation. So rapid adaptation is something that we also need to be concerned about. There's no treatment. Diseases are always fatal, whether they're humans or animals, and there's no vaccine. So prevention is costly and difficult and mostly based on surveillance and rather radical control measures like depopulation. And in humans, there's no particular method for prevention, aside from control in the healthcare environment, which is where a lot of human prion transmission has classically taken place, dealing with issues like contamination of medical instruments. But again, it's a difficult process because you have to identify the person who may be at risk of having infectivity in their tissues and then act accordingly very quickly. I won't go over the range of animal prion diseases again, just to point out that BSE and scrapie, in a sense, represent two ends of a spectrum of human risk. On one end, you have scrapie, with which we've been living for centuries, and there has been no definitive evidence shown that, in fact, in reality, people do get infected with scrapie. It can't be ruled out completely, but it has been studied to the point where we think we understand the disease as a low risk to human health. BSE, on the other hand, as people well know, is a known human pathogen, and it has caused an epidemic of human prion disease in the form of variant CJD in the UK, Europe, and 10 other countries, including Canada. So that's the other end of the spectrum, and we don't really know where CWD falls on that spectrum. It's a fairly safe bet to say that it falls between those two ends. It may fall outside. It may be absolutely no risk, or it may be worse than BSE. We don't know. But it's a fairly safe starting point to think that a working question is CWD more like scrapie or BSE? Human prion diseases, although they weren't called that at the time, were co-discovered by two German pathologists, Creutzfeldt and Jakob. There's a little bit of a backstory there because it's fairly sure that Jakob was a true discoverer, or at least had priority, and Creutzfeldt came along later and may not have even had a case of CJD in his lab, but the name has stuck, and it's now called Creutzfeldt-Jakob disease by most people. On the right, there's an image of the kind of microscopic destruction of brain tissue that takes place. Spongiform change is a characteristic of most, if not all, animal and human prion diseases, and it's really quite striking when you see it under the microscope because it looks like the brain is being eaten away. It's a little bit of an illusion because it's a vacuolation process, but it does come along with loss of neurons, which leads eventually to death. There are two major forms of CJD. One of them that people don't think about as much because it's sort of there all the time is what we call classic CJD, and it occurs at a rate of 1 or 2 cases per million people per year, just about everywhere where it's been thoroughly surveyed. In Canada, that is now translating into 60 to 80 cases per year, or more than 1 a week. So it's a fairly frequent phenomenon. And this is what we spend most of our time tracking in the Public Health Agency of Canada, because that's most of the CJD phenomena that we encounter. We look intensively into each one of those cases as far as possible to determine their causes and their characteristics, including the possibility that it might not be classic CJD, but something zoonotic. There are sporadically occurring, and most people use that as a synonym for spontaneous, genetically caused, and also acquired forms, meaning transmitted by infection. And those have all been through, with classic CJD, the healthcare environment, through contamination of medical instruments, or in most cases, as a matter of fact, through grafting of tissue from people who died with CJD, and it was unrecognized. The disease may incubate silently for at least 30 years, the classic form of CJD, and we know that because we can pinpoint the date of grafting of tissue grafts. The main form of tissue graft that's caused classic CJD by infection is called a dura mater graft. It's the outer lining of the nervous system that is used as an excellent repair material in neurosurgery. And it turned out that at least one batch of this commercial product was contaminated with human prions at the time it was produced. And that outbreak is still ongoing. We, in fact, identified a case in 2017 of a person who had received a dura mater graft in 1987. So the incubation period can be very long. And as I mentioned earlier, there's no known link of this form of disease to animal prion disease. Variant CJD is a very different matter. Although the numbers are much smaller, it's created a lot of concern because it is linked in a solid way to BSE. 12 countries have been affected, with deaths between 1995 and the most recent one in 2016. And Canada's had 2 cases, one in 2002 and one in 2011. Both were investigated and concluded to be imported. In other words, the individuals were exposed in other countries to the BSE agent. It can be transmitted by blood transfusion. This has been demonstrated in the UK with several very, very likely cases of transfusion transmission of variant CJD from donors who had variant CJD incubating but had not yet become clinical. They later developed variant CJD, and then a few of the people who received donations from those donors developed variant CJD. Another complication, which I think is very, very important and is still sort of awaiting full evaluation, is the fact that there seem to be thousands of silent carriers of variant CJD in the United Kingdom. They've been able in the United Kingdom, because of the availability of large tissue archives, to study the prevalence of pathology in appendices and tonsils that is indicative that those individuals have been infected with BSE. And as I say, the best estimate is that there are thousands, if not tens of thousands of people in the United Kingdom who have this subclinical infection. What's going to happen to them, what the possible consequences are for onward transmission, we really don't know yet. So, how do we take all of this information as background in light of what we know about CWD and find a way to address the issues of risk to human health? And of course, this wonderful map by Dr. Brian Richards deserves being shown again and again. I've taken the liberty of modifying very slightly his map by adding some blue highlights just to emphasize the rate at which the reporting of CWD distribution has increased since 2013 in this case. So the blue outline in Canada is, for Canada, the extent of reported CWD cases in wild populations in just about exactly 6 years ago, in March 2013. And you can see the comparison between that and the maximum extent of the gray area now. And similar comparisons can be made, of course, in the United States. So it's fairly clear that CWD is likely going to continue to expand in surveyed populations in North America, and as a result, the opportunity for human exposure to CWD, given that it's fairly safe to assume that the majority of those animals that are harvested are not tested for CWD, exposure opportunities for humans are going to continue to increase for a while. So it's something, obviously, that public health needs to keep a close eye on. So here, in a nutshell, are the key findings of humans' prion disease surveillance in both Canada and the United States over approximately the past 20 years. That's the period over which concerns about healthcare transmission and blood transmission and possible zoonotic transmission have been predominant in the United States and Canada, to the extent that both countries have funded intensive surveillance of human prion disease of all kinds. And in Canada, this has led, I'm getting these buttons mixed up, sorry. In Canada, this has led to the identification of a total of 972 cases of classic CJD of all kinds. There have been 2 cases of variant CJD found, as I mentioned, and no other forms of CJD have been identified. And what I mean by that is that we carry out neuropathology whenever we can on CJD cases that come to our attention. So between 70 and 80% of this figure of 972 of those cases have actually had autopsy and neuropathology examination. And this is a very good way to distinguish different sources and variants of human prion disease. Variant CJD, for example, has very distinctive neuropathology that is, to an alert neuropathologist, almost unmissable. So we haven't seen anything like that, and each of the cases that we've found fits, as far as the information we have indicates, with a known form of CJD. And the same picture with larger numbers essentially applies to the US. I'll get this right eventually, with obviously much larger numbers. The US has also had 4 cases of imported variant CJD and nearly 4,000 cases of classic CJD reported to the end of 2018. So the significance of this for interpretation of human risk from CWD is that surveillance covers the period of highest concern for human exposure in North America to CWD. In other words, the past 20 years or so. We both take an intensive approach, both countries, and we think that the negative findings here are therefore meaningful. It is more like evidence of absence than just absence of evidence. So the way we summarize this is by saying there is no direct evidence of human CWD to date that either the US or Canada is aware of. It doesn't say that we're not missing something, but the evidence is fairly strong. There have been 5 studies of targeted epidemiology of CJD in situations where people are thought or have been confirmed to have been exposed to CWD, and all of them had negative findings. It's important to note, though, that these studies are very difficult to conduct. There were only 72 CJD cases included in all of these 5 studies combined, because it's not easy to do long-term follow-up, and assessment of CWD exposure is, of course, very challenging. So it's reassuring to see that those small studies had negative findings as well, but of necessity, they weren't as powerful as comprehensive surveillance. So moving on to the primate transmission studies, these are the 2 species that have been used in attempted transmissions of CWD to non-human primates. On the left, we have the cynomolgus macaque, which is known as an old world monkey, and I'll show you a bit more about what that means in a second. On the right, we have a squirrel monkey, which is a so-called New World monkey, and that reflects their geographic distributions. Macaques are mostly found in Asia and Africa, and squirrel monkeys are found in Central and South America. This is an evolutionary relationship diagram that shows the relationship between, on the one hand, very distantly related primates, i.e. lemurs, New World monkeys, Old World monkeys, our closest cousins, the anthropoid apes, chimpanzees, gorillas, and so forth, and humans. Now, a considerable amount of attention is paid to the fact that Old World monkeys are more closely related on an evolutionary scale to humans than New World monkeys are. But it's also a bit of a mistake to say that Old World monkeys, i.e. macaques, are actually close relatives of humans, and therefore you might assume that what you find in macaques reflects what you might expect in humans, because the evolutionary divergence estimates based on extensive supporting information for the Old World monkeys, which are the closer of the 2, begin at 26 million years. So this branch point is a long time ago. So it's not really on the same scale even as what we think of as the degree of closeness of apes to humans. The divergence points here range from 6 to 18 million years. And New World monkeys, of course, are more distant related to humans, and the numbers here are even more impressive in terms of distance. So you can't automatically extrapolate from any of these experiments to what you might expect in humans. I hope people can read at least the key features here. There have been 3 studies reported in the literature to date, although Dr. Schubs is still ongoing. She's reported at conferences, which is a form of peer review, of course. These 2 other studies have been extensively reported. The first one by Richard Marsh and colleagues was published in 2005, and both of 2 squirrel monkeys that they infected by injecting brain material, CWD brain material, into the monkey's cranium became infected. In the case of the NIH study, essentially the same results were found with brain material both inoculated into the skull and fed orally. So there's a very high attack rate here. Almost all of the squirrel monkeys were infected. In contrast, the NIH study found no evidence, even with detailed, careful pathology studies, that the macaques had been infected. And they were also injected inside the skull and fed orally. Dr. Schub's results, which were from macaques, were in strong contrast to the NIH findings. So among other types of experimental transmission, they fed the animals both brain and muscle tissue. And in each of those groups, there was infection found, evidence for infection. So this is the most detailed slide. I hope you can read it at least approximately, but I'll make sure that I articulate what's on it. So squirrel monkeys are highly susceptible by the oral route. They are non-human primates, and this was reported in 2009. It's interesting to me and a little bit puzzling why this has not been thought about and discussed more publicly, because it suggests very strongly that there is evidence of primates being susceptible to CWD. In contrast, though, we have a much fuzzier picture with macaques, especially given that the NIH findings were negative and the APRI findings were positive. There were several differences in design of these 2 studies, so it's not surprising that they might end up with different results. I hear quite a bit of thinking and discussion where people are trying to decide which one is right and which one is wrong. I don't think that that's the right question. I think that both studies are valid, and the differences in design and also biological variability, which cannot be fully controlled, both probably provide the explanation for the different results. NIH quite justifiably interpreted their negative findings back in 2014 to indicate that a barrier protects humans from CWD infection. But the APRI findings suggest that this barrier is not absolute. So it's not like humans are immune to CWD. At least this evidence does not suggest they're immune. And this translates, based on the APRI findings with muscle, that CWD positive muscle should not really be considered completely safe for humans to consume. I know that that's a strong statement to make, but this is what the scientific evidence points towards, with the caveat, of course, that monkeys cannot be treated like humans in terms of the implications. I've already explained my little bit of a criticism of this statement that macaques are closer to humans and therefore the results are more relevant. I don't think that that is supportable. I think some non-human primates are definitely susceptible to CWD, and I think the caution is that you must be careful in extrapolating that to human risk. So to sum all this up in a perspective that is largely mine, but I think it's based on evidence, there's no direct evidence of human CWD so far, but human exposure is likely widespread, and recent scientific evidence suggests that there's no scientific basis for human immunity to CWD. And we should consider additional factors, such as the possibility that some people may be more susceptible, CWD prions may adapt to humans, and there may in fact be silently affected human CWD carriers. So all of this points to me to the conclusion that the risk of human CWD infection, albeit probably low, cannot be completely dismissed. And because human CWD could have very serious consequences, even if there were only a few cases, it adds to the rationale for thinking very carefully in terms of precautionary principles, whatever you take that to mean. And of course, the precautionary principle itself is the subject of a lot of discussion, with some people dismissing it outright and others thinking it's very valuable. I happen to think the latter. So personal and public health precautions, what would they look like? First of all, I would suggest that people be aware that CWD could be transmissible to humans. I will reiterate that it's probably useful to carefully consider testing your harvested animal, especially in CWD endemic areas. I'm certainly not the first to say that, of course, or to say that CWD positive animals should probably not be consumed. Something that I haven't heard in general advice is that it may not be advisable to consume cervid brain, especially in CWD endemic areas, but because that's where the majority of the infectivity may lie, it's probably not a great idea no matter where the animal is harvested. We should continue the surveillance and research for both CWD and CJD, and in whatever way we can, contribute to reducing the spread of CWD. Pardon me. And that's it. Questions or comments? Sorry, I'm waiting. Okay.
Thanks, big guy. Felix Barbetti from Niagara. Is there a time interval from exposure uptake to a positive indication within the primates or, well, humans? I heard an interval of up to 10 years in some report.
If you're thinking of CWD, we don't have that information for humans. We have variant CJD as something of an analog to that, and the median incubation period for variant CJD has been estimated to be around 11 or 12 years in the UK. In the monkeys, it was only a few years, in the case like 2 to 3 years, I believe, possibly up to 4 with squirrel monkeys, and with the macaques in the APRI study, it wasn't too much longer than that. If I remember correctly, it was 5 to 6 years. But those were direct, experimentally controlled exposures, intended to show infection if it was possible. So I think in terms of the incubation period question in general, given that classic CJD at least has been shown to be capable of incubating silently for decades, I don't think we can conclude that because we haven't seen human CWD yet, that it may not be incubating.
Thank you.
Hi, I'm Kat Lantang from Blood Watch. I'm over here, other side.
Sorry,
Hi.
these spotlights are
I'm just wondering, with your experience with CJD and VCJD, is there any consideration at the Health Canada level and an ongoing discussion about donor deferrals who are donating to the blood supply, especially in CWD endemic areas?
That's a very good and a very difficult question. Donor deferral with variant CJD was based on geographic place of residence during the period of maximum concern for whatever region you were looking at. So for the UK, for example, the period of highest risk for people who had come to Canada after residing in the UK and looking possibly to donate blood was between 1980 and 1996, when very definitive control measures were put in place to restrict the transmission of BSE and to prevent human exposure. So after 1996, it was considered negligible in terms of probability that a person living in the UK would then be afterwards exposed to BSE. you can see the difference between the UK situation and the way that Canada handled it and what we would ever be possibly able to do in Canada if it turned out that human CWD was a reality, and therefore on a precautionary basis, which was the basis for action with variant CJD before it had been demonstrated to be blood transmissible, what would you do to actually reduce the risk of blood transmission? I don't know personally what you would do. The idea of having a questionnaire, for example, that said, have you consumed cervid meat, I don't think would be defensible. It would be very difficult ethically, actually, to support that kind of move. So it's something that's being discussed right now, but the question is not an easy one to tackle.
Thank you. I'm John Ford from Owen Sound, Ontario. I have a question about the monkey studies that have been done with brain tissue and oral feeding of infected meat. Have those studies been peer-reviewed? It sounded like the one study only had 2 animals as a sample. You said they're valid. I just wondered if they had been peer-reviewed.
Peer review typically refers, without further qualification, to something having been published in a journal, in print, with anonymous peer review, the whole traditional approach, and people have the chance to pick apart the author's supposed findings in great detail. Another way of doing peer review is to present it at international conferences in front of large numbers of experts, where there's an opportunity for interchange of ideas, and open criticism. I mean, anyone who's attended a scientific conference where something controversial is presented knows that scientists are not shy to try to shoot something out of the air if they don't think it's valid. I've seen Dr. Shub present this at Prion 2017, and the experts did not raise any substantial objections. They asked questions of clarification. Her evidence is very good. I know from speaking with her of some of her unpublished findings, and yet the study is ongoing, by the way. That's one reason why it hasn't been published yet. So I am convinced myself that her findings demonstrate that transmission occurred. So your question is a very good one, but it's important to know that this is not just sort of a floater. Her findings are very solid.
Good morning. Very good talk. My name is Rob Hare. I'm from Keswick, Ontario. You had briefly touched on testing. Now, in a room that has a lot of hunters in it, and with the prevalence of this going to come to Ontario, it's just a matter of time. Is there work being done on testing methods? So primarily right now, I understand if you harvest an animal, the Ministry of Natural Resources and Forests would like to head if you're in an area where they're testing. But if it becomes more prevalent, are we looking at a rapid testing system so we know that if I harvest an animal, that it can be tested rather rapidly before it's consumed?
I think it's wise for me to pass that question to people who know far more about it than I do. It's obviously a bit of a tricky question because the economics of testing and the logistics of testing are not trivial. So I honestly don't think I'd be in a position to speak directly to your question, but there are plenty of people here who can talk directly.
Good morning, Tony Jackson. Thanks for the presentation again. One of my thoughts as you were speaking is that brain tanning is a very common practice for dealing with hides. Do you have any concern about the transmission through absorption or risk and monitoring of people who handle hides on a regular basis?
Yeah, that's a very important question as well. I mean, obviously it doesn't have to be food consumption of brain tissue that would lead to exposure to CWD if the animal did have the disease. I don't know enough about the practices around that to really make a suggestion as to what the potential for human exposure would be. It suggests to me that perhaps if there's an alternative, it might be interesting to seek that out. I mean, would other species' brain tissue do the same thing as cervid brain tissue would? I know that there are traditions among indigenous peoples to use the brain. It's just enough to do the animal's hide. And that's a separate kind of consideration, of course. But if there were an alternative, it might be worth looking into.
I guess my concern is that in remote parts of the country, you use the resources that are available.
Right.
So that's the natural practice, I guess.
Right, right. I think that's an example, actually, of something where there is really no good answer at this moment. I think in areas where the animal is harvested and no CWD has been reported, then the risk could be considered extra low. It doesn't mean it's impossible, but one shouldn't be overly worried, I would think, in an area that hasn't reported CWD so far. So I think that coupling decision-making on the part of individuals with knowledge of where CWD is could add some value for precautionary purposes.
Any more questions? No. And Mike, thank you very much. That was excellent. Thank you very much. I know, I always do too. I think many people here will recognize our next speaker. Daryl Rowledge is no stranger to the OFAH and has presented at this conference before. Additionally, he's been active on the CWD issue for many years. Daryl is an Alberta-based researcher, writer, policy analyst, and public interest advocate. He has spent over 25 years investigating, distilling, and speaking on a variety of public policy issues, from forests and wildlife to healthcare, sustainable development, and constitutional law. Currently the director of the Alliance for Public Wildlife, Daryl is leading an international effort to confront the crisis of CWD. Daryl has served as a special advisor to the Alberta Fish and Game Association on the Alberta Forest Conservation Strategy. He was a founding member of the National Caucus of the Constitution and the Environment and has served as the Wildlife Society's ad hoc committee on examining issues of domestication, disease, and the threats to wildlife. Daryl Rowledge.
Good morning. So I first want to acknowledge the territorial lands of the Mississauga and Anishinaabe people. I have the good fortune today of not having to expand on that because my colleague Brian Jackson from the Piikani Nation will be able to offer greater thoughts on that side. And it also has the benefit of me not having to try to stumble through the Blackfoot that he teaches me. Because I'm always suspicious that, I'm secretly saying something about the Queen's underwear, but so I have, now I have to go back. So I've been tracking this for a long, long time, and the essence of what I've been doing is to try to figure out how we get in these positions. And I've entitled this No Accident, Public Policy and Chronic Wasting Disease, because that's what this is. And before I dive into that, I need to clarify that I'm not a pathologist. I'm not a molecular biophysicist. I'm not an economist. I am not a lawyer. I'm basically an idiot because I can't stop looking at everything. And to illustrate that, I want to talk about my favorite idiot. This is actually a story from Herman Daly, an economist. He tells a story about this guy that was incredibly famous because he was known far and wide as the village idiot. And I mean, this guy was crazy. You could go up to him and you could have two different denomination bills. You could have a low denomination bill and a high one, a 1 and a 10. And you could offer the village idiot one or the other. and he would reach out and grab the little one and put it in his pocket, and he'd go away. And people started increasing the denomination of the bigger bill and doing it over and over and over again. He'd always take the smaller denomination. And one day a guy got really ticked off with this, so he got him cornered and he said, look, I know you're not stupid. You're going to tell me, why don't you take the bigger bill? And the village idiot said, well, if I start taking the bigger bill, people are going to quit making the offer. And the reason I think this is so valuable is that it has wisdom about sustainable economies. It has a lot to tell us about the very essence of government. And so when people think about government, very often they think about politics, they think about democracy, but at the core, what we're trying to do in terms of political science is try to carefully study the various issues that we have to deal with and try to figure out how we can make great decisions, intelligent decisions based on science and evidence to get what we want and avoid what we don't. The village idiot, he knew he wanted the money, and he wanted to avoid people quit making the offer. That's literally how simple this is, but the incredible complexity of the sciences that we have today make this really, really challenging. But the responsibility to do it is all based in the public trust doctrine. This is the essence of why we have representative government. So when we're dealing with this issue, we have to understand how this has played out over time. So this has been mentioned before. So beginning in about the mid-1980s, all of a sudden there was this crazy disease where cattle started attacking the farmers. They were acting erratically. They were tipping over dead. And this was the essence of the mad cow epidemic, or so-called bovine spongiform encephalopathy. Millions of animals were slaughtered. The agricultural economies were absolutely devastated, and the costs were enormous. Now, the official claims that this was not possible to transfer to people, there was no evidence that it had gone to people, they turned out to be tragically wrong. About 230 people turned out to be infected, and tens of thousands still carry the abnormal protein. We don't know what that will mean. But the bottom line in this is that the toll was eventually limited because changes in policy and protocol ended up containing the epidemic. So when it was found out that industries that had collected all this protein and rendered it down into ruminant feed, they were going around to the slaughterhouses, they would pick up whatever they could get, and they would put downer cows, anything that they could find that they could make protein out of, they were rendering it down into ruminant feed and feeding it to cows. So understand what's happening here is that you're taking an herbivore, making it a carnivore, and then making it a cannibal, and you're feeding it diseased relatives. And it turned out that the similarities to Kuru of the people, the 4A people in Papua New Guinea, were assisted the scientists in figuring out that this was in fact a TSE. And so they changed the policy with regard to the ruminant feed and contained the epidemic. Now, they looked at this really carefully because they'd made some mistakes. So in 2000, they issued the BSE inquiry report. Lord Phillips had 16 volumes. Yeah, I did read all this. And then there was another document that was produced by Lord Phillips looking at the lessons that they learned. And they knew that they'd made some mistakes because they weren't abiding the precautionary principle. The industry was allowed to go on for too long. It spread the disease wider, and it exposed millions more people. So the harm was greater. They said the precautionary principle should have been applied. And nevertheless, they stemmed the epidemic, and the world took note of that. Now, in my province in Alberta, in 2003, we had one mad cow. One. Instantaneously, the United States led 39 other countries to ban all of Canada's beef and beef products. It extended actually beyond that, and there were massive multi-billion-dollar consequences from that. But again, the policy changes to prevent the spread to food and feed chains was able to contain it, so we learned our lessons, or not. Over the same time period of BSE, this massive epidemic of a sister disease called chronic wasting disease was spreading. And in 1996, basically the same day that the UK government announced that people were dying from BSE, the Canadian government confirmed that CWD had been confirmed on a game farm in Saskatchewan. It was imported with game farm animals from the United States. CWD was declared a state of emergency in 2001 by the US Secretary of Agriculture. In 2004, Canada's expert scientific panel made a plea that we need to do everything possible to prevent the spread before it's too late. They also confirmed the origin of this, that we had imported it with game farm animals from the United States. So more recently, last year, the One Health Congress, which is kind of like the leading bleeding edge of health and healthcare in the world, they, at their conference, addressed CWD. We got it on the agenda, and you'll note that they hoped to look at the lessons learned from the BSE crisis. I was there, and unfortunately, a lot of that was looking at the reactions that you can do rather than prevention, and that's one of the problems that I want to address. So, for example, this is how we could have done a lot better. When CWD was identified down by Regina in 1996, we pleaded with the government to trace where this came from, to try to find out where it may have gone. They refused. They said, this is one-off, don't worry about this. We got this. We killed all the animals in the herd. So we pleaded to no avail, and it was found again in 1998 over by Swift Current. And again, they refused to figure out where it came from or where it may have gone. Then it happened on that same farm by Swift Current in 2000, and eventually the government said, this is Ag Canada then, they said, we need to figure out where this came from. And so they did. And they found out that there was a herd up by Lloydminster that was the original, it was the index herd for Canada. And in the 4 years that they delayed, they allowed that farm to ship animals to 40 different herds, and they infected 21. They spread this all over the place. This was despite all the pleas that we made to government to try to deal with this. You've seen some of these already. This is what it looked like in 2000. Then what we're facing in 2019. This is the largest, most contagious, most infectious biomass of infectious proteins in global history. The implications are dire, as you're going to hear more from Brian Richards and other people. This is a huge threat to our wildlife populations. It's a threat to deer all over the world. It is the issue of the plant spread is profound. So when I started hearing about the plant studies, I actually went into Chris Johnson's lab in Wisconsin and looked at what he was doing. It was incredibly elegant science where he tagged the proteins with fluorescence, and then using laser microscopy, he could track the endocytosis through the roots up into the aerial tissues of the plants, and it was in the infectious range, and the animals got sick. Claudio Soto did a different approach in Texas. He took wheat grasses and he exposed them to CWD on the outside that might simulate if an animal drools with saliva or feces or urine on the outside of a plant, and he basically power washed them to try to get it off. He proved with PMCA that it was still there, and every single lab animal got sick. So when I was hearing that stuff, I was enormously concerned about the implications for trade, because the bottom line is that when we look at international trade agreements, we realize that they're not based on science and evidence, they're largely political, and here was a scientific justification to put other countries ahead of us. So there's a comparison here on the trade side. I put the UK in geographic context here to show the comparison to North America. Either Wyoming or Colorado is bigger in mass and geographic area than the UK is. So when I say that this is the largest biomass of infectious prions, that's not exaggeration. It's hugely in that category, and it's highly contagious. So how did we get here? Well, we were pleading with governments to have them understand that you're not looking at the big picture. And this has sometimes been referred to, Garrett Hardin described this as the tragedy of the commons. And the way he described it, he used a common pasture as a formula where he could show that if people were able to add another cow to the common pasture, the pasture would inevitably be overgrazed because all the benefits would come to the person who had added the cow, but the costs would be shared among the many. And he used that economic formula to try to suggest that this was what was behind it. He's actually mistaken about some of this. The real cause, I would suggest, because some of the greatest tragedies have occurred on private land where the costs and benefits are shared by the same people. So what's really going on, I call it micro-market myopia. This is where we have a micro picture where we're typically looking only at a narrow view. We often base our decisions in the marketplace in monetary terms, sometimes in votes and other currency, and we have a short-term time range. I want this benefit right now. And is there a solution to this? Of course, we can do comprehensive analysis. We know that we can't just do narrow analyses. We have to do broad-based analyses. We know that biology and business are not just interrelated. One is a complete subsector of the other. There are no businesses, there are no economies absent life. So we need to keep that in mind. And we know that we can't look just short-term, we have to look long-term. And when we do that, it creates the opportunity for enormous benefit. The best model that I know of to show you that is the North American model of wildlife conservation. So I'd like to take you through this just very briefly. So if you think back a couple hundred years, a little bit more, the President of the United States dispatched Lewis and Clark out on their incredible journey across the continent to document the resources, the people, all of the characteristics of the continent. And if you read their journals, they literally ran out of words to describe the wildlife that they saw. So fast forward 100 years, and all of a sudden, Vice President Teddy Roosevelt is hunting in the Adirondacks when President McKinley is assassinated, and the Washington Press Corps says, oh great, now this damn cowboy is president. And that's kind of bizarre because he's admired deeply today. So why is he admired as the conservation president? Well, it's because when he came to office, all of that wildlife was gone. All of it. We're facing extinctions like we'd never seen. 60 million bison, 40 million antelope, dozens of millions of deer. And so the stuff that was there during President Thomas Jefferson's presidency was gone. And we were facing extinction. So the government that had the responsibility to take care of this public resource marshaled their best scientists to try to figure out what to do. And they held international wildlife congresses, and they mapped out some desperation laws to try to protect wildlife. So the first thing they had to do is figure out, what the hell, how did we get here? What was the cause of this collapse? Well, it was the marketplace. If you look at what happened, it was a cataclysmic result of putting a price on the head of wildlife. So when they put these principles in place, where they said this is a public resource, they took the price off its head, they were going to allocate wildlife by law, there was going to be equal opportunity for all, there was only going to be killing for legitimate cause, for food or predator control, but not for sale, and they recognized that wildlife was an international resource, so they had to cooperate with other governments. And then, sometimes called the Roosevelt Principle, they used science and evidence as the basis for public policy. So this model became the world's greatest environmental success story. We replenished an entire continent of wildlife while the rest of the world watched their wildlife disappear. It was a phenomenal result of good government. Good government decisions based on science and evidence. And we stumbled into what we now call sustainable development. Because when we got that wildlife back, we stumbled into the economic gold mine. The value of the wildlife commodity industry, not the commodity, but the activity industry in North America is huge. It's about $150 or $160 billion every year. In 1996 data, Canada got $12.1 billion from the activities of hunting, fishing, and wildlife-associated recreation that surrounded the public resource. Interestingly, that number is almost identical to the $12.3 billion that was the total contribution to GDP from all of agriculture. But we got two priceless benefits from this model as well. And the first is that if you're going to preserve wildlife as a public resource that's valuable only when it's alive, you have to protect the wild places. You can't have wildlife without habitat. And with habitat protection, you get biodiversity. And we don't even understand some of this. But the other benefit that we got was a tremendous protection of the health of wildlife. So how and why is that so relevant? Well, it turns out that most of our infectious diseases come to us from animals. And although this is true, there's a mystery that's involved in this. Because it turns out that it's really hard to get a disease to jump a species barrier. We've been talking about CWD going to other species. And it's even harder to get a disease that jumps a species barrier and then becomes communicable within that new species. So if these two things are true, and they are, then how do we explain that most of our diseases come to us from animals? Well, it's because of domestication. There are a whole bunch of issues that happen that accelerate the evolution and amplify the virulence of these diseases. So I'd like to zip through a few of these. The first is stress. So what happens when you catch a wild animal? Everybody knows this. It's terrified. Well, okay, so what does that mean biologically? Its heart is racing, its blood pressure is up, its respirations are rapid, it's basically into what's called fight or flight. It's doing all kinds of really cool things. It's shifting blood supply to the large muscle masses. It will clot blood from wounds more quickly. Well, all of that, getting it ready to fight or flee for its life, is enormously valuable. But there's another part to it, because the same endocrine system that does that is responsible for our adaptive immune system. Stress, chronic stress, compromises immunity. So then animals in captivity have all of these things. We have high density. We have to feed these animals large portions of the year, so we have to feed grasses and grains. And that brings all manner of rodents and their diseases and their parasites. And then there are all manner of vectors that are involved, insects, sometimes fomites. These are surfaces like plants where they can get diseases. And then there is just this massive increase in exposure. There is squalor. These animals are very often living in very unsanitary conditions. And then this starts to happen. Selection starts to happen. The ones that are susceptible start to die off. But then we accelerate this because we selectively breed for what we want. So we want eggs, we want wool, we want meat, we want all good things that we can sell, and we don't want wild animals trying to kill us. So we breed for docility. So the calmness or the docility of animals that have become more accustomed, they're less stressed, they become more disease hardy. So what we're doing, though, is not just changing the appearance on the outside of an animal. The changes that are happening internally and biologically are phenomenal. So this is a slide from Australia. They looked at what happens to brain size of domestics versus their wild counterparts. The best example I know is wild sheep, whose brains are 50% bigger than domestics. And it's not just that the domestics have smaller brains. The frontal cortex has basically disappeared in the domestics, so it's disproportionate. So what does this mean? Well, when we move animals, we're moving everything that lives in it and on it. And so we're moving the diseases to new environments, inviting new opportunities, and we're spreading whatever diseases the animals might have. So you put all this stuff together and you get this incredible increasing spiral of increasing virulence of the pathogens and resistance of the hosts. So domestication is basically a disease factory. Washington State did an interesting experiment where they took 6 healthy wild sheep, bighorn sheep, and 6 healthy domestic sheep, and they co-housed them. They just had them penned together. The first bighorn was dead in 4 days. The last one was dead in 71 days. And you don't have to do this experiment anymore. UC Davis summarized the data. 100% of the wild sheep will die from diseases the domestics don't even know they have. So if that's sheep, here are some of the diseases that have come to us from animals. So it's a very long list, and the amplification processes that are ongoing are profound. So we need to pay attention to this. So the North American model that kept our wildlife healthy and valuable only when it was alive was challenged beginning in the 1980s. And this was not just a challenge to put the price back on its head. This was a challenge that was extraordinary because they wanted to actually domesticate it. They wanted to purposely raise this for money. They called it game farming. Why do this? Well, these are the things that was suggested that they could sell. In addition to the breeding stock, which was the pyramid breeding scheme at the beginning of this, they were going to sell venison, velvet, urine for hunters, and so-called shooters. These are, well, you'll see what they are. So everybody knows what venison is. It's just meat. But this is what velveting looked like. These pictures were taken in Alberta. This is an absolutely brutal practice of removing living, growing bone tissue. It's very highly innervated. The nerve tissues are amazing. It's so sensitive that you can watch them shake flies off their racks. That's when this is done. And it's done for the sale of an aphrodisiac, that there's not a shred of scientific evidence that it is anything more than placebo. This is what urine products are. I've hunted my entire life. There's no actual reason to have this. And there are claims that, oh, this is urine contains so little prion that you don't need to even worry about it. Well, Nick Haley, that did a bunch of the original tests, is a good scientist. And he followed rigorous protocol. And I guarantee you that that he was testing was pure urine. What's in these bottles is not remotely pure urine. Everything is in there. There's no care taken. They don't insert a catheter into a urethra to get this urine. If these deer stand over these grates, everything is in there. So that's a problem. These are what shooters look like. These are genetically manipulated, mutant antlered animals that somebody will pay a lot of money to shoot. This animal actually didn't live to suffer that fate. There was a combination of things that led to a death. I don't want to speculate about that. But the entire industry, the promotion that was made in the 1980s to do this game farming industry was based in economics. That's what they said. But they had never undertaken any economic analyses. I happen to have a background in economics, and I asked them all the relevant questions. So where are the demand schedules? I want to see the measures of elasticity and diminishing marginal utilities. They didn't know what those things were. And they eventually admitted I couldn't see their benefit cost analysis because they didn't do one. So on the left here is Alberta's 11-page thin double-space analysis of what they did before they rammed this through. It was completely political. It was completely corrupt. On the right is what the government in Wyoming did. When the heir to the Campbell Soup fortune wanted to do this massive game farm in Wyoming under the Devil's Tower Monument, they just happened to try to get this under the nose of some of the best wildlife biologists in the world, Beth Williams, Tom Thorne, a whole bunch of people. And they were in Wyoming and Colorado because that's where the wildlife was. And they said, you want to do what? Fortunately, the Wyoming government commissioned the Game and Fish Department to do a comprehensive analysis. That's what it looks like. I was in Wyoming debating with John Dorrance and his lawyers when this was going on. And when I came back with a briefcase full of stuff from a bunch of these scientists is when I really intently started looking at what was going on here. These are the things that they identified. These were certain results, and why they testified, why those biologists stood up in Wyoming and said, don't do this. You're going to get diseases and parasites and genetic pollution, and you're going to lose habitat as soon as these fences go up. You're going to interrupt migration patterns. You're going to destroy the North American model of wildlife conservation. This has enormous economic costs, and there are all kinds of issues regarding how this is going to affect public policy and the social failures that will be associated with this. First Nations were vehemently opposed to this. These are letters that were to governments from some of the First Nations that I was working with, Treaty 7 Tribal Council, FSIN, CYI. So all across Canada, First Nations were vigorously opposed to this. Nevertheless, they rammed it through, and this is what we got. These pictures were taken, this picture was taken in Alberta. That's not a natural environment. If one of those animals has an infectious disease, they're all going to get it. So we passed this in 1987, 3 years. It took a little over 3 years, and we had a massive epidemic of bovine tuberculosis, just as was predicted. It went to cattle, went to bison, went to pigs, and 42 people had to begin treatment for tuberculosis because of elk farms. Canada lost our national TB-free status because of this. Cost us $1 billion. These are some of the source herds that we got. This one's in Colorado that brought animals in, but this came into Canada in 4 different provinces from 4 different sources and was just a massive problem. Now, in the spring of 1996, like I said, this almost happened the same day. The UK admitted that people were dying of BSE, and the Canadian government announced that CWD was confirmed on a game farm in Saskatchewan. So the other thing that we need to talk about is novelty. CWD, like all of the known TSE epidemics, is new, and it's a domestic disease. So we've talked about scrapie in sheep. That's a domestic disease. There's no scrapie in wild sheep. There's no TME in wild mink. There's no felid encephalopathy in wild cats, right? There's no BSE in wild bovids that we've been able to find. And in the case of CWD, how do we know that this has not been here a long time? Well, because we've done a lot of testing. You should find this, you saw the graphs, the prevalence should be, you should find this in about every 3rd deer if it's been around all the time. Well, in my province, we tested, before we found our first one, we tested 6,883 deer. And I actually did the math. The odds of testing that many and finding only one, if you should find it in every 3rd one, are pretty astronomical. They're 1 in a trillion, 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 keep saying trillion until you've said it 101 times. Or put another way, if you want to prove in particle physics that you found the Higgs boson or something, you have to do what's called 5 sigma proof. These are standard deviations away from the mean. You have to prove it within 99.99997. Just the 7,000 deer that we tested in Alberta is trillions of times beyond that, and you just heard this morning that Ontario has tested 12,000 without finding it. It is not possible that this has been here all along. So people who suggest that, well, we know that it's, we're only finding it now because we're testing, that's just nonsense. And they think that because it's been here all along, if you assume that, it can never hurt wildlife populations or it would have happened. They also say that, well, we know we've been eating deer forever. If it would have been a threat to people, it would have happened already. Every single part of that logic is wrong. So in Alberta in 2007, we tried to be able to contain this somehow, but we didn't. The public wouldn't put up with it. When they saw these pictures of what our cull looked like, and it wasn't properly explained, the public was up in arms. But they were not only up in arms because of this, they were up in arms because the government refused to shut down the ridiculous industry that brought it to Canada. They were continuing to spread it. So in 2008, I wrote this book, no accident, because it wasn't an accident, Public Policy and Chronic Wasting Disease in Canada. And I didn't release the book, although it was published. I instead sent it in a 3-inch binder full of all the documenting science to all the governments that were relevant in Canada, in 4 provinces and to the feds. And I pleaded with them to do their job, to do comprehensive analysis of this, to find out what makes sense to protect the public interest. They refused. Despite all the calls and letters, there was no response. I got sort of a response a year and a half later when the Harper government ponied up $1 million taxpayer dollars of your money and gave it to the game farming lobby because there were political connections. So this is a failure of government. It's not a failure of science. The plant studies, I said that I would offer some evidence of where these people, this is Chris Johnson and Claudio Soto, here are some species that are vulnerable to this. So we know that there is a scientific case for what Norway has just done. That's not the real threat. The scientific case is real. We need to be concerned about this. Other countries have a right to keep this out of their lands and their wildlife and their economies. But what's really going to drive this is when the countries and the companies find out that they stand to gain tens of billions of dollars in market share gains if they can put up barriers to our ag products. So after I found this out, I stopped going public and I started sitting down with governments, with ministers, pleading with them to do the right thing because I was worried that the trade barriers would arrive. So 2017, we published this comprehensive analysis. It was peer-reviewed, distributed widely. We did, Paul Sockett, the former director of infectious and zoonotic diseases for the Public Health Agency, he did with me, co-authored an update in October. I'm in the middle of doing another one. But our attempts to try to get government to pay attention to this are really challenging. We're not getting the responses that we need. So this is Norway announcing the trade barriers. This is what we need to do. We outlined in this what we need to do based on the sound science and the opinions of all the scientists. First thing we have to do is contain the epidemic. Everything you do with an epidemic is for naught if you don't contain it. So we have to stop the movement of live animals and product from game farms, equipment from game farms, and we have to stop hunters from moving carcasses. We have to make sure that nothing is going to the food or feed chains, and then we have to have a collaborative science and evidence-based approach moving forward to try to figure out what to do. So the choices that government are faced with are pretty simple. Based on the science, we can take the vital actions that we need based on the certainties of what we know, based on the risk and the impacts, the outcomes, the potential outcomes, and the nightmare scenarios. And nobody's talked about the nightmares yet. So I'll put two out there. What happens if we're facing the expansion of these trade barriers? What's ag going to do if all of a sudden they can't export? They're going to do what they always do. They're going to turn on wildlife. This is infected vermin. They're shedding these prions into our landscapes and are cutting into our bottom line. That'll be a disaster. It's going to be a huge, huge fight. What happens if CWD is confirmed in even a single person? How do we maintain confidence in our food supply if it turns out that people are susceptible to this? This is in the breadbasket of, this is Nebraska and Kansas and Iowa and Ohio and Wisconsin and Alberta and Saskatchewan. And there are trainloads of feed, billions of tons going into the concentrated animal feeding operations, feeding all manner. And we just found out, included in our update, that pigs, they don't seem to develop the disease, but they're amplifying prions. And it's in their lymph system. And we're potentially exposed. So what happens if we are susceptible to this? And I haven't talked about the real nightmare yet that keeps epidemiologists up at night. What would be the result if it turned out that this not only goes to people, but it behaves in us like it does in deer, where it's highly contagious? Where you could have it for 15 years, 10 or 15 years, be passing it around all over the place, right? So governments have a responsibility to ask these what-if questions. So the vital choices that they have is that we could do this, we could take the vital actions that we need to do to contain this, And we could keep our eye on the uncertainties, whether spiroplasma bacteria would have some effect. Or we could do the second choice, where we could have more study into the things that we don't know, even if, like the spiroplasma bacteria example, it's a distinction without a difference. Nothing about that is going to change the need to contain the epidemic. There's no difference. Official policy across North America is they've chosen the second. It was mentioned that there's a more study that's been announced by in the United States Congress. This is what we're doing. We're going to study this. We're going to continue to foster and amplify and spread this disease. This is the excuse that they give you when you press them. We haven't yet seen a human case, and we're not completely convinced that we're going to see one. So, like Health Canada that said after the macaque study that it looks like the only prudent course is to say that CWD has the potential to go to people. Okay, well, we had to include that modifier potential. It's valuable because it's a limiting modifier, but in science, this is a one-way modifier. There is no outcome out of this that is going to reduce the risk to deer. Finding whether it goes to people or not, the zoonotic question is a one-way modifier. It's not going to change the fact that this can be spread to animals through contaminated feed. It's not going to change the threat to our ag markets and our economy. And I went to the best ag economists and international trade experts in the world trying to find out what would happen if we suffer these trade barriers. That's the most imminent danger. And they said two things. First of all, the danger is greater than you're estimating, and I said it was when, not if. And they said that the consequences would be worse than I said, and I was saying it was going to push us into recession and we wouldn't know how to shut it off. They suggested that it was going to be worse than that because it would shake confidence in our food supply. So things can only get worse with the zoonotic question, whether it can go to people. That's how we have to view this. So the obligations of a government require the precautionary principle and accountability and polluter pay. The precautionary principle, this is a really simple thing. Where there's a potential of severe or irreversible harm, it's a very high bar to public well-being. The absence of absolute proof can't be used to allow or maintain a bad policy. In those kinds of cases, the burden has to be on the people advocating the risk to prove safety. Precautionary principle, it's pretty simple. Accountability and public trust. Everybody knows what polluter pay is. I assure you that the game farming industry has paid nothing for the pollutions that they've caused, the harms that they've caused. And people need to be mindful. Governments and officials need to be mindful. In Michigan, the ridiculous decision to switch to the Flint River water that caused, I think, 12 deaths from Legionnaires and poisoned thousands of people and children with lead poisoning, people are facing felony manslaughter charges. I just hope governments keep that in mind. This is probably one of the most depressing but also motivating things I've ever seen. Paul McCready, this amazing aeronautical engineer that did the Gossamer Albatross and the Gossamer Condor that crossed human-powered flight across the English Channel. When he was, before he died, when he was getting elderly, he was concerned about the state of the world and his kids and grandkids. And so he did this estimate of the difference between the natural world and the human side, the humans, livestock, and pets. So 10,000 years ago, humans, livestock, and pets made up less than 0.1% of the vertebrate biomass on land and in the air, not fish. By the time he published this, that number had changed so that 97% of that biomass was humans, livestock, and pets. That last 3% is what motivates people like me and the people in this room to try to protect wildlife. Wildlife. People want to get into an industry of so-called captive wildlife. Captive what? So this is all about mapping our future. Like we learned with the tragedy that happened that destroyed our public wildlife, we replenished it. We took that tragedy and we turned it into triumph because we had good public policy based in sound science. That's what we need to do again. So I can't finish without mentioning, because my filmmaker would kill me, that we're trying to put this into a film. So if any of you can help us out, we're still struggling trying to get the funding for this, noaccidentfilm.com. You're going to see, I think, a trailer, demo video of what we're trying to do. But I can't thank you guys enough for taking this issue seriously, for inviting all of us here. And I can't leave without thanking all the scientists that are here, because I wouldn't know any of this stuff without their help. They've put up with me over countless hours on the phone because I don't know what they know. And they've been so gracious and so generous. I need to thank them as well as you. Thanks.
Thanks, Gerald. That was very good. Could you comment on the fact that in Europe, in general, over large portions, the landowner has the jurisdiction over the wildlife. And in fact, wildlife is harvested and goes into the food chain in many portions of Europe. And could you comment then, say, if roe deer, which is a very broad ranging species in Europe, gets CWD, what you would predict, and maybe some of your comments based on your analysis here in North America, what you see might happen over there?
So I need to acknowledge, first of all, that I have limited understanding of some of the other models. There are models of conservation that work in other places. They are a bit different than ours. Some of them have been relatively successful. But in general, I don't think they change the rules of biology. They don't change evolution. The factors that are going into it all apply. So when we're dealing with the issues of different kinds of species, you know, I think we have to keep in mind that there will be a continued adaptation for new species, because as we get further down the road in this, we realize that the adapted strains, we're dealing with probably, I think, 7 different strains of CWD now. So this is not a static thing, it's dynamic. This is going to change over time. I don't know, frankly, enough about roe deer or the background of what's going on in Europe to respond to that. I know that there are people a lot better versed in that than I am. But what I can bring to the table, if anything, is the need for us to be humble about this and try to figure out how to work together based on the best science and evidence to try to get the result that's going to help all of us. And I just see that if this continues, we won't have a hope. If any of the worst case scenarios come about, I mean, it's going to be a disaster.
Good day, Daryl. Great job, by the way. Steve Elmy from Belleville. I've had pen deer in the past, about 10 years ago, and for this exact reason, I actually, about 1.5 years in, put the animals down and called her a day. Very expensive. However, I seen firsthand the stress and the levels of these animals that they go through, and it's not fair to them nor anything else. But at the time, we had the CFIA come out and do basically research on the farm to make sure that it was all legit. But what is CFIA doing to shut these down, or do they even care about it in regards to the deer farming?
Okay, so before I criticize what CFIA is doing or not doing, I need to recognize that people cannot put the blame on the people that are involved working for CFIA. So under the previous government, Minister Ritz demanded, he put together a task force, and it was motivated by money. They were spending way too much money on CWD. It was tens of millions of dollars, and it kept finding it, and they didn't know where it came from, and this was a disaster. It was costing, all this money. So he put together a task force to try to deal with it, and there were 18 members on the task force. 15 were from agriculture. Of the 15, 7 were directly from the game farming industry. So from a political science perspective, that's called regulatory capture. And I encourage people to Google it. This is where you've captured public interest with private sector interest or interference. It's not acceptable. So, and 2 of the wildlife advocates that were there were from provinces that they were not allowed to question the policy. So the first thing I need to do is acknowledge that Penny Greenwood and the people that were they're doing their best, but they were operating in addition under circumstances of compromised principles, because they had terms of reference that I've seen that demanded that they try to figure this out on the basis of budgets and politics, not science. So they weren't looking at this from a holistic kind of perspective as to try to figure out how to solve the problem. What they were doing is trying to figure it out in a way that would spend less money and not negatively impact the game farming industry. And what they came up with was at first a zoning approach, and then they, when that was objected to, I think by the industry, but I wasn't there, they implemented now what's called the Voluntary Herd Certification Protocol. And what this has done is that CFIA has basically washed their hands of this and dumped this on the provinces. So any finding in a province that didn't have CWD, or any province that doesn't go into this certification program, and we know that mandatory surveillance didn't work, and Brian Richards is going to be here tomorrow. He can give you the data from the United States where they had mandatory surveillance and it didn't work, right? So what CFIA has done by move to this mandatory, or sorry, the abandoned mandatory stuff and to this voluntary herd certification protocol is a complete violation of epidemiology and the public trust doctrine and professional ethics. It's not their fault. They were forced into this situation, right? It's not fair to the scientists where they were put in a box where they were essentially said, okay, you have to answer yes or no. Did you stop beating your wife? It was not possible to do what needed to happen, which is to contain the epidemic. They weren't given that option. So I'm actually interested in asking the Supreme Court via a reference case whether it's constitutional for the federal government and the CFIA to just, on a reportable disease, to just wash their hands of it and dump it on the provinces. So they did in the case of new provinces with Quebec. The CFIA said eventually that they would go in there and kill the animals and compensate the game farmer, because they have to do that, but only if the province would take it very seriously and kill a bunch of animals on the outside of the fence to try to make sure that it didn't spread or didn't come from there. But at the end of the day, it's not what we need. We need to stop all movement of this disease. It's the first rule of epidemiology, and they're violating it. So my heart goes out as well to the people that got into the legal game farming industry. These are not evil people. They're hardworking, honest folks that got into a legal industry. It's not their job to try to understand transmissible spongiform encephalopathies. I mean, how the hell is that going to happen? So we need to do this collaboratively. We have to have a science and evidence-based approach, and we have to be mindful of all of the things that are at stake, including what my friend Brian Jackson is going to talk about in a bit about treaties.
Thank you very much, Daryl.
Thanks.
That's great. I'm very bad at getting these pictures. So our final speaker from for the morning is Brian Jackson of the Piikani First Nation. Born and raised in southern Alberta, he attended school in Pincher Creek, Alberta, and furthered his education at SAIT in Calgary, Alberta. At the age of 20, he took over the family farm and ranch after the passing of his father in 1980. In 1995, Brian was elected to band chief and council, where he remains today. As a young boy, he enjoyed fishing and hunting with his father and participated as a guide in his father's outfitter and guide operation. He carried on the tradition of hunting for sustenance and to feed the many poor people of the Piikani Nation. Today, he still enjoys the rich tradition of hunting and fishing to provide for his family and many people who still depend on his generous rations of elk, deer, and moose meat. As the elder on his present-day chief and council, Brian brings a rich Blackfoot traditional and spiritual connection to the wildlife his great-great ancestors depended on long before newcomers set foot on the shores of North America. Brian Jackson.
Hoki ni kuuksua, ni tanaku, ni stua aapatsista, mi haputsi pikani ninnaks. Good morning, everybody, distinguished guests, delegates, presenters. It gives me great honor to be able to present a perspective from the First Nations view. I was introduced to this hideous CWD file approximately 4 years ago. I was an elected member of Band Council in 1995. I was 34 years old then. So you can do the math going forward. And today I've learned from my good friend Daryl that it's an issue that requires the attention of leadership, not only on the First Nations, but leadership across the national level within Canada. Daryl came and presented to our council, I believe in April of 2015, approximately 4 years ago. He gave a very detailed presentation on the impacts and how this problem was going to proceed if nothing was done about it. As my introduction stated, I have a history with wildlife and participating in hunting, not only for food and for sustenance, but for a way of life. It provided us with an income when we were younger. It gave us opportunity to provide to our people, the less fortunate people on First Nations that couldn't afford the good food that most of us today enjoy. So through my father's efforts and my grandfather's efforts, we provided rations to our people. And when Daryl presented, that kicked off an alarm in my mind, because I do that still today. And our First Nations people across Canada, of which most are under treaty, there are exceptions in BC where there are modern day treaties that have been negotiated with the federal crown. But for myself, a member of the Piikani First Nation, of the Blackfoot Confederacy, we are signatories to Treaty Number 7, better known as the Blackfoot Treaty. Our territory, our footprint was vast, from the North Saskatchewan down south to the Yellowstone, and as far east as the Sandhills in Saskatchewan. We had the opportunity of looking at what this hideous disease was going to do through Daryl and the Alliance. We moved forward and we made presentation. We presented at the Executive Committee of the Assembly of First Nations, where the National Chief, Perry Bellegarde, called for immediate action. A lot of the chiefs that were in assembly, the vice chiefs that were in assembly that day, were totally unaware of the situation. We presented, and from there we moved forward to a resolution that we gained in Regina in 2017, where they gave the mandate to the Piikani Nation, along with the Alliance for Public Wildlife, the opportunity to and efforts to promote and bring awareness to the Indian people, the First Nations people across Canada. So since then, it's been a constant awareness for us. Wherever we meet, wherever we talk. Daryl and I were recently, I believe it was in December of last year, in Ottawa, where we presented again, and we received another round of support for another resolution that would offer us the opportunity to continue our tireless work. As a leader, as an elected member of Band Council, we have a duty and an obligation to consult and inform our memberships on a wide variety of issues. Daryl came to our what we call a band meeting, similar to this, where he made full presentation on chronic wasting disease. The Assembly of First Nations Grand Chief, Perry Bellegarde, was in attendance. He was also given a presentation. From there, our people started asking questions. They started asking Daryl, they started asking myself, and leadership. Some of the leadership around my present-day council didn't know because they were unaware of it. So it's been the efforts, that's what we've been doing over the last 4 years, is trying to bring this awareness. We've also tried to promote resolutions and ways to solve this problem. It's a difficult uphill struggle. We've tried to meet with federal governments. The federal government has a fiduciary trust obligation to Indian people in Canada to inform them and to consult with them on issues that affect us all. The federal government has fallen short of that mandate. We've taken efforts to meet with Health Canada. We've taken efforts to meet with the Minister of Indian Affairs. Today, we haven't gained any real traction that we're proud to talk about. It's still a problem. So we've moved our focus not only from the federal crown, but to the provincial governments as well. The provincial governments who basically are participants in policy making that drive these policies that give the ability for game farms to operate are, how can I say it best, they're just not adequate. And they're not helping the problem. We are still suffering effects. Fingers are still being pointed. They'll say it's the government's responsibility, and mainstream points fingers back at them. We need to prove the science. We've engaged with people like Daryl and his team of experts, and that's all we ask. That's all we've been asking our federal government, is to prove the science. Prove to us that CWD doesn't pose a harm or impact or any kind of irreparable harm to our people. Prove to us and show us. We all hunt under treaty. These rights were given to us by our ancestors. In our case, in 1877, when we signed Treaty Number 7, we were given the right to hunt and fish for sustenance. That's being impacted. It's being eroded every year by provincial laws, and the federal crown sits back and does nothing about it. We are imposed by wildlife management units where we can't trespass. It impacts our way of life. More so, what causes my attention to this is what the wildlife means to our Blackfoot culture, the old ways, in which what I call them. These old ways are stories and ceremonies and song that have been handed down from time immemorial, long before the Europeans set foot on the North American continent. And those old ways are continuously being practiced today as we speak, and I'm part of it. And it's no disrespect to some of the First Nations across Canada, where someday somebody's going to wake up and say they had a dream, or their grandfather came to them in a dream, and next day I'm a healer, I'm a medicine man. A lot of that happens today, and I respect that. But with me and our circle, it comes from the old way, a transferred right that has come since time immemorial. And these songs and these ceremonies that mean so much to us, that gave us a way of life, are being impacted as a result of the threat of CWD. The thunder medicine pipe that was transferred to me in 2000, which we better known as the rider's pipe, it comes with songs. It comes with a deer song. It comes with an elk song. It comes with a buffalo song. And these songs and ceremonies last a whole day in the spring and in the fall. We pray so that in the spring that we have been given new life in the springtime to gather and collect and to survive another year. In the fall, we pray again so that we can survive the cold winter months, the cold winds that are ahead of us, as the old people will say. And in those songs and ceremonies are these animals, the deer, the elk, the buffalo. They're all a threat. What's going to happen to our ceremony and song if these are impacted? What's going to happen to these ceremony and song if we're not able to collect and gather during hunting season and preserve them till the springtime in the ceremony time? It'll have long, devastating effect. The elders, I spoke with elders last week before I came here, and I told them why I was coming here. There was 4 of them. They told me to speak of this because it's important to our people. Those old ways that were handed down to me are real, and they are from the past. I didn't wake up one day claiming I was a medicine man. I'm simply the keeper of a thunder medicine pipe, one of the oldest and most traditional ways, sacred covenants of the Blackfoot people. So with that, the title I chose when I was asked by Keith what the title of my presentation was, The Blackfoot People and the Survival of CWD. How will we survive it? How will our people move from the threat of what the government did to First Nations people through residential schools, through the treaties, and the promises that were made to our people, of all which we've learned to understand and move forward and try and make the best of our lives in mainstream society and be contributors to society in a good way. But how do we deal with this? How do we deal with somebody like myself who believes strongly in the old ways? How am I supposed to deal with a threat that may threaten the very substance that I gather and the very religion, the practice that I practice every year that my grandfather and his grandfathers handed down to him? It's difficult, it's hard to understand, it's hard to grasp, but we need the government's participation. We need government to wake up, smell the coffee. There's an issue that's imminent that'll affect groups like this. It'll affect groups like this back home in Alberta. So with that, I'd like to kind of cut my presentation short. Thanks to Daryl, he chewed into about 5 or 6 minutes of my time, which maybe is a good thing because I'm kind of getting hungry too. And I'm sure most of you are too. I'd just like to thank everybody for listening to me and hearing me out and all the information that's going to be shared over the next couple of days. It's constantly a learning curve for myself. I've learned new things this morning already, things that Daryl may have told me, but I wasn't listening. I mean, Daryl phones me at the craziest times, and And the only time I get to talk to him is when I'm driving from our tribe to Calgary. It's about 2 hours. So in that 2 hours, I try to catch up on my phone calls, and I spend most of it talking to that guy over there. So with that being said, thank you very much.
We might have some questions. Anybody have any questions? Well, that is it for our morning session. I'm going to be honest, I'm delighted. I've been putting this together for a while, and to see it go and to have such a great host of speakers, I think it sets a really strong tempo for our conference and a great foundation for us to go forward. So lunch today is provided. I'm not sure, we're a little ahead of schedule, so I'm not sure if it's ready, but it will be ready. If it's not ready, it'll be ready shortly. And we will reconvene at, I believe, 1:15 for session 2 on prevention. Thank you all very much.
Well, welcome back, everyone. I hope everyone enjoyed a bite to eat. This morning, our speakers brought us up to speed on what CWD is, how we got here, the situation that we currently find ourselves in across Canada. This afternoon, on the other hand, is all about prevention. What is Ontario doing to prevent CWD from coming into the province? And really importantly, what lessons can we learn from other jurisdictions that have dealt with the problem? Our next speakers are tag teaming their presentation. Chris Hayden and Tor Buchanan are employees of the Ministry of Natural Resources and Forestry in Peterborough. Chris works in wildlife policy and Tor in wildlife research. Chris has advised on wildlife health matters since 2012, working on files such as CWD, white nose syndrome of bats, chytrid fungus in amphibians, and many more. Prior to wildlife health, Chris worked for the MNRF on fur bearer management and rabies research going all the way back to the 1980s. Tor Buchanan is a manager with the MNRF Wildlife Research and Monitoring Section in Peterborough. Prior to becoming a manager, Tor worked in wildlife research for 10 years as a senior wildlife research tech, where part of his duties were to coordinate Ontario's CWD surveillance program, along with rabies research and control and waterfowl banding duties. Please welcome Chris and Tor.
Good afternoon. Lest there's any doubt, I'm Chris, that's Tor. Thank you, Mark, for the introduction. I wanted to thank Carrie and the Board of Directors, Angelo, and yourselves, the members of the OFAH, for the opportunity to talk about Ontario's chronic wasting disease preparedness and potentially response actions in the face of a detection of chronic wasting disease in the province. Tor is going to do the overview of our existing surveillance program, and then I'll come back and follow up with the response plan segments that we're discussing. Take it away, Tor.
Thanks. I just want to acknowledge one other person, too, who couldn't be here today. Larissa Niddick coordinates our CWD surveillance plan. And she had helped develop this project, but unfortunately she couldn't be here today, so I'm stepping in for her. Yeah, so as Chris mentioned, we wanted to talk to you about Ontario's CWD surveillance plan and response plans. How do we move through the slides? This one? Ah, there it goes. Okay. Thanks. So why do we care about CWD? Ontario is a little bit unique in North America right now. We're getting to be on the more unique side of not having chronic wasting disease. We've really covered the basics of CWD this morning very well from Evelyn's presentation, so I'm not going to go too much into those details, but suffice to say, if we did detect CWD in Ontario, it would be very very big problem for Ontario. For our hunting community, it's important. The deer hunt is very important to our hunting community here. It's important economically for Ontario. It's a large generator of revenue for the province and for the economy as a whole. It's important to our Indigenous communities who live in Ontario. It's very culturally significant for them. And if we do get CWD in Ontario and it becomes entrenched, it's going to be very tricky, as other jurisdictions have discovered, to eliminate that disease. So really the best strategy is to keep CWD out of Ontario. And if we do get unlucky enough to detect CWD in Ontario, we really want to detect it early to have any chance of an effective control program. So really what I'm going to talk to you about is surveillance and the importance of surveillance on detecting CWD very early when it comes into your jurisdiction. Okay, so we've already seen this map a number of times, Brian Richards' North American CWD map. You can see lots of endemic areas in the west. More recently, in the Midwest through Wisconsin, we've had large outbreaks as well too. And even more recently than that, in the eastern seaboard through Virginia, West Virginia, into Pennsylvania, large wild outbreaks in those areas as well too. And I'll talk to you a little bit more about Quebec's case. They unfortunately couldn't be here today, but we do have some of the details of their outbreak as well. Sorry, their cases on a game farm. Just to zoom in a little bit closer to Ontario, our situation is unique again here, and for the better. It's different than the rest of North America, where we have excellent geography in the Great Lakes. So we have all these really big water bodies that surround our southern borders, really impedes natural movement of deer populations through those areas. And in between those Great Lakes, we have very fast flowing rivers, which limits, it's not impermeable, but it does limit the deer spread from those other jurisdictions around us that have CWD. So it really does work in our favor, our geographic situation. That's not quite the case in the northwest of Ontario, that butts up against Manitoba and Minnesota, but in the south here, we're pretty blessed with that. And if you see this map, you can see the pink dots are wild cases and the yellow dots are on game farms. Every jurisdiction that surrounds Ontario, except for Manitoba, does have CWD, or at least was detected in New York State, and all the other states have active cases of CWD occurring. So we are literally surrounded by chronic wasting disease. Just to bring you up to speed a little bit on the situation in Quebec, this is new news. Just in September of this last year, 2018, they detected their first case of CWD, and that was on a game farm which had a very large number of red deer in a domestic herd there, up to 3,000 red deer on two separate farms there. So it's a very large herd there. Canadian Food Inspection Agency came in and culled those herds. All 3,000 of those animals have been removed, and about 2/3 of them were tested for chronic wasting disease, and through that they detected 11 cases of CWD on those two farms. This is a really grave concern for Ontario. It's very close to our border, about 15 kilometers away from the Ontario border, and I talked about that good geography Ontario has. The Ottawa River in that area is actually quite permeable, and likely deer are able to cross that river no problem through swimming or even across the ice. So it's not one of the faster flowing river borders that we have. So it is a concern. It's very close to Ontario. We've been working closely with our counterparts in Quebec to keep up to date on their situation, and we're pleased that they have taken such an aggressive response and initiated very rapid control programs there. So that's very positive. Now, if we talk about the Ontario CWD risk model and surveillance program, CWD mitigation, if you're going to have any hope of control of CWD response, it really has to get early detection of those first cases. And really, there's probably likely only been one successful outcome of CWD in North America in New York State. Presumably, they had a fairly early detection. So that's what we would hope for here as well, too, in Ontario. Early detection, and then maybe some control methods might actually have a chance of eliminating or at least controlling CWD in Ontario. So we do annual surveillance program in Ontario, and it's a risk-based program which uses a number of different factors to determine where the greatest risk of CWD might be each year, and that changes year after year. So we look at these from the science out there available across North America. We look at these risk factors, and this list here is in order of importance. So we feel these are the most risky sources of CWD at the beginning to decreasing levels of risk at the bottom of this. So first of all, game farms. They do import animals from across North America in many areas. Zoos also bring in animals as well too. Taxidermists might be working on a deer head from another jurisdiction that has CWD. And neighboring cases or outbreaks in neighboring jurisdictions. So we look at the distance from that case, the intensity of that outbreak in neighboring jurisdictions, and how long ago those cases were occurring. So that's our most important factor in the risk model that we have. Next, we'd have elk and deer densities. So through hunters submitting their postcard surveys, we're able to do a crude calculation of relative densities in some of those WMUs, wildlife management units. And presumably, if you had CWD, a higher density of animals in that would facilitate more spread more rapidly. We look at prior sampling efforts. So when was the last time we went to that area and checked for CWD? If we just did it last year, it's going to decrease in importance in that risk model. If we haven't done it in 10 years, it's going to increase in importance in that risk model. We also look at unstudied cervid populations. So we believe we have some escaped wild red deer in the North Bay area. We don't exactly know what that number is, but that is a risk. We don't know the source of those animals or necessarily what diseases they might have. And other exotic animals on the landscape. Local M&R offices get these reports a couple times a year of a Sika deer or a fallow deer out in the landscape. So we try to incorporate that into the risk model as well. Getting down into low importance in the model, it doesn't really have a lot of impact, or deer aggregation areas, so wintering yards. If a lot of animals come from one area to one wintering yard, there can be disease transmission at those higher densities in that area. And also winter severity may cause yarding activities to occur more often. So you take each of those risk layers, and we calculate a lot of these every single year. We take each of those, combine them all into a computer model, and you can see the map on the right there is a bit of an output map. So you combine all those, and it'll show in a heat-based source where the greatest risk was. This is the actual example of what we did in 2018. So we put all those risk models in, those risk factors into a model, and it outputs at the WMU, at the Wildlife Management Unit level, what the risk is to each unit. So we take an area, a chunk of geographical area, about 12 to 14,000 square kilometers, where the highest aggregated risk is, and usually that's where we'll do chronic wasting disease surveillance in that year. And you can see in April of 2018, the computer model suggested we should go to southwestern Ontario. There's a number of game farms in that area of the country. There's reasonable deer populations in that area. It's in reasonable proximity to the outbreak in Pennsylvania, the south end of the state. And it'd been 8 years since we had last tested that area. So those are some of the reasons why the model would have suggested that area in that year. And so then we chew off those wildlife management units. We always keep our boundaries to wildlife management units so hunters can understand clearly whether they're inside the surveillance zone that year or not. And then our crews will go out and sample in that area. So that was all kind of planned in April, once we get the postcard survey data back and tabulated. Then in September, as we're gearing up to go and do that area, we get the news that Quebec has had a case of chronic wasting disease very close to the border. We had just done Eastern Ontario the year before in 2017, so we'd done surveillance in that area very recently. So we're really overriding the model here and making a human decision that we also need to go back to Eastern Ontario. And our minister did direct us to go and do surveillance in Eastern Ontario that year as well. So we did go back there. We only did one wildlife management unit, WMU 65 that year. We really wanted to focus concentrate our efforts into that one wildlife management unit directly across the border from those cases in Quebec and intensify our surveillance in that area. So we sent out 7 crews, and a crew consists of 2 wildlife research technicians. They go out in a truck, and they look for hunters, and they look for hunt camps. They go and ask the hunters if they can collect a sample from any deer they've hunted. We also have depots too, so to facilitate hunters that might be hunting in the archery season or in the muzzle loading season after the main gun hunt. We try to get all people an equal opportunity to submit their samples, and it just increases our sampling as well too. So we do set up some freezers out there at local tackle shops or at the local M&R office where hunters can go in and drop off their samples if they want it tested. It's completely voluntary in Ontario. So the results were in Eastern Ontario, we were able to collect 300 samples from that one wildlife management unit, which is pretty good from one wildlife management unit. In Southwestern Ontario, that Niagara to Guelph type of area we showed you, we got 457, which is very close to our target of 460. So we're about on target for both of those zones, what we'd like to get in one actual year. And CWD was not detected in any of those areas. Now, I'd just like to point out one thing too about the Quebec case is that, as we've heard today, CWD can have a long incubation time, and just because Quebec has done a lot of testing outside the fence in wild deer in Quebec, and we've tested our deer on that side of the border too, certainly doesn't mean we're out of the woods yet, and we'll need to test in those areas for a number of years to come, and I know Quebec will be doing that, and we'll be considering that as well too in Ontario to make sure that we can keep an eye on any potential spread that might have occurred from that farm. So since 2002, when we first did our first pilot study, we've done all areas of the province at least 3 times, all areas of the province that have significant deer populations. So we've been through the whole province at least 3 times now. We don't do it in order, we do it by risk factor, but we have gotten to all areas of the province. We've sampled over 12,000 deer to this point, and all have been negative for CWD. We've never had a case of CWD in a wild deer in Ontario, in a wild cervid. As was mentioned this morning, we did have several cases back in the 70s at the Toronto Zoo and a mule deer there, but we've never had one in the wild in Ontario. Besides testing healthy animals from hunters, it's also really important to opportunistically test any sick deer that you might find too. These are the animals that have CWD-like symptoms or just any kind of sign of sickness. We try to get these in for testing. They can be relatively expensive per sample, where we can probably collect a deer from a hunter and have it tested for maybe $50 during the hunt. When we go to collect these, we have to have a staff drive all the way out there, collect one deer sample, and bring it back to the lab, which is quite expensive, but really is worthwhile when you consider the different risk factors. And research from out west has shown that those animals that are sick are at 10 times greater risk of being positive for CWD than an apparently healthy animal hunted during a deer hunt. So we do make every effort that we can to collect these animals and get them in for testing. And this is really an area where we need help from deer hunters in Ontario and from the OFAH members to make sure that the word gets out and that we hear about these reports of sick deer and get them into the lab for testing too. Just going to talk very briefly about EHD disease here. It's not on the agenda for today's topic of CWD, but it is recent news in Ontario in that we had our very first case in 2017. So it's caught the attention of deer hunters in Ontario recently, and the symptoms of EHD can be confused, general sickness can be confused with CWD too. So we feel it's important that hunters in Ontario know the difference between the 2 diseases. So epizootic hemorrhagic disease, EHD, is quite common in southern US, and it's often reoccurring in those jurisdictions every single year. Often it will flare up into larger die-offs of deer, but it's almost always present in the deer populations there. It really doesn't have a large effect for the most part on deer populations in the south. It's caused by a biting midge which carries the virus, and that midge is basically unable to survive the Ontario climate. So each summer, these flies, midges, can get blown up northwards on southern winds. And we might see outbreaks in Michigan or Ohio or states like that. And then eventually, even if it's a long, hot summer, even into Ontario, which happened in 2017. But generally, the flies die off throughout the winter and with the first frost, so your risk is back to 0 at the end of the season. And it depends really on the next summer if they're going to blow up again or not. So it is not endemic in Ontario. So we did have 2 cases in 2017 in the London area, and no reported cases in 2018. So it appears it did not blow up last year. And EHD does not affect humans too, to the best of anyone's knowledge. So here's some difference in the signs of CWD. And I would try to keep it fairly simple. Take-home points that you guys could remember is that EHD is really a concern in Ontario in the late summer, fall months. So you wouldn't expect to see a sickly-looking deer sick with EHD in the middle of winter or in the spring in Ontario. That would be fairly unlikely, whereas CWD would occur any time of year. Deer that die of EHD tend to be found in water bodies, so through their sickness and hemorrhaging of blood, they often will go to water courses to cool off, to lower their body temperatures, and take in more fluids. And they tend to die there. The disease is very acute and rapid, and they tend to die quickly and will actually be found dead in those water bodies. That's not necessarily a symptom of or a sign of CWD. So CWD animals tend to die over a longer, protracted fashion. They will slowly emaciate and look really skinny, whereas EHD dead animals will tend to be still quite healthy, like they've died really rapidly, so they still have their fat masses, whereas the CWD deer is slowly deteriorated over time. So just back to CWD, as we've heard today, there are no effective treatments or vaccine. In order to have any chance at implementing a response program, you really need early detection. So that's why it's very critical that we get the word out to OFAH members to report their sick deer that they see out in the landscape, and to participate in the voluntary submission of deer samples when the MNRF crews come around to collect samples. Also, the Canadian Wildlife Health Cooperative phone number is there as well too, and they will test any sick deer at any point in time as well too. So it wouldn't necessarily have to come through the Ministry of Natural Resources. CWCH would also test that animal. With that, I will hand it back to Chris.
Tor had maybe one day's notice as to having to present today. All right, well, thanks, Tor. We've seen the surveillance aspect that's been ongoing for several years within Ontario. We have also had, since 2005, a response plan, prevention and response plan in place, and it has, for a couple of years, been under review, a review which we hope we're getting near the end of. Obviously, we have to review policies and plans for a number of factors, and as we heard from Dr. Merrill today, there's a host of new science that has emerged about CWD since the 2005 plan was put in place that plays into the review process. We've had substantive change in this ministry's own mandate since 2005. We're not so much a hands-on ministry as we were back in 2005. The mandate of other agencies involved in a potential CWD response, such as the CFIA and OMAFRA, has changed as well. We have a much more diverse society in Ontario than we did in 2005, so there's different publics with different perspectives on wildlife management, and in considering CWD responses, we have to look at their different sensitivities in order to garner the support of the broad public and the hunting community. And human dimension science has become a factor now. As looking at other jurisdictions that have had CWD, we can learn from their experiences on how to communicate and engage hunters and the broader public in a supportive approach to managing the disease. The goal of the response plan, therefore, is to have a coordinated response, collaborative coordinated response that, internally we have to coordinate amongst several of our internal divisions. We have a provincial services, regional operations, and policy divisions, each of which would have roles in a CWD response. And we have to coordinate also with our other partners in potential responses, such as the CFIA and OMAFRA, in the event that there's CWD found on a farm. And of course, hunters, landowners, and the OFAH itself are partners in this response as well. We'll put in, back up a bit to the plug with the Canadian Wildlife Health Cooperative as well. We rely on them for disease diagnostic services, not only for CWD, but other diseases, and they would provide us with expert messaging and diagnostic services. Okay, so since 2005, we've taken some actions, and at the time we put the original plan in place, taking some regulatory measures to help protect the province and prevent CWD from coming in. Of course, we have in place the prohibition on the use of natural lures and scents made from cervids. That was put in place in 2010. Such a prohibition is intended to keep prions out of the environment, because we know they can exist in virtually any part of an infected animal. We have had since also 2005 and elaborated on in 2010 regulations on the import of cervids, both live and hunted. The import permit requirement exists for live cervids being imported into Ontario from other jurisdictions onto farms, or in transit through Ontario, or even for slaughter. The permit requirement currently applies to the 4 native cervid species of Ontario: white-tailed deer, moose, caribou, and elk. And as well, we have a prohibition on the entry into the province of high-risk parts of cervids hunted in other jurisdictions. And as you know, the high-risk parts include any part of the antlers, head, brain, eyes, tonsils, hide, hooves, lymph nodes, spleen, mammary glands, entrails, internal organs, or spinal column. The regulation does, of course, allow an exemption for clean skull, antlers, and skull cap, tanned hide, and skin of the head if enclosed in a sealed container, if a hunter wanted to have a mount made of his kill. Under the Fish and Wildlife Conservation Act, there is a section that states that a prohibition on allowing the release of cervids from farms, and allows also the ministry to recover costs incurred in recovering or killing any such farm cervids that do escape. There is associated with that also is a $300 set fine, something of a disincentive. So given that we are in the process of revising the plan, we have opportunity to consider additional actions to enhance the province's preventiveness, as well as to improve any response that we might have to undertake. So possible additional measures that we might take to reduce the likelihood CWD enters into Ontario is to broaden the import restrictions. So impose import permit requirement for all species of live cervids that may be coming from other jurisdictions, and even go so far as to prohibit the import of live cervids from any jurisdiction that is CWD positive. Similarly, a possible action might be to prohibit entry of hunter-killed cervids from jurisdictions that are CWD positive unless the kill has been shown through testing to be CWD negative. Another possible action we could take is to prohibit the use and possession of natural lures and scents for all purposes, not just hunting, but where someone might be using them for photography or just viewing of deer and using these lures to draw a deer in. The prions that are potentially present in those substances don't really care what purpose they're being used for, so it makes sense to prohibit them across the board. Prohibitions on feeding and baiting of cervids under all or under specific conditions is also something that can also be considered. Obviously, baiting and feeding of cervids and of wildlife in general is a pretty contentious topic. We receive letters from individuals requesting that the ministry consider banning feeding of deer in particular. And I've been at public meetings where people have stood up and said that, no, I'll go to jail before I'll stop feeding wildlife. So there's a whole spectrum of sensitivities on that subject that we have to consider, which makes regulating it very difficult and enforcement even more difficult. And as I said, there's only a $300 set fine for an individual, a farmer, who allows for the escape of cervid from a farm. Obviously, that set fine could be increased as a further disincentive. So those are all measures that we can consider looking at as we review the plan. Furthermore, beyond the preventative measures, we would want to look at what exactly are we doing in response to a possible CWD detection in the province. We want to ensure that if CWD is detected, as we heard from Tor, we heard from Dr. Merrill earlier this morning, that a prompt and effective response is key to controlling any outbreak. So we want to make sure that we have the legislative and regulatory means in place prior to detection of CWD to allow for that effective response. We want to ensure we have the ability to establish control and surveillance zones and allow for additional measures that limit disease spread, such as obviously initiating a concerted depopulation of wild deer within this control zone, this response zone. Suspend open seasons in an area where depopulation is actively going on. This would be largely for public and hunter safety. Open seasons such as that would be hunting seasons and possibly trapping seasons in the area within which the control zone, within the area of the control zone. It obviously makes sense to require mandatory submission of cervids harvested within the control zone area for testing for CWD. It's a critical factor in determining prevalence of the disease within the area of the outbreak. And use this prohibition on the movement of high-risk parts of any cervids harvested in a control zone from moving outside of that area to help limit prion spread. Another possible action we could take is to modify the authorizations that are issued to ministry-authorized wildlife rehabilitators who may be rehabilitating deer. Such a rehabilitator located with a control zone, we could look to retract their authorization insofar as they would be barred from taking in deer or rehabilitating or releasing deer. So broadly, beyond an immediate CWD response, there is the possible inevitability that we have to live with CWD across a broader landscape, in which case the province has to be ready to look at changes to how we manage deer in the recreational deer hunt. Adoption of policies that encourage continued hunting and hunting pressure so as to maintain suppressed deer populations in the proximity of a CWD outbreak. Those, you can think of a variety of different means, increasing tag rates, potentially even reducing license fees, 2-for-1 sales, what have you. Anything that helps encourage hunters to continue harvesting deer. So these actions that are proposed are very similar in many respects to those that were undertaken in Quebec recently, and which in turn are very similar to how New York was able to eradicate CWD in their outbreak on wild cervids. In both cases, I think, well, Quebec is too soon to tell, I guess, but in New York, got pretty lucky and was able to eradicate the disease at the very early point of detection. Of course, the actual circumstances of any detection in Ontario could vary quite widely. You can imagine CWD detected at Marine Land and Game Farm in Niagara Falls, downtown Niagara Falls. The response that agencies would take there would be substantially different if it were detected, in rural South Central Ontario or even Northern Ontario, in the Rainy River area, for example. So what I wanted to show is a actual kind of little tabletop exercise scenario based on the response plan elements. So what I've done is just selected a random location within Peterborough County for this hypothetical outbreak of CWD on a cervid farm that doesn't actually exist, but we're just going to play with the exercise here. So there we have about 30-odd kilometers to the northeast of Peterborough, a little bit north of the town of Warsaw, a hypothetical cervid farm on which there is the first case of CWD detected. So the first thing we're going to do there is to establish the enhanced response and surveillance zone. Our guideline for our response plan is a 30-kilometer radius around that point. For Northern Ontario, it would be a larger radius, but for Southern Ontario, looking at deer behavior and movement patterns, 30-kilometer radius would be appropriate. So there's your 30-kilometer radius from Hastings up into Kawartha Highlands Provincial Park. It's kind of staggering to imagine that within that area, there's almost 3,000 square kilometers of land base. That's 2,900 2,800 kilometers. That, for your reference, is WMU 74A and B, within which we know there's an average, or we estimate there's an average of about 2.8 deer per square kilometer. So one of the first steps one has to do, outside from initiating depopulation in proximity to the index case, is to attempt to determine prevalence of the disease in that area. So this is conducted through a helicopter survey, which would take approximately 30 hours of helicopter time, using a double count methodology which corrects for habitat-specific siteability in a forested habitat, forested landscape such as Ontario. So we got there, okay. So this would be undertaken simultaneous to the initiation of a depopulation exercise in proximity to the farm. If we believe we have an opportunity to extinguish an outbreak, depopulation would occur with haste, and for that the ministry would look at engaging the services of a professional company of marksmen who would undertake depopulation. They can remove a large number of animals in a very short period of time, again, similar to what was undertaken in Quebec this past fall. So having determined how many deer we have in the area, we start removing deer from the landscape and testing them for CWD, and we start to get an initial sense of the prevalence of CWD in that area. And this will give us a sense of how low we have to push the population. If the disease is not prevalent, we try to aim for a lower density, about 1 deer per square kilometer. That would mean eradicating something like 1,200 deer from that entire 30-kilometer radius. If we find, though, as we remove deer, that the prevalence of the disease is higher, a higher deer density overall is allowable, and we start to begin to think that we may not be able to contain this, but we nevertheless carry on and continue harvesting deer. At the same time, also again, engage some of the regulatory measures to enhance the recovery, potential recovery or control. As I said, we would suspend licenses to hunt and trap within that depopulation area, the red zone in the center. There would be mandatory testing for CWD of all cervids harvested throughout the entire zone, the ESRZ, the 30-kilometer zone. Prohibit movement of high-risk parts. Prohibit baiting and feeding within that zone. I think that's a pretty safe bet that even if more broadly we don't want to ban deer feeding or baiting across the province, we do want to control it within the response zone. And as I said, modification on the authorizations held by wildlife rehabilitators. These actions go on for 3 years minimum, looking for the point at which we no longer detect infected deer. And that's kind of the gauge that is used. If you go 3 years without detection, you can consider yourself CWD free. Some would say 5 years, 3 to 5 years. If you get out to 3 years, you're probably looking pretty good. If, however, the disease continues and we're having difficulty eradicating it, it may move beyond and be detected beyond that area, then we're looking again at augmenting or modifications to how we manage deer more broadly. Potential manipulation of the age, sex, tag rates, and license considerations to make hunting more palatable to hunters within that area. And that's what it looks like. In a very superficial way. There are other elements at play here. As I said, this scenario is in the case of an outbreak on a farm. Obviously, the Ministry of Agriculture, Food and Rural Affairs has a role, as does the Canadian Food Inspection Agency, for what happens on the farm, and their policy on such an index case would be to eradicate all animals on the farm, try and attempt to stamp the disease out. Potentially compensate the farmer for his losses. This is basically the approach, again, that was undertaken in Quebec. So that kind of is like the ideal scenario where you can bring all guns blazing into a situation where you may have the ability to control CWD outbreak. As I said, other scenarios may not be so favorable, but you try and plan for the best and hope for the best and plan for the worst. As I mentioned, of course, the plan overall is under review. We are hopeful that in the coming weeks we will receive permission to post the revised plan draft on the environmental registry for public comment. That will hopefully occur this spring for 45 days. And we encourage all members of the OFAH to consider how they feel about this plan. And with luck, we will be finalizing the plan in early fall and associated regulatory changes to have them in place this fall. Now, we just have some questions to ponder, and perhaps we have time, I'm sure we have time for discussion still. What are you hearing? What concerns you the most? What advice do you have towards long-term hunter support in a CWD response situation? How can we keep hunters engaged and increase participation in CWD surveillance? So I'll leave it there and have some questions. I hope Tor didn't go too far because there may be some questions of a more technical nature.
Back on slide 19 or 20, you made reference to cross-border. One, two, three. And then he disappears on me. Back on slide 1920, you made reference to enforcement, or excuse me, dealing with cross-border. We don't have borders between Manitoba and Ontario and Quebec. And we do have members who go and hunt the species at risk. Given that there is no equivalent to Canadian Border Services, how are you going to enforce, besides education of hunters, that no, you can't bring your mounts back unless they're already cleaned, which means engaging a taxidermist in the jurisdiction where the animal is taken. And I know personally of a case where an individual hunting down in one of the U.S. states that has got, and we've already had this conversation, took a buck from an area that is a known CWD area, brought it back, Canadian Border Services, where you been, where you going, was down hunting in Ohio or Michigan, whatever it was. No questions as to whether or not you were bringing back any kill or any parts.
Yep.
Now I understand that Canadian Border Services, does not enforce provincial regulations or legislation. And I was told there was an agreement in principle, but in fact there is no agreement in principle, and therefore it's unenforceable. And given that Ag Canada and CFI have washed their hands of their responsibility with CWD, how are you going to be able to implement all these changes quickly enough in order to minimize the potential risk?
It's a challenge.
Two questions.
Two questions. Sounded more like.
And you got a half an hour.
We're going to have to work more closely with Canadian Border Services. So I also heard in the Northwest situations where American hunters are bringing large quantities of hay and feed over the border on trailers, which I would have thought is directly enforceable matter from Canadian Border Services, but somehow they're allowing this just to come over. So there will have to be more discussion, I think, if we're trying to keep Ontario clean and free of CWD. So it's not much of an answer, but that's where we're at right now.
Good day. Good day. Bill Blackwell. I'm from Port Elgin. I'm way at the back here. Just to give you a perspective, you're asking questions about the OFH might think, and I may not speak for everybody, but you know, I had a threat in my house. I had this pile of oily rags beside the furnace, and I knew that sooner or later, if I left them there, it would catch fire. So rather than getting the family all together and explaining the fire plan and when they should run for the phone and run for the hose, I just got rid of the oily rags. So in gaming farms as the oily rags, why are we not making a concerted effort to get rid of the oily rags?
I think they call that an analogy. So the elephant in the room, that isn't in the room, is deer farming and the fact that OMAFRA has oversight of game farms. Without delving into their mandate and trying to rationalize why we have deer farms in Ontario, you can rest assured that we have had considerable conversations with OMAFRA, more so in the last 6 months than in the past 6 years, certainly. And actually, we have to thank your federation for sort of forcing the issue across both ministers' desks, and it's appreciated. You know, your guys' engagement is terrific on this file. It's greatly appreciated. I was told recently that there are yet additional pressures on OMAFRA that supersede chronic wasting disease, but we are engaged in discussion, and there's hope, one crosses one's fingers, that in the coming months there is going to be an improvement, get some of those oily rags at least off the floor and away from the furnace. I'm not at liberty to delve into some of the details and things, but I wouldn't say that there's hope, and I'm optimistic about change if that wasn't the case. So hope that's an answer.
Well, thanks, Chris and Tor. Given that you are the official representatives of our provincial government, I'm sure we could have had probably another 15, 20 minutes of questions. I'm sure there are a lot of important questions that people want answered, or at least want answers from our government on. But we have to move on, and our next presenter is Rich Davis. He's the manager of Manitoba's Big Game Health Program. In 2005, he was appointed to manage programs to eradicate bovine tuberculosis from the Riding Mountain area and to prevent CWD from infecting the province's wild cervids. To date, the Big Game Health Program has prevented CWD from entering the province and is recognized internationally as a leader in CWD prevention. In 2016, Rich was invited by Norway as an expert on a national CWD prevention response to help them develop containment eradication actions to address the disease after it was discovered in that country. Pardon me. Rich has been an active hunter for all his life, is a hunter education instructor, and a member of the Manitoba Wildlife Federation. Please welcome Rich Davis.
Well, good afternoon, everyone, and thanks, Mark. You have to bear with me. Been a long day. I'm one of the ones that got bumped out of the sky when the 737s weren't allowed to fly, and they bumped me from a nice afternoon flight yesterday to a 4 a.m. flight this morning. So it's been a long day for me. If I stutter, if I start falling asleep, I apologize right now. And speaking of apologizing, there's kind of a rule when you make a presentation. You're not supposed to apologize for the presentation. Now, that being said, I'm going to say right off, I'm sorry for my presentation. You see, when Keith asked us to present here, to come out and present to you folks of the Federation, most of my presentations in the past, and I've presented to Manitoba's Wildlife Federation, to a number of their clubs, to communities, to First Nations, to individual hunting clubs and groups and whatnot. And when I present on chronic wasting disease, it's always doom and gloom. You know, I look around the room and I get those big eyes and open shocked mouths, and you talk to folks one-on-one, and some of them they're almost scared to go hunting because of the disease and because of the implications and the worries about it. And now finally I was able to come out to this meeting and make a presentation that's a little more upbeat, a little more positive. You guys have already heard all the bad stuff from other folks. I get to tell you some of the good stuff. So I made my presentation, my title for my presentation, I thought I got to come up with something upbeat. And what I came up with was, Manitoba's CWD prevention program, a success story. Then I thought about that a little while later. That sounds very arrogant. And I really didn't mean that to sound arrogant. So I took it off the title. So and I did promise a couple people here that I would apologize for it. Again, I broke a rule about making presentations, but Manitoba is successful so far in keeping the disease out of the province. I can say that with some confidence, and again, I'm an optimist. I'm a glass half full kind of a guy. The stuff that we have done, I think, has really helped prevent the disease from getting into the province. I think we have a little bit of luck based on the geography of the province, but some of the hard work that's been done by not only my program, but also before my program was even started, has really paid off, and we've been able to stay and keep the disease out of the province. And I'm not going to say so far. Now, when you think about how do you prevent a disease like CWD, it's like they say with pictures in the internet or on social network or social media. It's like pee in a pool. It's out there in the wild. How do you stop it from moving around in the wild? Well, we thought about that. We tried to come up with better things that we could do to prevent the disease from being moved, for being transported, and also to reduce the risk or the transmission of the disease in wildlife itself. We asked ourselves questions. Where is the disease now in not only in Canada but in North America? You've already had presentations on the spread of the disease across the country. How is it being spread? We pay attention to people that are a lot smarter than us that do the research that tell us how the disease is spread from animal to animal and how it's being transported from one location to another. We're not researchers, we're wildlife managers, so we take that information, that the hard work that's being done by a lot of different groups and people, and we try to use that information to our benefit to try and reduce the disease risk and the spread. Now, all these questions I got listed here are things that we have already asked. Are you guys getting kind of a feedback there, or do I sound all right? Or as well as I can sound? I'm getting some kind of a feedback here. Okay, good. We asked ourselves, what kind of actions can we take to prevent the disease from getting in the province, to slow that spread and slow that transmission? And of course, we, down at the bottom there, we started asking ourselves, do we have the legislative authority to be able to stop it? We had to make some changes in our legislation. We modified our acts and we put in regulations specifically targeted towards chronic wasting disease prevention. And again, what else can we do? Has everybody seen this map? I will note that Ontario is included in this version of the map because of that one positive in the Toronto Zoo. There was a mule deer that tested positive for CWD quite a while ago. So if you want to talk about a success story, Ontario was successful. They had CWD and they got rid of it. Well done, you did good. Now, this one, though, is dated from last year, and it doesn't have Quebec in there quite yet, so that map is going to change. So we looked at, we followed the progression of the disease. We looked at how it was spreading, and, where is it showing up? Where does it start, or where did it first start? Where did it first show up, and how has it been progressing and moving out? Obviously, a major way that it's showing up or a way that it's moving is on the back of a truck. I've seen this one in this picture as well. It's moving, and it's being moved by us, moved by people. That's how it got into Canada. That's how it got into Korea. That's how they think it got into Norway. They're not absolutely certain, but they're thinking feed sources. Somebody buying some kind of a supplemental feed and putting it out for their reindeer populations, salt licks, One thing in Norway that they do use quite a bit of are lures of salt lures to draw their semi-domesticated reindeer herds in for harvest. They'll put out salt licks for the animals to congregate. They shoot them and then send them off to market. So there's a possibility that they bought some of that stuff. And we've all seen those ads. Wild TV has so many different ads about all the different kinds of the new miracle attractant for deer and everything. What does that stuff do? Brings them right to one particular spot. The deer and the elk are going to come to that one particular spot, and they're basically eating out of the same bowl. If one animal is infected, there's a good probability, or probably a good chance there, that other animals will be exposed to whatever that animal has at those particular single point source of pollution or point source of infection. And again, you've seen this map. This is why we're terrified in Manitoba, how close it is to the border. There is no geographical boundary between us and Saskatchewan. It's just a made-up line on a map, and it's all prairie. So its habitat has changed quite a bit over the past 100 years, 150 years-ish. Used to be more prairie. Now there's more forest out there, but it's all in riparian zones, making perfect habitat for white-tailed deer to survive and to move into areas where they haven't been in the past. One of the good things, though, is we don't have nearly as many mule deer. And I don't know, it seems like the prevalence in mule deer is significantly higher than it is in white-tailed deer. Now, whether or not mule deer are more susceptible to the disease, I'm not sure of that. I'm not a researcher, I'm not a scientist, but it may be an effect of more mule deer are in the endemic areas. Their population densities are higher in those areas than in white-tailed deer. But regardless, we don't have as many mule deer. We have had people approach us and ask to allow hunting of mule deer in Manitoba for disease control. We're still debating about that one, but we really don't have enough mule deer in the province to warrant an actual hunt, unless it's a disease control issue. Another thing we're concerned about, of course, is the environmental contamination. You've probably seen this picture as well, and I stole this. I did give credit at the bottom there. How hardy that disease-causing agent, the prion, is. Not only, it doesn't survive just wholly inside the body, but once it gets out, it can persist for so long in the environment. And that's a major concern for us. You get that one deer that has a disease, one elk that has the disease and dies, like that area, that spot is now considered a high risk or a highly contaminated spot. And how do you get rid of that? That scares us. And one thing I will point out, at the bottom there I mentioned about sewage treatment. One of the issues that we've run into in the past is dealing with abattoirs, about being able to, how does an abattoir that kills elk from a farm, what do they do with their effluent? Not just the SRM materials, the specified risk materials, the brain, the spinal column, et cetera, but when they're washing their boots off or they're cleaning the killing floor, where does that stuff go? Does it go into your sewage system and pumped out onto fields, or where does it go? And we know that sewage treatment, well, scientists know sewage treatment does not deactivate the prions. So there's concerns that we have there, and I'll explain a little more about that. So these are the things that we're fairly certain that occur when there's CWD. There's nose-to-nose direct contact between individual animals, nose-to-nose, et cetera. I know there was some discussion on fetal movement and whether or not it's the calf that gets it from the dam or however that works. Regardless, if the adult has it, chances are the kids are going to get it. So how that transmission actually works mechanically, that could be debated, but from a management standpoint, it occurs. Now, indirect contact, again, once those prions are outside of the body, all that fun stuff with saliva being left on a food source that another animal might get to, urine tract or left in areas where there's high concentrations of animals, and also, again, transferred at what we call our unnatural sites. And that's deliberate feeding or baiting for the animals, and also accidental, hay bales that are left out by a farmer, not intentionally to feed deer, but they just left them out. Five gallons of coffee and I'm still thirsty. Go figure. And also, the disease is transferred from animal to animal at those sites, but it's also moved in the back of a truck. I printed out my notes, but I should just read it there. It's bigger, I can read it easier. Just a little more, pardon me, just a little general info on wild cervids in Manitoba to give you some perspective. We don't grow a lot of cervids. We don't grow a lot of elk. We don't grow a lot of deer. Moose, we have a fair number, but we're having trouble with them right now. Our white-tailed deer population province-wide is roughly 150,000-ish, but that number fluctuates a fair bit. This is their absolute far north of their range is in Manitoba, and they're greatly affected by weather conditions, seasonal conditions. This year, 4 years ago, we had 2 years in a row of very harsh winters for white-tailed deers, and our populations crashed. I'm sorry, I take it back. 6 years ago, our populations crashed. And we had to adjust our management process. For licensed hunters, we reduced our hunting seasons, and we also went to a bucks-only system to let the deer rebound a little bit. We had a couple of good years, populations came back up, but this year we're now concerned again about a potential crash in our white-tailed deer populations. Had a long winter. It's still a winter in Manitoba. It's been very cold. We had a long cold snap and lots of snow. That's the killer, really, is having lots of snow. The deer can't move around as well, and they can't get to feed. Brings into play some of the issues we run into, though, when they're yarded up in these deep snow years. They're concentrating even more so at more concentrated sites. More potential for disease transmission rather than being spread out a little bit. And also, just to mention, we have woodland caribou. There's a population of woodland caribou, and we share barren ground with Ontario and Nunavut and Saskatchewan as well. That's on the wildlife side. Just wanted to get you in perspective there. And again, mule deer, they're considered extirpated in Manitoba. There were once more in the province, we believe, but because their populations are so low, we've listed them as extirpated, and that means they're protected from hunting. Now this little spot here, that's our game hunting area 27. Not a very big spot, but I'll show you, I'll get another slide of it, it'll show it a little better. In that game hunting area, I flew an aerial survey 2 years ago. We covered the area 100%. We counted over 3,000 white-tailed deer in that spot, in that game hunting area. We counted 26 mule deer. Two groups, 12 and 14. Not a lot of mule deer. So, now to everybody's favorite. Now, in 1996, Manitoba got into the elk farming business. Farms were set up with the encouragement of the provincial government. They were capture farms. The elk populations were stable or growing in some areas. There was concerns about depredation issues where the elk are feeding on bales and et cetera, running through fences. The solution was to capture some of these elk and start an elk farming business. At the time, we only allowed, or the province allowed, capture of the Manitoban elk. It's a subspecies of elk, Manitoba, or Manitobensis. At the time, there were around 90 farms that became established. It was smaller in the beginning. The capture farms got a bunch of animals, and they sold those animals to as seed stock to the other farms in the province. About 90 were established at the peak. Now, the products, we didn't allow hunt farms, but we did allow for meat farms, for velvet, and for hard antler production. and for breeding stock for other farms in the province. And of course, we demanded provisions to be put in place to make sure that those animals were Manitoban or Manitobensis and weren't, the genetic purity was kept there. So that there wasn't crossbreeding or there was some goofiness with red deer hybrids or anything like that. We wanted to make sure, in case those animals did ever escape out of the farm, that they didn't genetically impact the wild herds. So we were successful in getting some restrictions put on there. And we also wanted the animals tagged, big dangly ear tags, so we could identify them easily. And very strict records are supposed to be kept by the elk ranchers. Now, in 2002, our good friend CWD started showing up, and we began to put in restrictions. We banned imports of elk from outside jurisdictions. So it was only Manitoba elk were allowed in Manitoba. I'm going to give a shout out to our Manitoba's version of this group, the Manitoba Wildlife Federation. They were instrumental in getting this kind of stuff done. They were adamant about keeping Manitoba elk Manitoba and making sure that CWD wasn't brought into the province on the back of a truck like this. So they were very key. They're a very vocal group, and they have a very strong lobbyist policy to make sure that this stuff doesn't happen. So I'm just giving them a shout out. Like, those guys are doing good. And this year, the last couple of years, they've really started taking the CWD issue by the horns and greatly increased the communications with the hunters and greatly brought up sample submissions to me from hunters just through their efforts and through their communications within their smaller clubs. Now, in 2002, we put this regulation in place, and no imports, no live animals could be brought into the province. Now, we had lots of arguments, we had lots of pushing back from elk industry and even from our own ag folk about us being too draconian on this. We're restricting trade. There's even threats of suing us under the provisions of NAFTA because of this, but we were adamant about it, and with the Wildlife Federation's support, we were able to stick it in place. And of course, 5 years later, our agriculture department backed off a little bit and said, okay, you can now bring in elk into the province if it's from a jurisdiction that has been negative for chronic wasting disease over the past 5 years. Provisions we put in place, though, we demanded that they had a surveillance system that was at least as adequate as Manitoba's. And Manitoba, all on-farm deaths are required to be tested for CWD. All animals that slaughter are required to be tested for CWD. We have mandatory sample submission from hunters in our disease control zones. No jurisdiction matches that. So we've been effectively able to shut down live import of elk into the province because of those provisions. Now, to me, that's kind of weak legislation. That leaves a loophole or leaves a gray area there that somebody could bring an elk into the province. But it's there, and it's worked so far. Now, currently, it seems like that industry has really fallen by the wayside in Manitoba. We have 16 licensed farms as of December 2018. 16 licensed farms. About 2,000 head, with only 3 or 4 actually large production farms in the province. Most of the head are on those farms. The rest of the numbers, 5, 10 animals, 15, 20 animals, are on smaller farms, and they're basically pets. They raise the animals for a little bit of meat for the family, and some of them only have, I've seen like 3 animals on a game farm that's licensed. Those little stars, those are the locations of the farms, just to put it in perspective. When I talked about the effluent from the slaughterhouses, in 2007, agriculture also started to allow animals back into Manitoba for slaughter, to go direct for slaughter. The trucks had to be locked and sealed so they wouldn't drip any fluid as they got to the rendering plant, or pardon me, to the abattoir, the slaughter plant. But we got a call from our ag folks a year, maybe 2 years ago, saying there was a farm in Saskatchewan that wanted to ship animals to a abattoir in Manitoba. The abattoir was the one that was actually pushing for it because they're trying to increase some of their production. They weren't sure if that could be allowed under this. So we reviewed it with our ag counterparts, and we looked at it and said, well, what's happening with the effluent? How are they washing the trucks? How are they making sure that they're not hauling urine and junk all over the place, or what are they putting back in the truck after it goes? What about the wash water? Where's that going? Talked to the abattoir, and the way that they were handling the effluent is it went off into their pit, their sewage pit, and when it got full, they would pump it out onto the fields, adjacent fields, agricultural fields. Then the sludge would be scooped up and hauled off to a landfill site. That wasn't adequate because we know how persistent the CWD prions are. They're going to survive that. As a matter of fact, it's going to make it worse. They're going to dump it onto clay fields, like clay particulate soils, where it becomes more infectious. It's going to be picked up by the plants. Other animals are going to feed on it, including elk and deer in the area. So we said, oh, wait a minute, this can't happen. We don't have the legislative authority to say no, except our environment department does. And what happened there, that particular abattoir was only licensed to handle cattle, pigs, and chickens. It couldn't take elk or deer. So again, weak legislation, but it worked. We were able to keep those animals out of the province. Things we got to work on, obviously. Now, what have we done to try and stop this? There's a ban, again, under LEDA, live cervids can be brought in from jurisdiction that's test negative for the past 5 years. So there is some restrictions there, but every other cervid besides elk is falls under the Wildlife Act, under the control of my director. And we have to, the only way we allow exotic cervids or native cervids into the province is if they're permitted. We have to give them a permit, and we don't give them permits, simple as that. So we banned it. We're not going to allow live import. That includes animals going to the zoos or to petting zoos, or somebody wants a pet Nipponese deer or something. Nope, you're not getting it. So basically, Manitoba is effectively shut down for imports of live animals. They can't be brought in a truck. Now, we banned the import of unprocessed cervid carcasses. Now, this regulation was put in place back in the day when we thought CWD was similar to BSE. It was contained within the SRMs, the specified risk materials, brain, the spinal column, the head, the hide, hooves, mammary glands, entrails. So we banned importing that. Hunters could still go out of the province, shoot a deer, bring it back in as long as all that stuff was left behind, and they brought basically the meat back. We did allow them, or we do allow them currently, to bring the antlers back with the skull plate attached as long as all soft material, soft tissue is cleaned off and it's been disinfected with a 2% bleach solution. Capes can be brought back, but they have to be in a sealed container and delivered to a taxidermist within 5 days of import. We are in the process of reviewing this because this was, again, put in place when we thought CWD was similar to BSE. If we could stop the deer from eating the infected brains of deer, then maybe we could stop CWD, but now we know that's not the case. So we may increase the restrictions there. We're actually looking very hard at that. Now these signs here, we found the same problem that has been mentioned earlier in some of the presentations about, and even one of the questions, like how does a hunter from Ontario know what the regulations are in Manitoba or Saskatchewan? We've got to do a better job of communication between us, these jurisdictions, between government agencies, and try to figure out a way to make the rules the same. It's not only us that has that difficulty, the U.S. has the same. There's restrictions on, you're in a CWD endemic zone, you can shoot a deer, you can bring it to another CWD endemic zone, but you got to cross these states that don't have CWD, so you got to follow every one of their rules, and it's a dog's breakfast. Trying to make sense of those regulations is tough. So we got to do a better job at dealing with this. And back in the day, we collaborated with Ontario Ministry of Natural Resources, and we bought these big signs and stuck them up on the highway. Took us a while to get them up, but we do have signs up. I've got roughly 16 highway signs in place. I'd have to double check. But we're doing that, and we're trying to put it into each other's hunting guide and on their website to try and educate some of the hunters on this stuff. And people wonder, like, why do we need these kind of signs? Like, if you're going in someone's, into the province, you should know the regulations. It's because of this kind of fun stuff. This is Quebec hunters that traveled to Saskatchewan and into Alberta. They shot a bunch of deer, 14 of them to be exact, and they were heading back to Quebec with these deer. We seized them in Manitoba. There was a bit of a gray area there because the regulation states you cannot import the carcasses unless they've been cleaned of all those SRM materials, but the regulation was a little weak in that if you're driving through the province, is that actually bringing it into the province? We charged them anyway. So we seized it. These all tested negative for CWD, but we took it away from them and sent them on their merry way. We've prohibited possession of substances containing bodily fluids from cervix. And there was a question earlier about the Canada Border Services Agency. I'm pleased to tell you that a year or so ago I got a phone call from those folks. There was a truck that was coming up from the US that was heading into Manitoba, and it had a couple of crates of deer urine in the back of it. The little bottles, they were packaged up, and they were being shipped to Alberta, actually, to go to the Bass Pro Shops in Alberta or something. But the CBSA folks in Manitoba stopped the truck and turned them around. So they do enforce Manitoba regulations. It may be sporadic, I don't know, but I was pretty impressed and glad. I honestly didn't think that a federal agency like that would care about Manitoba's goofy regulations. But anyway, we've banned the possession of these province-wide. So you can't have any of these materials. And that's just some examples of them. There's lots of different ones. We did find that there's a lot of artificial attractants, the synthetic ones. So there really was no impact to hunters that rely on this kind of stuff. And even to the folks that sell it, they didn't have any problem with it. So we made sure, but we didn't have any real fight over that one. Another one, next part that we've done in Manitoba, we've designated our disease control zones. Based on the information from Saskatchewan and watching the disease spread there, we set up the Great Wall of Manitoba, which is, this area here is designated in regulation as the chronic wasting disease surveillance zone. In that area, pardon me, that's a mandate. Go back there. Go back there. That's the, we have our CWD regulations are in full force in that area. The area along the bottom here is a voluntary zone along the U.S. border. We started as everybody else does. The disease showed up in North Dakota, so we started doing voluntary sample submissions along the U.S. border. We designated an area, 2 townships at 12 miles, easily or seen from the ground with the mile roads we have. We started collecting samples. Collected them for 5 years, no positives. We talked to North Dakota. We were pretty satisfied with their wildlife sampling, and we decided, okay, enough is enough. We'll stop sampling for CWD. 1998, though, bovine TB was found in Rocky Mountain National Park in livestock and later in elk and deer. And that caused a whole whack of things, long issues. During the time we started collecting mandatory samples from hunters around Rocky Mountain National Park, which is this area right here. That's Rocky Mountain National Park. We also did a subsample for chronic wasting disease. We later on increased this area for bovine TB to include this area here, which is our Duck Mountain area, which is a high elk population and deer population, just to be sure the disease hadn't been spreading into another elk population. Mandatory samples were required from the hunters, and we did a subsample for CWD as well, all negative. Done in 2003, we designated that area that along the Manitoba-Saskatchewan border as our CWD surveillance zone. We increased the area where mandatory sampling was required. And now in 2018, we've reinstated that voluntary area along the U.S. border, set up drop-off depots. I've got 31 drop-off depots set up for the hunters to submit samples. The idea is we may end up turning that zone along the U.S. border into mandatory as well. We just want to see what our response is. And again, the Wildlife Federation has been great. All the clubs along there have been encouraging their members to provide samples. So our numbers actually came up pretty good. I'm running out of time here, aren't I, Mark? Okay, I got to get going quicker here. Now, just zoomed in a little bit, there's our bovine TB zone around Rocky Mountain National Park, and our chronic wasting disease zone goes all the way north to the little town of Flin Flon, and now south all the way down to the U.S. border and across the U.S. border. It's an older map. Mandatory sample submission along the Saskatchewan border in those game hunting areas. Now, licensed hunters are required to submit biological samples. Some folks ask about that mandatory sampling, like how many people actually bring in the entire head from a nice trophy buck. I figured about 80%. I looked at the number of samples that I got. I looked at the number of samples that hunters claimed that they had submitted, or pardon me, claimed they had shot, compared the numbers, and it comes out to be about 80%. Different levels of participation, but not too bad. And again, along the U.S. border, just request the samples, or voluntary samples. But we're starting to get some bite into that. It's only been open for 1 year, but we are starting to get some samples coming in. To date, over 10,000 white-tailed deer have been tested. All the little dots you see there are different samples I've collected up to 2017. I don't have it updated yet. But it shows you samples that we've collected in the past from the voluntary area along the U.S. border. That doesn't include the ones from last year. So these are the ones from 97 through 2002. And all of these, that's a Rocky Mountain area. You know, there's 20,000 samples in there. It's hard to pick out an individual dot. But we haven't found CWD in any of those animals that we've been testing. And we sporadically test for, well, whenever I get a moose that comes in, I'll test it for CWD. Mule deer gets hit by a car or accidentally shot, and we test them, or if it shows with signs of clinical signs, we test those as well. Now, feeding of cervids, this is another question. It's illegal to bait cervids in Manitoba for the purpose of hunting, for licensed hunters. That regulation does not apply to Indigenous, to First Nations, to Treaty, to Métis, or to Inuit. They are allowed to harvest wild foods without restrictions like that put in place. Now, in Manitoba, of course, like for licensed hunters, it's illegal to bait for deer. You can't put out any kind of bait for deer. Now, in 2014, we had a little bit of an issue in our CWD control zone with a group of First Nation hunters. There's an old abandoned elk farm. They took the feedlot from the elk farm. They put a bunch of bait in the middle of it, hay bales and whatnot, waited till the wild elk came in and then shot them. You know, they posted the pictures on Facebook and all that, and there was a hue and cry from the public, from the media, from other treaty folks, from other Métis folks. Like, they didn't think that was right. But our officers looked into it, and there's nothing wrong with it. It wasn't illegal. Our minister got angry. Everybody got angry. So we said, okay, it's an obvious disease risk. They're bringing these animals in and feeding them in one particular spot. You know, we got to stop this, but we didn't want to infringe on rights. We can't infringe on rights. So we started our consultation process, and we did what's known as Section 35 consultations with all the First Nations and Métis groups that use those areas for hunting, for harvesting wild foods. And overwhelmingly, they were in favor of a feeding ban. So we were allowed, or we put in a ban that prohibits feeding of cervids at any time of the year in our disease control zones. So there's no feeding of cervids allowed in those areas. We also look for risk sites. Now, that's one of the things that people ask me for a fair bit, is how do you find these sites? Like, how do you know where the bait sites are put or people are feeding? Well, we go and look. That's simple as that. You can't see them from the road very often. You usually got to get up in the air. So every year we fly an aerial survey, and I look for disease risk sites. At the same time, we count cervids. We look for congregations of animals, and we try and get a sense of the density of the animals. Same time, I'd look for ear tags in elk, look for escapees, and look for all the natural concentrations. There's me by the helicopter. I just want to show you that picture because I'm the one that flies in the machine. And I get tired of seeing the helicopter pilot's eyes get big when he sees me walking up to this little tiny machine. It gets off the ground, even with me in it, I swear. Now, what we're looking for, this is a picture that I took of just a group of elk there. I'm looking, in this, obviously 3 elk, easy to count, but what I'm looking for are ear tags. Because we have had a number of animals in Manitoba that have escaped from elk farms. If they escape from an elk farm in Manitoba, the farmers contact Manitoba Agriculture, they're required to, and they're usually given 30 days to try and get that animal back in the farm. We don't have a fine or anything. I found that interesting that Ontario actually charges the farmer for escapees. We try to work with them to get the animals back. But we have shot animals in Manitoba from Saskatchewan with Saskatchewan ear tags. So every time I fly by an elk, I look for those ear tags. This was just my attempt to try and take a picture because it's difficult trying to see those ear tags from the air when you're buzzing by in a helicopter and those things are running 100 miles an hour. But as you can see, like even high resolution zoomed in, you can't see. You can't see definitely there's no ear tag there. The best way is just looking at it visually, sitting in the actual helicopter and looking for it. We look for accidental feeding sites. This wasn't one that I found. This is actually one from Trent Bollinger's study out of the CWHC in Saskatoon when they were looking at CWD. Now this deer here did test positive for chronic wasting disease. They set up trail cams at these sites. It did die. That's why they tested it and found it was CWD. And other deer, of course, were seen feeding at these sites. Accidental site, though. It's not something we can charge somebody for feeding. It's just grain that was spilt when they were transferring the grain. We look for illegal baiting. Baiting's illegal. Pretty simple. Got a tire there with salt, big pile of grain, and there's a tree stand sitting up here if you can see it. And this is right up along Riding Mountain National Park. No feeding in the park. Pardon me, no feeding. No hunting in the park. There'll be no feeding or I'll get angry. But these are impossible to see from the road. Rolling hillsides. You could drive by a mile and you never see something like that. So we have to use a helicopter to look for these things. We even thought about using drones, but apparently you can't fly a drone with a camera over private property. That's a rule that we didn't know. We look for deliberate feeding. Our friends the deer running away from feeding sites that are set up so folks can have their toast and cereal in the morning and watch the deer outside. Now, what do we do about these sites? That's another big one I get. Our officers are given the powers to do that. We have the authority, legislative authority, regulatory authority to enforce our no feeding, our no baiting. We also gave them the kind of a blanket authority that the officers, with their discretion, they can order to remove any substance, anything that is attracting cervids or putting wildlife, people, or livestock at risk. So they have the authority to go and order somebody to remove. If they've left a bale out in the field, they can tell the farmer to move that. If the farmer doesn't, then it's a charge under failing to follow the officer's order. So they have that authority to do that. Some areas we post as we designate an area as no hunting. Someone's been baiting. We can put posts a half mile around the area for no hunting. And direct charges for baiting for cervids for hunting, and also in our disease control zones, charges for feeding cervids, just a flat out charge. As well, order to remove, all that stuff comes into play. Now the other ones that we look at, we find, are kind of accidental. The deer feeding at a farm site. Heavy snow year, they're yarded up, they're getting into bales. This farmer actually didn't do too bad of a job. Oops, they've got the bales stacked on end and double row. That actually helps prevent deer from getting into them and elk from getting into them. But there's plenty of stuff where they busted bales up that the deer are feeding on. So we want to definitely address that as a potential disease risk. Other ones, bale grazing. This is gaining more and more popularity. Obvious attractant for high cervids or high cervid population areas. This is right up against Rocky Mountain National Park, our highest elk population. But what do you do? How do you stop that? Like, we're not anti-farmer. You know, we don't want to prevent somebody from doing these kind of practices. That's good environmental farming right there, but it's bad for the elk. So what do? This kind of stuff. We've provided hay storage fences for over 175 farmers around Rocky Mountain National Park. We're starting to provide barrier fences for farmers in their CWD zones. Just starting to. They're 8-foot high game farm fences, about an acre in area. Cost about $6,000 that the government is paying for the farmer. They work. That's the best way to keep the elk out of there. I don't have pictures of it, but some of the other things that we've done are livestock protection dogs. For instance, in that bale grazing situation, if that farmer had a livestock protection dog, you know those big white shepherd dogs that chase Wile E. Coyote around? Those dogs are actually, we've been working with our ag folks on some experimental work on that, putting those or getting those dogs to bond with livestock, with cattle herds, and it's working. There's some really good evidence to suggest that they actually stay there, and they're not sheep dogs, but they're not with the sheep. And the idea is they chase away coyotes and wolves, and just their presence will keep deer and elk away from the cattle herd. And we started that with our bovine TB to try and stop that disease transmission, but it does work. Another one you could use are what known as 3D fences, which is basically an extra strand of electrified barbed wire put out from a regular fence. Gives a visual block for the deer and elk. Plus it gives them a little zap when they touch it, but it prevents them from getting into areas where they're feeding the cattle and getting in and mixing up and feeding at that common feed spot. So basically, I'm done. I've been rambling on for a long time, and I see lots of people nodding off. That's my 2 o'clock time frame there, Mark. There are 6 things that we have done, and just to reiterate there, we banned the import of live cervids. In effect, it's been banned. We've restricted carcass import. We've prohibited feeding in our disease control zones, and as well as baiting. We've banned the substances with bodily fluids. We conduct active surveillance in our disease control zones, and we conduct our regular surveys or survey areas, and we look to mitigate disease transmission risk sites. That's it in a nutshell. That's our six little steps to try and prevent the disease from getting into Manitoba, and it seems to work. And we're always looking for better ways of doing things, and always trying to fine-tune things and looking for whatever we can do to prevent this disease. So far, it seems to be doing pretty good. Now, I won't go too far into this, but we do have our action plan in place. Chris talked about Ontario's action plan. Ours is fairly similar, but we are currently working with CFIA and with Manitoba Agriculture to refine it and to upgrade it. Like CFIA will still respond in Manitoba to a positive farm as long as there is an active wildlife response outside of the farm. So what our policy is pretty straightforward. It's a scorched earth policy. We draw a 20-kilometer circle around the positive farm or positive wild animal, and we kill every cervid in that 20 kilometers. Now our plan is to go in there before the first calving season. If we detect it in November in wildlife, we'll get in there in February or March and kill everything in that 20 kilometers. We'll also create a 70-kilometer zone and set up additional hunting licenses or opportunities, figure out how many animals we need to detect for the disease, and go from there. It's not pretty. I'm not looking forward to it. I figured out in that game hunting area 27 with those 3,000 white-tailed deer, I drew a circle on the area and counted the number of animals in there, including moose and elk and 2 mule deer. I'm going to have to kill 1,750 of them in the first year and keep doing it every year for 5 years until we get 5 years of no positives. So we'll keep doing that. We'll keep it serve it free. It's not pretty, but that's our response plan. I'm done. Yep. Okay, any questions?
I will take one question.
Apologize for talking too long.
Hi, I'm Helen Gerson from Canada Border Services Agency. And I just wondered whether Manitoba has approached the CBSA to work with you at the border, as did Ontario and Quebec. And we are working, can't directly enforce provincial legislation, but we can share information when there's a threat to the environment. So border services officers can inform provincial authorities that a hunter has come in, or they can also inform the hunter that they cannot bring the carcasses into the province. And if they do, then their information will be shared with the provincial authorities.
I'm not the one to really answer that question. That's more of an enforcement question. But I know our officers have worked and have discussed things with CBSA on a number of occasions and work with them on the CITES permits and export of things. I don't believe anybody actually talked to them about that little incident prior to that incident when the urine was being brought into the province. But the CBSA did that kind of on their own, as far as I know. And I give them total thumbs up for doing that. But that is a good point, and something we should be looking at, I think. Try and work on that.
Thank you, Rich. Thank you. Thanks a lot, Rich. I did let Rich go a little bit longer because I think their prevention strategy is fairly robust and admirable. So, and also because it's a coffee break. So I figured we'd eat into our coffee break a little bit. I'm going to take at least a 10-minute coffee break, maybe 15. It is, if we could be back here as close to 10 after 3 as possible, I'd really appreciate it. Thanks. Okay, our next two presenters hail from New York State. Jim Farquhar is currently the Chief of the Bureau of Wildlife for the New York Department of Environmental Conservation. In 2005, when CWD was detected in Oneida County, Jim was the regional deer biologist in that area. Jim was involved in the field response planning and actions during this incident. Since 2005, Jim has overseen continued surveillance efforts directed at determining prevalence of CWD in the affected area. He also played a lead role in drafting New York's interagency CWD response plan and risk minimization plan, both of which are now in place to guide efforts to reduce the risk of CWD entering the state or to respond in the case of a new detection. Justin Gansowski is a district supervisor of USDA Wildlife Services in central New York. Justin started working for Wildlife Services in 1997 as a wildlife technician. He has a degree in wildlife management from the State University of New York, Cobleskill. He has worked with double-crested cormorants, white-tailed deer, gulls, and feral swine to protect agriculture, natural resources, property, and human health and safety. Please welcome Jim and Justin.
Well, thank you. Yes, I am the Bureau Chief now, but in a previous life, I was a practical field biologist with some actual skills, and I appreciate you folks inviting us here so that I get to relive those days just a little bit today. I'm not being a overpriced bureaucrat today, so really appreciate being here. Another comment I want to make is I think this is just a fantastic event here. Not only do you have, maybe myself excluded, some really outstanding speakers and some really great information being shared. But the fact that you've got this has been put on by anglers and hunters, folks that are out in the field, and you've got that kind of an interest, tells me something very, very positive about the outlook for CWD management in the future. And as I'll talk through this presentation, you're key to this equation, and we probably haven't talked about that quite enough today. Assuming it's this green button. I want to start our story a little bit before 2005 and the response that we did in 2005 when we detected CWD. I think it was Chris who said earlier at one point, made the comment that New York got lucky. And, you know, that probably I could sum it up and stop right there and walk out, because I think luck was a big part of what happened in our case. However, I think sometimes you make a little bit of your own luck, and I think some of what we saw from Ontario and some of what we saw from Manitoba speaks to that a little bit. If you get your plans in place, if you're working on these things, and if you're kind of paying attention, you probably have the potential to influence some outcomes and potentially at least slow down or at least reduce the risk of CWD in the area that you're interested in. Prior to 2002, like everyone else in the eastern part of the country, probably the eastern part of the continent, we weren't paying a lot of attention to CWD. It was this thing that was occurring in the western states. It really was not that significant to us, a handful of states. Yeah, we were paying attention, but you know, it wasn't important. But in 2002, I think, as we all know, that's when CWD was detected east of the Mississippi River in Wisconsin, and that did get our attention. So we started the process of CWD management in 2002 in New York State. That started with regulation, and that process can be very, very painful no matter which jurisdiction you're from. But one of the things we did was ban the importation of live deer and elk into the state. And that actually was probably one of the key things that helped us out there. We initiated a feeding ban, and that was amazingly painful when you were working in an Adirondack portion of New York section where feeding was very, very prevalent. We hosted a meeting that I like to say 200 people came to and 198 were peed off. It was pretty ugly. I really didn't think there'd be tires on the car when we left that night. But we implemented it, and to a fair degree, people are complying with that feeding ban in New York right now. And then we put parts and carcass restrictions on CWD positive states at that point in time so that they could only come in with the clean, boned out meat, the clean skull caps, all of the things we've talked about. And this gets to some of the cooperation. Cooperation with our Department of Ag and Markets with a herd certification program with a separate set of regulations that they promulgated that got us into the testing of the captive herds, sort of parceled them out as certified herds, all deaths tested, and what they call special purpose, where a percentage of the deaths were tested. And the purpose of those were real simple. We wanted to see if we could prevent the introduction of CWD into the state. If we had it, contain that spread, and obviously, first and foremost, for a wildlife agency, protect the health of the wild deer herd in New York State. And those remain our goals to this day. So just to give you just a little bit of a graphic, a pictorial of New York State, this is what happened over the course of 2002 to 2004 before we detected CWD and when we really got started and were beginning to at least pay attention to the disease. Obviously, a lot of holes in New York State at that time, but we tested about 3,500 deer in those in the period of those 3 years. Might add in 2002, because that detection came, I think, in April or May or maybe June in Wisconsin. We actually sent staff out and probably collected several hundred deer during the summer months so that we would have a sample, a representative sample around the state in order to assure hunters going into the fall that we were fairly confident we didn't have CWD. And then we started working with hunter-killed collections from that point on. So along comes 2005. In late March of 2005, a deer was sampled as part of routine surveillance on a farm. Incidentally, that deer was butchered to go to a game feed charitable event at a fire department, and it was consumed by all of the attendees before we got their test results back, so that created a whole nother issue with our Department of Health. But nonetheless, we got that positive result. Almost at the same time, a second deer died on a farm about 4 miles away, and that deer was also tested. So in the course of about a week, we got 2 positive detections on farms in New York State where we'd not had anything. So both of those herds were depopulated, and we detected a few more deer there, and Justin may talk about some of that effort as he gets up here afterwards. And then I'm just going to talk for a few minutes about what we did in April of that year, and I have to say inside of 2 weeks we're on the ground collecting deer out of a zone. So we'll talk about the Oneida operation. And this is where the cooperation comes in. Within our state agencies, obviously the Department of Environmental Conservation, with whom I work, our Department of Agriculture and Markets, and the Department of Health were there. USDA Veterinary Services provided a lot of insight and input to us, and of course Wildlife Services that Justin worked with, worked hand in hand with us in the collection and also in depopulating those captive herds. Can't probably go much further without talking about Cornell University. At that time, we didn't have our wildlife health program fully up and running. Now we do, but nonetheless, we had a good relationship with Cornell and the diagnostic lab, the cooperative extension folks. And a little bit later, the Human Dimensions Research Unit did a whole lot of human inquiry on the response of, perceptions of hunters, perceptions of local residents, and perceptions of agency staff on CWD and how we did and what they felt the risks and concerns were going forward. And then we got some help after that also from SUNY ESF, some graduate work looking at the risk of spread and interactions between deer, not only in the containment area, but in other areas. So we've got some predictive models that help us anticipate where deer might move on the landscape, and we can use those now. So just really briefly, we initiated incident command structure in order to do the collection in 2005. I'm not going to necessarily recommend that you have to do that if you get a detection in your state. I'm not going to say not to either. Incident command can sometimes take on a life of its own. I think we probably could have done probably as well in New York without that structure, and maybe even a little bit better, but nonetheless, ICS does bring a lot of resources, it brings a lot of structure, and in our case, I can't say that it didn't work, so I won't say too much more about that, but maybe over a cocktail if you want some of my concerns about it, we'll talk about it later. So initially, we established a 10-mile perimeter around those 2 index herds, and you can see those 2 dots on the map is where they were in the town of Westmoreland. The blue circle is 5 miles, and the red circle is 10 miles out there. And the idea was to collect about 400 deer off of that landscape for testing. Again, at this point, it's not a containment area. It's a sampling zone, if you will. And really, the operation was not to depopulate the wild herd. It was really to test for presence and prevalence of CWD on the landscape. First step, know what you've got, and then make your plans from there. I won't talk too much about some of this stuff because Justin will probably cover some of the shooting activities and some of the actual in-the-field stuff, particularly the night shooting that USDA took the lead on for us. Our agency decided to handicap our staff in collections and sort of said, well, you can go out with 1 gun, 1 bullet, and only go out for a few minutes at a time. USDA, our agency, said, oh, you guys are professionals. You can go do whatever you feel you need to do, and they were a lot more effective than we were too, so it's good to have them there. We also made use of a lot of other parts of our agency to assist in that, so when you had shooting teams out there, you shoot the deer, you could actually call them in, and our operations staff would come up and collect the deer and move it so that shooting staff could stay focused on shooting, and other folks could come and whisk them away. We had access crews, and that's important because this is mostly private land. We used access crews to literally go knock on doors. We GIS capabilities in these days, picked out parcels of land that might be suitable for taking deer, and then because we can't trespass on private property, went out and actually knocked on doors and got permission. And those access crews actually took a form with them where the owners could say, you can go into this field, I don't want you driving through the fence, notify me when you come, do this, do that, whatever, and it was handed to the shooting crews so that they could comply with landowner wishes. And it kept goodwill up. And then we had roadkill collection crews, and we were actually picking up every roadkill deer in that area. We had the New York State Thruway in there, so we actually had a pretty ready source of dead deer. One thing I just want to touch briefly on, that is actually me about 15 years ago or 14 years ago, a little bit younger and with a little bit more hair. This actually is from one of the meetings. We held some public meetings in the area, did a lot of press. I might add that my role in those meetings was to do sort of the technical presentation on CWD, and I'm not even going to cover that because it's been well covered today. But we also had our director of our Division of Fish and Wildlife there to talk about what our plans with respect to the wild herd was. And we had the director of the animal industries for our Department of Ag and Markets in the room. So really put on a good united front, talking to the public that we were working together and trying to figure out what was going on. Might add that Ag and Markets was doing all the trace outs, trace backs, trying to figure out where deer had come and gone from, and really put a lot of effort into the captive side of that equation. So when it was all said and done, we collected a fair number of deer. We didn't quite get to the 400, but we did detect 2 positive deer. Those 2 positive deer were, I guess I'm in Canada, so I'll say roughly 1 kilometer apart from each other out of that big circle. So literally on the same farm. We did test them for relatedness, and they were not. And we tested a lot of deer where we didn't get a positive result. But still, 2 probably suggested we had it on the ground and a somewhat low infection rate. That's April. So after that, we amended our CWD regulation. We went to establish a containment area, and I'll show a picture of that in just a second. We required a mandatory deer check and submission of sample in that containment area, and that was within a day. We also had a number of deer processors in that area, so we worked with them so that they could be alternate sites for people to take their deer to, and they would remove the head and save it for us so that we could pick it up. So we actually had multiple sites where people could drop them off. And we would collect the tissues in a check station, that very nice check station we got built because of CWD. So that's one, okay, that's the only benefit that came out of it. We got a nice building out of it. Also within the containment area, we prohibited the possession of roadkill deer, we prohibited the collection and sale of deer, elk, or urine, and we banned rehabilitation of white-tailed deer in that containment area. And just for a pictorial, what we did was, and you can do this any way you want, you can define it by roads, you can define it any way that's practical for people to understand where your area is. What we did is any place that 10-mile circle, any town that the 10-mile circle touched became a town within the containment area. And in some cases, that meant, because they're pretty large townships, that the containment area extended 20, 25 miles away from the index farm. So in 2005, just to recap, there's the sample collection locations in those green dots superimposed over the boundary of the containment area. We pulled in 1,610 deer. 1,400 of those were hunter-killed. We probably had very good compliance that year, probably 80 or 90%. And one of the things we did, somebody talked about CWD testing being fairly quickly. This was a ELISA test, but we were guaranteeing results for containment area deer within 7 days so that people could kind of get on and understand what, if anything, they needed to be concerned about with their deer. We also ramped up very significantly our statewide sampling, trying to get at least 5,000 additional deer from around the state, and we did exceed that. So we really put an effort into understanding what we had in New York. People have talked about weighted surveillance. I threw this slide in here because since about 2006, 2007, 2008, somewhere in there, we decided to go from just some kind of a numeric sample to something weighted, and that kind of came along with Kristen Schuler at Cornell in our wildlife health program. So we do much like a lot of these other Manitoba and Ontario have spoken about, and I think somebody else earlier did. We've analyzed where we've got deer farms, where we've got taxidermists, where we've got other entities like that. And of course, as you can see, there's quite a bit of weight to the Pennsylvania border right now because, as we all know, Pennsylvania's got CWD, so all of our border counties have an enhanced risk here. And just graphically, this kind of shows how our sampling over time's been. We've tested over 52,000 deer now in New York since we began. Obviously, the spike in 2005 and for a few years thereafter when we were testing a lot, and then we went to that weighted surveillance. But we're still testing about 2,000 to 2,500 deer a year based on that program. So many of you may not be aware of this, but in 2018, early November of 2018, we got a false alarm in Chautauqua County, which is not all that far from here in western New York along the Pennsylvania border. We had a presumptive positive tested at Cornell by ELISA test, and that pretty much got a whole lot of things happening on the 8th of November last year. It was about a week away from the opening of our regular big game season, and our response plan would suggest that if you're very close to a season, we will use hunter-killed samples instead of going out and doing a collection because there's going to be a lot of deer available. But we're going to have to put a regulation in place, a mandatory check, and a whole bunch of other things. It was a scramble for a few days. We got the sample out to the national lab. They came back and said, boy, it didn't come back positive, but we want to be sure. We want to run another test just to be certain. So we had to wait a few days because we had a holiday in there. So we started to do a number of initial things to set the wheels in motion for what we were certain was going to be a CWD response. And why were we certain? We're right on the Pennsylvania border. This is just an example of known escapes from Pennsylvania deer farms over about a 10-year period. I didn't put the movement of captive cervids in Pennsylvania on there because the lines are so heavy that you don't see the outline of the state. So that caused us to dust off our plan, and I want to talk about plans just really briefly. Our plan is not that long. I didn't pick it up before I came here, but it's maybe 15, 17 pages long. It's not that long or detailed, but it does spell out at a high level what our strategy might be at certain times of the year, what happens in, as somebody else mentioned, if it's a detection in a captive facility versus wild. It speaks to the establishment of a sampling area, and it speaks to a whole bunch of other things that have to fall into place: communication, cooperation with the other agencies, and how we're going to go forward with captive and wild efforts going forward. So by the 13th, 5 days later, we're still making plans to do this because we don't have that final result yet. And we're working on building a containment area, getting all of those things in place. Now, mind you, a lot of people had to drop whatever they were doing and pick this up to try to get that response in place for when we could have access to deer. And that includes a lot of people in our central office and pretty much all of the wildlife staff in our Region 9 office that works out of Buffalo. And there's a lot of little logistical things that go on here, and here's one of the take-home messages that I think I'd like to share with you. If you get a detection, if you never have one, you got plans in place. It speaks to a lot of things that you're going to do. But in this case, the little red star there is in Keantone is where the detection came from. We started looking at things. We drew a circle around it for a sampling area and said, okay, we don't have a deer cutter anywhere near there. Maybe we ought to expand that out a little bit, that sampling area. So we went out a little further and we picked up one. Well, Chautauqua Lake's in the way, so probably not too much of an effort there, up there. We've got a major highway that's probably at least a semi-permeable barrier. And we got Pennsylvania where we might have CWD to the south. So what we ultimately decided to do is expand what would be our sampling zone to include a couple more meat cutters as alternatives. I guess, well, and also some taxidermists in that one. I won't go back to that. So just those logistical things that you're not thinking of in advance, you might want to be thinking about if you have to implement a control. One of the things we had to our advantage, because we'd done some upfront work, we knew where our meat cutters and taxidermists were. So out of that, we developed just a list of things we could have done in that particular 5 or 6 day exercise, maybe a little bit better and upfront, so that we could get past some of those logistical challenges. I just want to cover a few some recommendations that I'm going to throw out. Some of them, I guess, be directed probably at agency people, and then some of them perhaps at you folks. the hunters out there and how you can help. One thing I just want to say is, in our case, we've got excellent interagency cooperation. New York State Department of Agriculture and Markets and DEC have sort of joint responsibility over captive cervid facilities, so we have to work together. And while perhaps the DEC biologists would prefer that there not be cervid farms in the state, that's part of our reality, so you work with them and you do the best you can to come up with other means to minimize your risk. And other folks today have talked about that. We've got bans on import of cervids into the state. We just simply don't allow that on the farms, live cervids. And we've got very, very strong cooperation with them. They pay good attention to the farms. We're starting to do joint inspections with the ag and market staff now to the cervid facilities, in part to get to know them better, but to understand them and understand their strengths and weaknesses. So there are things you can do even in the absence of getting rid of something. You can minimize risk by thinking through it and working together. Have your plans and use them. Communicate frequently about CWD. The booklet you see there on the screen is something we got out this year with our risk minimization plan. It talks about response, surveillance, and risk minimization, and kind of covers the whole gamut. And it's been pretty popular. We've given out literally thousands of those. And so it's out there, and we're trying to, and it's got some recommendations for hunters. And last, and someone else said this, I think Ontario did a tabletop exercise. Do it, because you'll find out pretty quickly there's a lot of things, a lot of logistical things that you haven't thought about that become necessary. You know, in our case, everything, you know, that goes out to the public's got to go through a press office, all sorts of connections that need to be made, and you need to work very, very rapidly. In our case, we've got a governor that's interested, wants to know what we're going to be talking about, what we're going to be saying to who and when. So you have to work through all of those details, and if you got those things tacked down, you can probably mobilize response literally within a few days if that's what you need to do. Some recommendations for hunters. Use credible sources of information. Obviously, you're getting some of that here today. I think anyone that's paying attention on the national scene, blogs, some of the writers, some folks that don't want to take CWD seriously, there's a lot of bad information out there. Take some of that information for what it's worth and really try to work through the details and maybe listen to the folks that know something, dismiss some of the other. And you folks have been here. Share this information with other folks. Support your wildlife agency. It's real easy to criticize a wildlife agency for doing things wrong, and sometimes we do make mistakes, but I'll guarantee you there is nothing to be gained to me or any of the staff that work with me in doing a poor job at this. There's absolutely nothing to be gained. Other than a nice check station, we don't get anything. And it's not even job security. It's job shift, because we're not going to hire new people. We're just going to have them do other stuff and probably not be doing other important wildlife work. It's a tremendous strain on agency resources, and obviously as a hunter, if you want fresh, good, wholesome, healthy meat to feed your family, now you've got that whole concern to work through. So support the agency, support the rules they put in place. They're designed to minimize risk and hopefully keep your herd safe for you. So follow all those rules. And one thing I don't think I can stress enough is, you know, where you can, minimize risk by proper disposal of those parts and best practices where you can. So we generally recommend in New York, where you can dispose of in the waste stream, you can get that stuff out of the way and get it off the landscape in case. Because we're all in this boat together. And I'll thank you, and I'll turn it over to Justin. And in the spirit of cooperation, we are cooperating, but we did put together two separate slideshows.
All right, there we go. Oh, thank you for inviting me to come up here and speak today. I'm with USDA Wildlife Services, and our niche, what my normal job is, our niche is to protect the public, natural resources, agriculture, you know, really from damage, wildlife conflicts. So that, by doing that, we're very well suited for something like this CWD and rapid response. Because we're out there, and that is our job, is to, catch or capture, sample wildlife pretty much every day. But we also weren't really prepared for this ask when we were asked to mobilize in this quick of a fashion. So, we actually were asked to do a rapid response twice back in 2005. Because first, our New York State Ag and Markets called us to do the depopulation of the captive cervid herds and wanted us there in 24 hours. So, today that's something to think about, is what you need to do as an agency or as a group to make sure that when it does happen and you have your plan in place, are you really ready to respond? So we were asked to get there within 24 hours because they were highly concerned that there's poor fences. This is only a small site, but there's 20 deer there, and are they going to get out? At that time, I believe Veterinary Services or the state, one of the two, paid indemnity for that, and that was part of the deal. If the indemnity wasn't paid, then they wanted us to come in and depopulate that herd and stop this as quick as possible. So, relatively straightforward, but just something to try to think about. Are you truly ready to respond to that call? Do you have that in place? People or contractor ready to go? Then for us, the second was responding to the sampling of free-ranging white-tailed deer. As Jim mentioned, there was no positives found in the wild yet, so this is strictly a sampling effort. And they wanted us, we really had 3 days from the time of the phone call to get ready. At that time, I was actually a wildlife disease biologist, and in order to get there and get ready, we had to bring a person in from Maryland. We didn't have enough people on staff at that time to handle this, so it's a lot of stuff. We had 3 days to prepare for this event, which ran from April 11th to the 29th. We were working the night shift. As he mentioned, DEC was doing dawn and dusk. This was under the ICS coordination, and I guess I can say a little more, is that was a real pain in the butt for the guys working the night shift, because every day I had to go to a meeting at, I forget, 1 o'clock in the afternoon. So a typical day, just so you guys understand, doing a sampling effort at night on this isn't, for us, was we would usually get up and start our day about noon. Go through all your equipment for the night, gear everything back up, clean out the truck, clean the coffee cups out of the, get them out of the way for the next day, pile in some food for the night, and go to our mandatory meeting at 1 o'clock or whenever it was. But it had to be at that time so they could get the people out for the dusk effort, which we also did. But then we would go out to the farms that the crews lined up and look at them ourselves, because you can't do any work at night without seeing it in the daylight. So we basically worked from noon till about 3 in the morning or so every day. This is a sampling effort when we couldn't use hunters to do the work, right? This was because it's in April. But another thing about using an agency like us is that you can do this in most situations, even places the hunters can't go. That is a benefit for the future, right? Urban, suburban areas is fine. That's kind of work we're doing all the time anyway, so we're used to working in those environments. You get your samples right from the target area where you want. So after we did find our first, positive in the wild, we then tried to focus. They may have drawn other lines, I don't know, but I just know the team in that zone said, let's concentrate right in that area. So you don't get that with hunter harvest, right? But the prep time is high, and the cost is high. The one benefit we had through us is we had trained biologists out there conducting the work, people that did this all the time. To give you an example, we currently have about 35 or 40 employees in our state. Only 6 are trained to do this work. So we don't just let everybody do this because you're using high-powered rifles in the middle of a city or urban area. We want to make sure it's at night. So have specialized equipment. So FLIRs are much more readily available today, but a good FLIR costs you $10,000. So if you want to be able to see really well at night, that's the kind of thing that helps. Maximize the most samples for your money. Using suppressed firearms, being able to shoot from vehicles. If there's barriers in your way, that's something to think about now too, to add to your plan or to your regulations, so those special regs can be put in place. You really need, if you want to be a fast, effective, and get this over with, you need to have those barriers removed. And so a team for us we didn't even, in 2005, we weren't really even afforded a full team because we just couldn't, the money wasn't there to do it. But a normal team would be a 3-person team. We only ran a 2-person team. You have a driver, a person running the FLIR, and then a shooter in the back of the truck. And everything is done from the vehicle. It's very safe, and it's very effective, and it's very humane. We always strive to meet our AVMA, American Veterinary Medical Association's guidelines for all removals. So quick, just some stats. Right, in 2005, we worked 17 days. We had 4 people there. Those 4 people worked 1,100 hours, so basically 16-hour days. The whole project collected 292 deer. Of those 292, the nighttime operations took 198. So that kind of shows where you want to focus your efforts, at least in the habitat that we had. Agricultural, very wooded, and that's where we did get 2 of the tested positive. So we took the map that was handed to us on day 1 that Jim and those guys had drawn. Since we knew we only had 4 people, we just did a strict, here's your zone. Everybody took their one zone and learned it. I had zone B. So by the time 17 days was over with, I knew every road in that zone pretty darn well. We're handed a stack of papers from the crew that was out there knocking on doors. Every afternoon, I would drive to my zone and go talk to the landowners, see where it was safe to shoot on their property, make sure it was clear. I wanted to double check if that landowner said you can only shoot 2 deer, make sure that was the case. The last thing you want to do is agitate the public at this very stressful time. So some of the recommendations I can give you guys for this going forward, is, like I said, if it's a similar situation, is focus your resources on the night operations. Look to adding, new technology some now we're using in certain places interactive GIS property maps. So instead of being handed a piece of paper with, this is your property you can work on, it's right in the truck on the computer screen. You can see it, you can tap on it. Kind of the same with our mobile app. We've now developed a mobile app. They just wouldn't let me stick any pictures of it up yet. The government doesn't want the resource out. But it's that simple. It just puts a dot on the map. So instead of us have to try to get a person come in to call in to come pick up the deer, they could just look at their phone and see the dot we put on it. Use some of that. We've also developed our own FLIR on the rooftops now that helps us spot wildlife, and it's got lights and laser designators and stuff like that. Clearly define your goals. I think that's been said a lot. Use your plan. For us, the land access was key. We needed to have access to private property. Think about that ahead of time. You probably need as many or more resources to gain that property than you do actually to remove your samples. And that, from my standpoint, where I sat during this whole 2005 incident, that was the biggest thing. We lacked a lot of resources on the night crew. So a lot of times I would sit there and wait half hour, 45 minutes for the crew to come pick up the deer. So that's time I can't go remove more. That was the bottleneck. in the situation. And there was just no way at that time to remedy it. But that's lessons learned. Hopefully going forward, we could fix that if we had to do something like that again. The support personnel just weren't allocated to the right spots. And that was my overview.
Can you hear me okay? Yep, sounds like it's on. I've heard a lot about testing. Brian Fitzsimmons, by the way, from London. Heard a lot about testing from different speakers at the conference so far. And I got some questions in regards to it. You're saying that the entire head has to be left for testing. Originally, I thought you needed samples of the brain or something, but I'm curious as to why you'd need an entire head. As far as your testing goes. I'll just put these all out here for you. I'm curious about the cost of testing. Is it very expensive to test for the CWD? How long does it take for results? And I've heard, I think, the first gentleman there saying you were getting results in a week. Is that normal? I'm thinking as a hunter down the road how long I'd have to wait. And then how complicated is the testing? Is it easy to set up a lab somewhere where the testing can be done? Or do samples are there only a few places like Cornell University, for example, all the testing has to be sent down there? Does it need a really complicated lab? And that was it. So, sorry, a bunch of questions all in a clump.
I think I can hit a few of those. No, you don't need the head. I mean, basically, at that time, in 2005, we collected the obex, which is a piece of the brain stem, the lymph nodes, and the tonsils for those deer. Now the testing we're doing is just with lymph nodes. The thing is, with most people who are harvesting a deer, they're not, particularly a doe deer, they're not keeping the head anyway. So when it goes to a meat processor, they're removing the head, they're removing the hide, they're cutting up the deer, the people pick up the boned out meat. So that head was going to go into a discard pile anyway. So we would get the whole head and then we would extract the sample from it. So it was just easy to do that. We did have to make provisions for the nice bucks, the taxidermy mounts. We had a couple of taxidermists in the area who agreed to take those heads in, cape that out and take it completely off the skull and then provide that skull with the parts we needed in it. And then that piece of cape and the antlers could either be mounted at that facility or taken out of the containment area. So we don't need the whole head, it was kind of convenient. Testing, I'm not the expert on that, but all of the labs that do the testing that are being accepted have to be accredited and accepted by the National Veterinary Services Laboratory in Ames, Iowa. Cornell is one of those facilities, and Brian Richards probably know better than I do, but there's probably, 20 other, some odd labs around the country that do that and are accredited to do it. Thing with testing is difficult. In our case, in the containment area, we made a commitment that we're going to get those samples tested and results back within 7 days so that hunters could move on. Elsewhere in the state, that priority was not put on them, and it was 3-4 weeks. And I think it really depends on lab capacity and how quickly they want to move them. But we had Cornell very interested in taking care of those and putting a priority on our containment area samples. At that time, it was probably in the $30 range. I think generally now they're saying about $60 probably a sample. Probably varies a little bit.
Thanks, Jim and Justin, for coming up and giving us that presentation. Paul Mussel, question is, what cost, can you put a dollar when this started and your teamwork and all the other staff that was kind of thrown in? Any ballpark idea what it would cost your state to do what you did?
Just keep in mind we're government, okay? I'm not sure we ever completely figured it out, but the number that we used after the 3.5 weeks that we were on the ground there was over $1 million was spent. And think about it, you've got in rough numbers about 80 to 100 people working 12, 14-hour days. So there's overtime, there's all the other stuff. We're putting people up in hotels, we're buying them dinners and meals and all of those other expenses. So it was not cheap. In our case, I think we'd argue indeed it was.
Okay. Before I introduce our next speaker, just a couple quick announcements while I have a captive audience. When we adjourn, after Dr. David Westaway's talk, John Ward from Camillus Knives will be doing a knife sharpening demo and a Q&A right outside the doors there, right beside Steve Elmy's rack stacker display. And a quick note about tomorrow's agenda. The 9 a.m. speaker, Dr. Vic Adamowicz, was unfortunately unable to attend due to the grounding of those Boeing 737 planes. So we have that free spot. We are now going to start the agenda at 8:45 tomorrow morning rather than 8 o'clock, so you get to sleep in a little bit. Breakfast will start at 7 a.m. So food at 7, festivities begin at 8:45. Okay? Dr. David Westaway is a molecular biologist with a special interest in neurological disorders. He earned a degree in biochemistry from the University of Sussex and a PhD in biochemistry at the University of London. He completed postdoctoral training at the University of California and is the current director of the Center for Prions and Protein Folding Diseases at the University of Alberta. The goal of his current research is to understand the common forms of dementia. Dr. Westaway's research uses molecular techniques to address shortcomings in our understanding of these diseases. Please welcome Dr. David Westaway.
Good afternoon. So I'd like to thank the organizers for bringing me to this extremely well organized meeting, and I also am appreciative of the title of the meeting, A Conversation About CWD. Now, I actually used to live down the road in Etobicoke, so I got quite misty-eyed seeing the traffic on the 401 and also realizing its potential for generating a roadkill for testing. But anyway, I want to share some stories with you. Primarily, I think the way we're thinking about this disease and to emphasize the important and sustained investment in Alberta to tackle prion diseases. And I also realize that I'm the last person for the afternoon, and everyone's blood sugar level is dropping, and so I have one story I want to share with you that's a little bit of a trial balloon. It's maybe a narrative that's about 180 degrees opposite to narratives you're typically used to hearing. It may be a bit mind-blowing, but maybe it'll also keep you awake, I hope. So let's go. So we have some questions. We're always generally concerned about prion diseases. They affect humans as well as animals. And one issue is, how can this funny little molecule cause different types of diseases? They haven't been touched on so much today, but there are sporadic prion diseases that are not initiated by a known infection. We're interested in the way the molecule goes wrong. And of course, we're very much interested in how we deal with these diseases and ultimately stop them. Now, the talk is broken up into 4 topics, basically. Here they are. A little bit more about prion replication, some new thoughts on vaccines, some stories, some understanding we can gain from natural prion diseases and their pattern of inheritance, their genetics. And lastly, I'll come to a genetic approach to CWD. And for each of these topics, these are the sort of how we'll open them up and explore them a little bit more. Now, first of all, let's start off with prion and prion replication and any the problems presented by prions, but maybe also some frailties where they might be susceptible to being stopped. Now, the key, you've heard a little bit about this. These diseases are caused by proteins, and the key parameter, the key thing that goes wrong, is to do with shape. Cells in our body carry out a number of activities, and proteins in our cells are sort of like the verb in a sentence. Without a protein, nothing happens. But the way that these proteins carry out their activity is intimately tied up with how they look like in three-dimensional space. We had this version of this schematic earlier. The basic issue in prion disease is you have a normal protein that we all make. We make it every day. We probably make hundreds of millions of copies, and then every night we degrade it. It's labile, but when things go wrong, there's another form of this protein called the scrapie form, named after scrapie disease, that causes all the problems. But really there is the essence of how the disease amplifies is a conversation between a molecule in one shape, a molecule in a different shape, and the bad actor molecule convinces the normal protein to flip over, and then you've got two copies of the bad protein instead of one. So I'd again underscore that prions are not some cryptic bacterium that we failed to discover in the past 30 or 40 years. They are prions, they're not bacteria, they're not viruses. Now, one way to conceptualize these molecules and these diseases is to have a schematic or a model, and without getting overtly high-tech, you can do something like this in your kitchen. If you want to build your own model of a cellular prion protein, this is roughly what it looks like. So we have a front region that is floppy. We have a switch region, which is some of these mutations you heard about earlier map into that region, seems to control decisions, and then you have this folded up part of the molecule at the back. Now, if we revert back to scientific diagrams, these molecules, if you stretch them out, just in like thus-wise, they're not all identical. So basically, prion diseases you can classify as to whether the back of the molecule gets into the wrong shape. And here are some diseases. So fatal familial insomnia, Creutzfeldt-Jakob disease, of course, chronic wasting disease, scrapie. And if you look at the front, these are not exactly the same length, and that is intentional. So the bad form of the molecule comes in slightly different flavors that we would call strains. We can do this length measuring exercise to distinguish them, but there are other criteria as well. For example, the pattern of pathology in the brain will shift as well. There are other versions of prion disease where the middle of the molecule and the front of the molecule can go wrong as well. So most of what I'm going to say is going to apply to the diseases where the back of the molecule goes wrong. And so we are quite interested in this switch region here. It can interact with other proteins. It probably interacts with other components of the cell. And if one is lucky, all the interactions are positive, fruitful, and the person or the animal stays healthy. But if exposed to infectious prions, an animal would end up with CWD. Roughly, the human equivalent would be CJD. I'm not saying that CWD causes CJD, but that's in terms of the length of the molecule, it'd be similar. And if the switch region goes wrong, then you get a disease called NOR98 in sheep, or Gerstmann-Streuer syndrome in people. So that is a whirlwind tour through the molecular actors in this story. So I'll share with you a few things about vaccines. So If we want to deal with these diseases, we can go back to the drawing board and consider what we can interrupt, what we can block to stop things happening. And roughly speaking, that we have different types of targets. They're in the left-hand column. So we can go after the protein that's in the wrong shape, or we can go after the gene that makes the protein. So on this list, the top approach is to make antibody against the bad form of the protein. This is fairly standard, and that would be a sort of vaccine approach. That is an area of very active investigation, and I'll show you one example. Where do the pitfalls lie? Well, because this PRP-scrapey molecule comes in different, slightly different flavors, if you are unlucky, the antibody will inactivate most of them, but if a different flavor doesn't match with it, then that flavor will escape and the agent will replicate. And this is a phenomenon that can be called strain evasion. Now, we can think of small molecules like aspirin or something that we give to the person or the animal, and that would also interact with the bad form of the protein and stop it doing what it's meant to do. And that approach would also have the same potential pitfall of strain evasion. You might stop most of the molecules replicating, but if one of them is not quite the right shape to match, then you're not going to stop that particular one. We have an interest in this middle part of the molecule called the switch region, and we are trying to decode that, as it were, to figure out the Rosetta Stone of how that works. We don't quite know how it works now, so I have listed it as being a little bit theoretical, but that would be a possibility for the future. Now, if we focus on the gene that makes cellular PRP, you need cellular PRP to make the bad form of PRP, then if you got rid of that, which you can do in mice, and the mice are pretty much fine, and goats, some goats naturally don't have it, they behave, as far as we know, in a fairly normal manner. The advantage of that is that if you don't have any precursor, if you don't have any fuel to start the fire, then nothing can happen. If there's 5 different strains, it'll block all of them. So that has some advantages, and there are versions of the prion gene that naturally confer resistance, partial resistance, or in one case, apparently total resistance. There is a logistic problem of when breeding animals that the genes get diluted out with each generation. I'll talk about that in a little while. And lastly, there is an alternative strategy that deals with genes not getting diluted out with each generation, and that is a little bit more near the cutting edge of research, and that is something called gene drive. It's a trick, and there's also a reverse trick called gene break to turn it off later on. So I'll try and explain that to you just in a few moments. Now, so in the Prion Center at the University of Alberta in Edmonton, we have material from CWD animals and can purify it, and these are pictures from my colleague's lab and of how some of the infectious material looks like under the electron microscope. There's these fibrils you could see here, and then you can, by using some image processing, you can work out what their cross-section looks like. So gradually we can learn more about how the molecules are folded up from these types of analyses. But the main thing I'd like to share now is to use our knowledge of the structure of infectious prions to guide the therapy. So a few years back, using prion isolate that's propagated actually in mice, there is a structure of how the molecule is actually folded up in the bad form. The normal form we've known about since 1996, but for technical reason, the bad form is very it's rather insoluble. It's difficult to work with in a lab, so a special technique had to be used to do it. But what you see is that it's wound up in a sort of a ladder-like arrangement. Now, interestingly enough, we are, of course, familiar with and worried about prions that replicate in mammals, but there are also benign prions that live in other parts of the animal kingdom, and one lives in a fungus. And it doesn't do anything to people or animals, it's just a biological phenomenon where the fungus can make a switch in its lifestyle. But anyway, the point is that the exact way the atoms in this molecule are folded up was worked out for that one, and they're shown in this schematic here, this other bad molecule, well, not a bad molecule, but other type of infectious protein from the fungus is called Het-S. Now, when you sort of zoom in, there's a sort of ladder-like arrangement they call a beta-solenoid, and each section has 4 rungs of the ladder. That's one molecule. Then there's another one here, and another one here, and another one here. Now, when our colleagues went to look at this other molecule from a fungus, it actually comes in units of 2, not 4, so it's 2 rungs of the ladder. But the structures, this is a very high, very precise model over here. This is not quite as resolved, but you could tell that the structure is rather similar. Anyway, this led to something that we call the light bulb idea, that actually we could use this innocuous fungal prion to help us in our search for antibodies against the normal bad prion that affects mammals that we're, of course, extremely worried about. So I can see that my slide is looking a bit, starting to get techie looking, so I'll go through it a bit quickly. But basically, these triangles are the rungs in the ladder, and just as you may know that the DNA code is made up of 4 letters, well, the protein code is made up of 20 letters, and that's what these letters are around the outside. Anyway, in the ladder, certain parts of the molecule are lined up nicely. I'll show you a picture in a second. In the regular cellular PRP, we can look at this green bit and this turquoise bit. They're sort of in different places. They're not next to each other. But you can do some, what we would call protein engineering, to put these molecules here onto the fungal prion. You put them on the outside so the antibodies can recognize them, they're accessible, and then you use this type of molecule to, immunize mice, and then the mice will raise antibodies against this molecule that we call a molecular mimic. It is not itself infectious, but its shape looks like the real thing. And we heard about ELISAs for testing. Often ELISAs use an antibody. If you get a good reaction, it makes color or less color. So you just measure the color, which is often a yellow in these ELISAs, and you see how much color there is. That's what these pictures are. And this just shows if you immunize mice with this synthetic molecule, they start to generate antibodies. In fact, there are quite a lot of antibodies because you can dilute them out a whole long way and they still give reactivity. And I'm not going to go through the second schematic as what we would call a negative control. In any event, so now there is this candidate, what we call a monoclonal antibody against the shape of the infectious protein, and that has been tested on a number of different types of prion disease. Again, the more bigger the bar on this axis, the more color, there's more antibody binding. So these look quite promising. And over here are samples that all the samples that should not react with this special antibody. So these are experiments done by my colleagues, Andrew Fang, Lily Tang, Holger Villa, are steps on the road to getting a useful PRP-scrapie specific antibody. Of course, it could be a new diagnostic, but one would hope that by sticking an antibody on the bad infectious protein, you'll stop it replicating. This is one particular strategy, but there's also another strategy underway at the University of Alberta, and our colleagues in Calgary have yet a different strategy. So we hope by the end of the day, with these strategies, we will have different approaches to get antibodies against these molecules and thereby generate smart vaccines. Now I'm going to change gears here and just start into the genetics and draw upon a couple of examples that we have known about down through the years. And one of them is scrapie in sheep. We've heard that mentioned earlier today. And the other one is this rather strange and macabre human disease called kuru that was propagated by ritual cannibalism. Now, work on prion diseases, they didn't used to be called prion diseases, they had all sorts of names, but it started after the Second World War, and there were two schools of thought as to what a prion disease was. So one school of thought was it was, in fact, a purely genetic disease. It was a regular recessive disorder, and that school of thought was propagated by a guy called James Parry. And the other school of thought was in some ways more sophisticated, and that was there was an infectious agent that would affect the animals, in this case the sheep, but depending on the genotype of the sheep, they would be more or less susceptible. And the second point of view is much closer to what we think of today. So here is a scrapie of sheep, and it is a transmissible disease. So this is, of course, sounding rather like CWD. It's not quite as contagious as CWD, but it has some contagious aspects. And you can have sheep that might, through fluids or whatever, they might spread from one animal to another. You can have sheep in a pasture, remove them, bring in sheep that should not get scrapie, and they will get scrapie. So there's an implication the environment is contaminated. And then the last thing that remained to be done was it's a brain disease. You make an extract of brain from an affected animal, you inject it into an unaffected animal, and the animal gets sick. So that proves there's a transmissible agent, and that was the basis of how people study prion diseases in the lab. Now that's all well and good, but in fact, with some backward looking, it became clear that the other school of thought was not totally wrong. So the idea was that there was a recessive gene controlling appearance of this disease, and this was called the S gene, and then it doesn't cause the disease to appear spontaneously. There are mutations in humans that do that, but it is a susceptibility gene. But the point of this slide is really to give you an idea of the timeline, to go from the beginnings of understanding to getting effective outcome measures. So the S gene concept was published in two papers in Nature in 1960 and 1962. There was a gap of about 20 years or so where molecular biology had to be invented, and then we learned how to identify the PRP gene. People then looked at the PRP gene in sheep and discovered a small change that was present, comparatively common. And then it became clear that this small change in the gene was, in fact, a recessive susceptibility gene. So now we've got as far as here. Then you have to check it by some genetic experiments. Now we know what the protein looks like. It got mapped onto where it is on the back end of the protein. In here, there was some selective breeding happening in black-faced sheep in the States, and it is moving closer to eradication in that particular instance of domesticated prion disease in domesticated livestock. But so here we go from 1960 to close on 2020. So you should be getting an idea that these things are sometimes not so fast. Of course, we hope to go faster because somewhere in here, the whole of the techniques of molecular biology, they weren't invented yet. But once they're invented, the pace picked up. Now, the other issue or story about genetic modulation of prion disease comes from Kuru, and I've told you this is this rather strange disease in Papua New Guinea caused by ritualistic cannibalism, and that was a transmissible prion disease, transmission by the oral route, and that has examples where there's 30 years between a person being at one of these so-called mortuary feasts and them getting disease. So these diseases can be extremely slow, but there was also a sort of follow-up story to this earlier story that won Carlton Gajicek a Nobel Prize. So the follow-up story is so this is where the disease was occurring in New Guinea. It happened in the Southern Highlands in the so-called Foray region. And then many decades later, people went back to look at the genetic composition of the people living there, and they realized that right in the epicenter, that people had there'd been what's called selection pressure. There was a spontaneous resistance version of the prion gene, and it's very subtle, but it also seems to be extremely effective. Here's the 20-letter code. I mean, obviously it's not even the whole of the protein. The print is rather small. Anyway, the point is that one bead on a string in this code change produced total resistance to prion infection. And this has been checked by taking that same change, chopping it out, moving it into animals, and validating it. So that is quite an astonishing story. So if you have a really endemic situation, eventually there's a chance, not that the animal will know to have the right mutation. The mutations will just happen spontaneously, but the animal will be very much protected now. This is an example of a natural resistance allele. Maybe there'll be some for CWD, but also maybe we might want to think about speeding up that process. Which brings me to the last bit. So we've all heard about sexually transmitted diseases, and I'm going to try to explain to you about sexually transmitted prophylaxis, which probably sounds a bit mysterious, but hopefully it won't be mysterious in a few minutes. So the issue really here is to take aim at CWD with genetic approaches. Now, mammals, we all know about the Genome Project. Mammals have about 30,000 genes, and generally speaking, in prion disease, there's only one gene we're interested in. So there's one of those 30,000 genes. We know that there are natural variations, the prion gene, that cause resistance. I just told you about one. But when animals are breeding together, each generation, the gene repertoire gets diluted out by 50%. And I'll show you that in a little schematic right now. So this is where something more to say. So just a few words about genetic engineering. And some of these are a little bit tongue-in-cheek, but it's just to open up the conversation. So I have one here. I don't like genetically engineered organisms because I don't want to eat DNA. I mean, that's a bit glib and facetious. But there is instances when if one is driven to the wall, then one might need genetic engineering. Now, genetic engineering is not something invented by humans. It's been around for hundreds of millions of years. It's just that humans can use it for their own particular human purposes. And organisms encode their own genetic engineering tools to help them survive. In practice, a genetic engineering tool is normally a protein that messes around with DNA. So, this problem, so you have a resistance gene, we'd like to maybe use it in an endemic situation, but every time the animal with the resistance gene breeds, half of its DNA has been lost, half of its genes. So here's a schematic. We have 2 copies of every gene, and I've just colored them differently. Here's 2 copies in a male, 2 copies in a female, and the one to keep an eye on is this one in red. So here are 4 offspring, and you can see that only 2 out of 4 have got the red gene. And so each version we call, it has a genetic name, an allele. Now, the trick is to do something, so if I just go back, the last regular inheritance is called Mendelian inheritance. So, but what we want to obtain is something called super Mendelian inheritance, where here is a resistance gene, but with this yellow arrow, some DNA magic happens here, so we get 2 copies of the red gene, and then when the mating happens, that if you follow the red one, you'll see that all of the offspring have got the resistance gene. So this can speed things up. Now, I hasten to add that in this approach, we are seeking selectivity. We're interested in one gene, and we are scrupulous to avoid 29,999 other genes that we just don't want to go near. We just leave them as they are. So this is a concept, and it was figured out, first of all, from looking at simple organisms. There's always 2 copies of the gene. In this example, from simple organisms, you just have to look, there's 2 copies. One up here is gray, and the other one's different. That's a bit like me having red and white genes in the previous diagram. But this is a piece of selfish DNA. It copies itself into the other copy. At the top of the diagram, there's only 1 copy of this. Down the bottom, we got 2. So it's copied itself. And conceptually, what we'd like to do is to copy extra copies of prion genes that confer resistance. And in the media, you will have probably heard about this thing called CRISPR-Cas9. There was this thing about the engineered, and the story in China we don't want to go into. We're just worried about animals with prion disease. But anyway, it's a modular sort of biotech system. There's 2 bits to it. There's sort of green sofa-type shape thing here, and then you put in a piece of DNA to guide it, RNA, and then it will go in and do precise genetic engineering. So this looks a little complicated, but basically, let's start at the top, and then we'll skip the stuff in the middle, end up at the bottom. This animal would be capable of sustaining a prion infection because it's got a regular PRP gene. Once some engineering has been done, this PRP gene has been swapped out so it no longer works, but we've put in backwards some DNA tools that will knock out other genes when it finds them. It's sort of like a DNA cruise missile. That's the idea. So we're in the middle of testing this. I hasten to add we are practicing on mice, but the idea is to have animals where all the offspring in mice, they're called pups, where you have a male animal carrying this, and not just half of its offspring will carry the resistance allele, but all of them. So that's something, it's a little bit science fiction-y, but in practice, and there's some technical niceties about where you have to open up the PRP gene. I won't go into that. But we have done this in mice. We have made this special so-called CRISPR that goes in and inactivates the mouse gene in the inheritable cells of the animal. It's what's called the germline. And here are just some examples of what is happening. The longer-term project it's a bit like a construction project. You have to put everything together, like you have to put in the foundations and the framing and the plumbing and the electricity to make it all work, but we have a list of things we're doing to make this system work in mice to get to be ready. It's to be ready in case we need it in larger animals. And then there's also a thing called gene break that we're working on that can turn the whole system off if people are particularly worried about it. But anyway, this, I hope, gives you the flavor of the types of things we can do these days and might be pretty useful if we really get in a tight corner with a CWD epidemic. So I will briefly thank the members of my lab who carried out some of these experiments, and then I will just give you some conclusions. So through understanding what these molecules look like, the shape of the protein, the gene that makes the protein, we'll have options to deal with chronic wasting disease. Probably these types of technologies are going to take a while. It's not going to happen in 2 years, but also the time span of these diseases is much slower anyway. Thank you for your attention, and always open to new suggestions. So thank you very much.
Thank you. Thanks for a great talk. There's so many fascinating things in there, but I'll try to limit my question to the latter part where you're looking at using genetic components and CRISPR and so on. Mike Miller made some comments about the 225FF mule deer where they were breeding for homozygosity to try to figure out whether they could get immunity. And the practical applications of this, that particular genotype is really weakly conserved in nature. And his response was that they were not really all that successful. He says it's not that they didn't look like mule deer. That was followed up by Tracy Nichols, who said that her comment was that Mother Nature didn't like them, that they, for some reason, they were weakly conserved. So are there any parallels to trying to figure out, I think that there might be some really valuable insights with the kind of work that you're doing, because if we end up breeding or finding ways to get genetic immunity to CWD, what are the potential issues that we might be overlooking where for some other reason that there has to be a reason why this has been weakly conserved?
Yeah, I'm not quite sure which allele you're talking about to breed to homozygosity. Yes, but of course there's the PRP gene is there for a reason. If you put the animals are living in a very harsh environment, you might say a goat living on a farm without a PRP gene is not really being exposed to the environmental rigors of cervids out on the landscape. I mean, I get it, and the answer is you'll find out pretty quickly. The one way to do this is just not to take out the whole gene, but to use something that's already been tried in nature. And for example, I gave you the example of the natural resistance gene that emerged in New Guinea in a certain population of humans, where you can just take that very precise little bit that changes and drop it into a cervid PRP gene. The intention with that would still do whatever it's meant to do, and things would go on okay. But I think you'd find out pretty quickly if the animals really had some problem. Yep.
Yeah, thank you very much. I understood just about all of that.
Thank you. I feel great.
But quick question, I guess. Once you develop this cruise missile, hopefully you do, how would that be administered to an animal that was infected? Would it be through injection or orally, or do you have a choice? Do you have to, I mean, what would be the administrative process?
Yes, so instead of like taking 100 animals and then injecting them and then seeing if it worked, the approach would be a little different. They would be instead of rounding up the animals and injecting them, you would take a few animals and put the gene inside them, and then they are natural vectors wandering around on the landscapes. So it's a different approach, and I realize it might seem a little far-fetched, but one of the disadvantages of the vaccine approach is to vaccinate animals or people. You need to often one injection to raise the antibody. It doesn't work. You have to keep going back, and you'll be following the animals on the landscape there. So that's not perfect either. This would have the potential advantage that it's a sort of like a one-shot deal, that might be some special animals wandering around that can produce sexually transmitted prophylaxis. Yes, something like that. Yep. Yep.
Well, thank you, Dr. Westaway, for that extremely informative presentation. And thanks, actually, to all of today's presenters. That concludes today's sessions. We're going to adjourn momentarily. Before I let everybody go, most importantly, cocktail hour is at 5:30 in this room. I believe the cocktails are actually going to be out in the foyer tonight. And as far as I know, just a quick reminder about John Ward from Camillus Knives doing the knife sharpening demonstration and the Q&A at the booth right outside that door back there. Okay. Thanks for your attention, everyone.